Hello everyone. We would start within 2 minutes. Good afternoon, ma'am. Garima ma'am, Tabassum ma'am.
Good afternoon.
Can we start with the presentation?
Yes, please.
Respected judges, esteemed guests, and participants, a warm and hearty good afternoon to each one of you. I, Maithili Vyavhare, welcome you to postgraduate oral presentation of 4th Student Research Congress themed Innovation for Better Health. This event is organized by Sri Vileparle Kelvani Mandal's Dr. Bhanuben Nanavati College of Pharmacy, co-hosted by University of Mumbai and industry partner ACG Worldwide and SciTech Center. Now, as we embark on this intellectual journey, it's my honor to introduce our distinguished panel of judges for this session who bring with them a wealth of experience and expertise. Our first judge, Dr. Garima Garg ma'am. Ma'am served as a principal of Meerut Institute of Engineering and Technology. Ma'am has remarkable 22 years of experience in academy, research, industry, and administration. She has completed her graduation from Sardar Vallabh Patel University, Vidyanagar, Masters of Pharmacy in Pharmaceutics and Pharmaceutical Technology from LM College of Pharmacy, Gujarat University, and received a doctorate in pharmacy from Tirthankar Mahavir University. She was a recipient of Gates Scholarship with All India Rank 64 and received a gold medal for her PhD work. Ma'am is actively involved in academics, administration, organizing professional events, seminars, scientific workshops, and much more. She has participated in various national and international conferences and presented several papers in India. Ma'am has more than 40 publications in peer-reviewed international and national journals. Ma'am has 4 patents and 1 Indian patent has been granted. She is associated with several professional organizations like IPA and APTR, where she is Distinguished Life Member. Ma'am has guided around 28 MPharm students in pharmaceutical science. She is also trained in SAP under Train the Trainee program. We welcome you here, ma'am. Our second judge is distinguished personality in this field with 23 years of academic experience, Dr. Tabassum Khan ma'am. Ma'am holds HOD in pharmaceutical chemistry in SVKM's BNCT College. Ma'am has completed her graduation, master's in quality assurance and PhD in pharmaceutical chemistry from CU Shah College of Pharmacy, SNDT Women's University. She is featured as top 2% of scientists in the world. Ma'am has participated in various national and international conferences and presented several papers. She has 7 government-funded research grants. Ma'am has done more than 80 publications in peer-reviewed international and national journals. Ma'am has filed 3 patents published and 2 patents have been granted. Ma'am is associated with several professional organizations like IPA, SPER, FIP, and AAPS, where she is a distinguished life member. Ma'am has guided around 29 MPharm students and 5 PhD students. She has contributed in academics, research, industry, and various administrative sectors. We welcome you here, ma'am. As we progress through the presentation, I wish all the participants all the very best and may this platform serve as a catalyst for innovation that contribute to better health. Now, participants are requested to make note of guideline for postgraduate oral presentations. Presenters are requested to rename themselves as per allotted code numbers, if not already done. Presenters cannot reveal their name or any affiliation. Each presenter is requested to stay alert, switch on their cameras when their code is called out. Pre-recorded video will be presented on the screen. Only one author is allowed to answer for Q&A session. I kindly request judges to confirm that they have received the scorecard.
Yes, we have received.
I would like to thank Garima Madam. Let me take this opportunity to thank Dr. Garima for accepting our invitation and sparing your time with us. So thank you, Dr. Garima. I'm sure your inputs, valuable inputs will help students to further understand the scientific intellect of the program. As well as thank you, Dr. Tabassum, for accepting our invitation to be a judges. And all the student participants, remember all the rules and regulation which the team has shared with you. So thank you, Dr. Garima, as well as Dr. Tabassum. Thank you very much. Over to you, team.
Thank you, ma'am. Now I would request NPH SRC 001 to get ready. We are about to share your video shortly.
Good morning, all.
MPHSRC 001, are you in the meeting? Okay.
Today I am going to discuss on my research topic entitled as Red Rice Radiance, a novel red rice-based emulsion for skin protection and glow. The changing environmental conditions, lifestyle, eating habits, and stress has affected the individual's health. Further looking into the skin health, air pollution, UV radiation, and oxidative stress has an impact on the skin health, which has accelerated the skin aging process, hyperpigmentation, and even the skin cancer. Here you can see in the diagram, the UV radiation generates the ROS, that is reactive oxygen species, which causes the oxidative damage and activates some enzymes like metalloprotease enzyme, hyaluronate enzyme, and elastase enzyme, which causes the collagenase breakdown, decreases the hyaluronic acid level, and causes the elastic breakdown respectively, which results into the loss of flexibility and strength of the skin, which results into the formation. Also, due to oxidative damage, DNA mutation and lipid peroxidation takes place, which also causes the wrinkles. Synthetic products are the targeted ones and effective when used for a short period of time. However, regular application of the same may cause the skin damage as they mostly contains parabens. So we have selected the red rice as the red rice consists of the various phytoconstituents such as proanthocyanidins, which treat pigmentation. Gallic acid itself has the antioxidant properties. Anthocyanins has the antimicrobial properties. Due to this, red rice do not generally contains do not generally requires any antimicrobial agent for its preservation. Then the vanillic acid, it is also the antioxidant agent. Comeric acid, vitamin B3 smoothens the skin surface. Vitamin E moisturizes the skin surface. Hydrobenzoic acid such as ferulic acid, cynipic acid, and the gamma oronazole. We have developed an emulgel composed of the red rice extract blended with the almond oil working as a penetration enhancer and moisturizer. Red rice is a rich source of proanthocyanidins, as mentioned now, and hydrobenzoic acid derivatives like ferulic acid, cynipic acid, reported as a free radical scavengers. Whole red rice grains were extracted in the methanol water system 80 to 20 to obtain the polyphenolic constituents. 1 to 2% of the red rice extract formulated in the carbopol gel base and screened against the UV radiation induced skin damage. Here is the proof of concept for the lab scale prototype. Here is the formula of our emulsion. The ingredients are the red rice extract, which is the API, almond oil acts as an emollient and skin hydrating agent, rosemary oil acts as an antimicrobial, carbopol, gelling agent, glycerin, antimicrobial, emollient, and a co-solvent, and water as vehicle. The emulsion is tested for its sun protection activity, DPPHSA, and UV induced photo damage study. Here is the present state of our technical development. The emulsion was tested for these parameters such as homogeneity, pH, physical stability at room temperature, spreadability, and irritation test for the 45 days. And here we can see the emulsion is stable for the 45 days, and also again it is again till date it is stable, but the actual stability study is still pending. We also tested the total phenolic content and total flavonoid content of our emulsion. Total phenolic content was checked by the following CO method, and total flavonoid content by aluminum chloride colorimetric reagent. The total phenolic content, the values are 705, and the total flavonoid content values are 317.075. SPF, sun protection factor. Sun protection factor was measured by using the UV visible spectrophotometer, and it was calculated by the Mansus formula. The sun protection factor of the extract and 2% formulation, the value was between the 15 to 20, which shows the sun protecting ability of a gel against the UV radiation. The viscosity study is also conducted for the 45 days to test its shearing capacity. It shows the thixotropic property for the 45 days, and also after the 45 days also. Also, we have checked the oxidant activity and percentage of free radical scavenging activity of the gel by DPPH assay. Then a proof of concept for the preclinical screening. This is the simulation chamber in which we have placed animals for inducing the inflammation and irritation. There are first 6 groups. The first group was normal control. The group 2 was UV radiation plus heat exposure. Group 3 was UV radiation, heat exposure, and marketed formulation. We have used the Ayur herbal as the marketed formulation. Group 4 for the 1% herbal, group 5 that is 2%, and group 6 is only base. Among these 6 groups, I have shown the results of only 3 groups. The first one is the untreated group, the second one is the market standard, that is Ayur herbal sunscreen, and third is the red rice extract gel, 2%. Here you can see in the screening of formulation, uh, in the control group, normal in the control group, we have observed the normal epidermis and dermis of the animals. But in the untreated control, we can see the hyperpigmentation, hyperpigmentation and hyperkeratosis and inflammation on the skin surface of the animals. But in the marketed standards and 2% red rice extract gel, no inflammation and hyperkeratosis and parakeratosis was observed. This data is supported by the lipid peroxidation assay. Lipid peroxidation assay is performed to measure the percentage of the melanodialdehyde produced after the oxidative damage on the surface of any animal. Melanodialdehyde is produced after the oxidative damage. Here you can see in the standard, that is Ayur herbs, the p value is of 4 stars, and in the 2% formulation it is also the 4 stars. And in the disease control, that is in the untreated control group, the value is more, that is amount of melanodialdehyde produced is more. So we can say our gel has the sun protecting factor same as that of the marketed standard. Novelty and uniqueness of the product is that product, a carbopol-based gel, exhibited a significant effect against the UV-induced skin damage. Quantification methods of the gallic acid and routines are validated. Red rice extract is blended with the rosemary oil, which acts as a natural preservative. It is a paraben-free. Then it has the multi-targeted effects such as sunscreen, anti-wrinkle, anti-aging, and for the maintaining the skin tone. It is the viable product with respect to safety and cost. The antioxidant activity, that is IC50 value, was greater than 150 micrograms per ml for the 2% gel. Anticollagenous activity was 21%, and sunscreen effect, SPF factor, is 15%, is confirmed in the laboratory. Marketed applications. The formulation has versatile application as a skincare product. The product can further be optimized for therapeutic use as depigmentation agent and for skin diseases. Future perspectives. Red rice extract in combination with seed oil encapsulated in pectin extracted from the soil and gel with natural preservative can be done as novel formulation. There is a scope of combining the zinc oxide with the formulation, which will soothe the SPF factor. The waste generated from the red rice extraction can further be optimized to formulate the face scrub, hence waste will be valorized. IP strategy. The provisional patent has been granted for this project. This is the provisional patent number, and this is the date for the grant. These are the references, and thank you.
Thank you for the presentation. Can I ask some questions, please?
Yeah, ma'am, sure.
Yes, ma'am.
Okay. One thing I wanted to know that you have done the sunscreen, sorry, SPF value with UV. Okay.
Yes.
So,
Yes, ma'am.
have you tried using determining the UVA and UVB? What is the SPF value, UVA and UVB?
Ma'am, it is 15.
Okay, SPF value is 15. But have you tried the
Yes.
UVA radiations and UVB radiation? There are two rays which are considered.
Yes, ma'am. We have not tried it separately, but we will do that.
Yeah, it is possible with the UV. There is a method for that. Okay, this my suggestion. Another thing I wanted to ask Because right now, most of us are using the mobiles and computers and laptops, etc. So there is
Yes.
a blue light concept is the exposure to the blue light rays is very high. Okay, more
Mhm.
than the sun, we are exposed to the blue rays. So,
Yes, yes.
have you taken in because you have already applied for the patent, but have you tried? You can try. What is my suggestion is, have you
Yes,
seen
ma'am.
whether anything which are using can be used for reduce the damage by the blue radiation?
Yes, ma'am. Yes.
Have you gone through some
Uh,
literature regarding the same?
no, ma'am. I haven't gone, but we will
Okay.
see for it.
Yes, it's because as your product, and another thing I wanted to know whether you have tried with the only almond single ingredient, like you have used rice, red rice
Red
extract.
rice.
Okay,
Yes.
what if the red rice is not there? Only almond oil and rosemary oil is there. Is there is a significant difference in the you can say the SPF value and antioxidant effect?
Uh, Ma'am, the antioxidant effect is more of the red rice extract as it contains the phytoconstituents like I have shown, gallic acid, vanillic acid, and hydrobenzoic acid. So about the
Okay.
antioxidant activity is mainly because of the red rice extract, and the almond oil is acts as a penetration enhancer and moisturizer, while the rosemary oil acts as an antimicrobial agent. So that we have not used any parabens in our formulation. Uh, that's it.
Uh, okay. Mm, thank you.
Yes,
It's
ma'am.
a very good presentation, and it's a good thing you have applied for the patent also. Okay. So
Yes,
it's
ma'am.
a nice work because a lot of work is going on in this area. It's a novel product. So hope you all the best for your work.
Thank you. Thank
Okay.
you, ma'am.
And I would like to add one suggestion. You please study the same formulation with blue radiation also. Try because it is easy to replicate. Okay?
Yes, ma'am.
Okay. Thank
Yes.
you very much. Thank you very much. So nice presentation.
Thank you, ma'am. Thank you
I
so
have
much.
questions. One is, how are you going to standardize your red rice extract? What is your standardization parameter?
Ma'am, we have standardized our red rice extract by HPTLC method. We have calculated the bound phenolics and unbound phenolics by that, and total flavonoid content, total phenolic content, gallic acid and rutin trihydride present in the red rice is
No, no.
also.
Suppose I take a red rice from a different location. So am I going? Are you planning to standardize it with TPC and TFC or
Yeah,
anthocyanidins, which is the active? Have you done that? Have you
we will.
standardized the extract with respect to something, or you have used the extract whatever you have got?
Yes, ma'am. We have standardized it by TPC, TFC, and also by HPTLC method. We have quantified the gallic acid and rutin trihydride present in the extract.
नहीं, but gallic acid is something which is present in 100 plants उठाओगे उसमें 75 plants में gallic acid होगा. So
Yes,
is that the correct standard? So look back into your project, okay? Because gallic acid is
okay,
present
ma'am.
in many plants. That's one. Second,
Yes,
can
ma'am.
you go to the you've not numbered your slide. There is something where you have reflected DPPH acid. Can you go to that slide?
I don't.
Ma'am, please repeat which slide.
Yeah, this one only. This one. Okay, so over here, when you say, is this the red rice grain extract or is this the formulation?
Ma'am, it is the red rice grain extract.
So the response of red rice grain extract and flour is the same, if you see.
Hmm. Yes, ma'am.
So why did you prepare the extract at all?
What, ma'am?
Why did you prepare the extract then? You put the extract in your formulation, right?
Yes, ma'am.
So when the response was the same, what was the reason of you adding extract? We could have just used flour, right? Because it's the same
Uh,
concentration.
yes, ma'am. The blending of the oils with the extract was uniform, so we have used the extract.
What was the nature of the extract? Powder tha, sticky tha, what was it?
Ma'am, dried powder. We have dried the extract.
Okay, and what standard did you use in DPPH assay? Because I don't see the graph for standard. Okay, can you go to the next slide? So in TFC and TPC you've used a standard, right? Not this one, sorry. In TPC and TFC you've used a standard.
Gallic acid and butane trihydride.
Right. So do you think you needed to use those standards in your extracts and almond oil and flour? Think about it. Nice project, but good.
Yes, ma'am.
Thank you.
Thank you, ma'am.
Thank you. We will move on with MPH SRC 002.
Hello everyone. My topic of presentation is formulation, development and evaluation of probiotic tablets. These are the content of my presentation: background and rationale, introduction, method, result, and conclusion. In background, importance of probiotics. Why probiotics are important in our health? Because probiotic gain the potential health benefits, and they increase or improve our gut health, immune function, and overall well-being. And scientific research supports the role of probiotics in preventing and managing various health conditions. Then dosage form. So, probiotics available in liquid form as well as solid powder form. And the advantages of tablet form is because of ease of handling, precise dosing, and consumer familiarity. And probiotics in more suitable and stable form as the tablets. Additionally, stability and validity, dosage accuracy, consumer compliance, and market demand. Probiotics are sensitive to environmental conditions like heat, moisture, and oxygen. When formulating a tablet allows for better protection and preservation of the probiotic strains. The tablet formulation provides a controlled release mechanism for survival of probiotics through the digestive system. Then dosage accuracy. Tablets allow for accurate dosing of probiotic strain for the ensuring the consumer receive the intended amount of each administration. Then consumer compliance. Tablets are convenient for consumers and the market demand. Probiotic supplements in tablet form is significant in the market. Introduction. Probiotic strain. Probiotic strain promote our health balance of gut microbiota, supporting various aspects of health. And common uses of strain is they improve our digestion, our GI issues, and enhance immune functions. And they also improve the GI microflora and managing conditions like irritable bowel syndrome. Then different strains have different specific effects on their health balance. I use the Lactobacillus sporogenes or Bacillus coagulans is commonly used the probiotic for the digestive health. It maintains the balance the gut microbiota. It improves gut function and issues like bloating or irregular bowel movements. And Lactobacillus sporogenes combined with antibiotics and antidiarrheal. Then methods. I use a tablet compression method in well set condition and control with respect to heat and moisture, and follow the procedure of tablet compression. And I evaluated the pre-formulation study of probiotic powder as well as tablet mixture. Probiotic powder flow properties are bulk density, tap density, Car's index. Also, the tablet mixture flow properties, bulk density, tap density, and Car's index. And this tablet mixture used for the compression process. These are the tablet composition. In that probiotic powder, I use the probiotic powder, lactose, HPMC, magnesium stearate, sodium alginate, and talc. I use the probiotic powder taken 100 mg, lactose 100 mg, HPMC 82 mg, magnesium stearate 3 mg, sodium alginate 9 mg, and talc is 6 mg. This quantity are taken for only one tablet. Tablet is a weight of 300 mg. Then evaluation parameters of tablet. Bulk density, tap density, Car's index, then hardness, then thickness, diameter, weight variation, friability, and the cell viability count. Hardness determined using Monsanto hardness tester. Weight variation is performed as per procedure described in Indian Pharmacopoeia, and the friability is determined or calculated by using their formula, that is percent friability is equal to loss in weight divided by initial weight into 100. Then the cell viability count is cell viability count tested using the standard plate count method. Standard plate count method was performed after making the serial dilutions of the revived bacterial cultures. And the results, the hardness observed the 8.1 plus minus 0.081 kg per centimeter square. In thickness 3.3 plus minus 0.081 millimeter. Diameter 8.82 plus minus 0.021 millimeter. Then weight variation is 301 plus minus 0.95 mg, and the friability of 0.52%. All the results are comply to their standard results. And this is taken for the average. Observations are taken from the n is equal to 3 average ratio. Then cell viability count in different medium. Dissolution medium, I use the in acid medium and buffer medium. Acid medium, buffer medium, and water. Acid stage is 0.1 normal HCl. Their cell viability count is 5.9 into 10 to 2 CFU per ml. And then buffer medium 6.8 pH, 4.2 into 10 to 2 CFU per ml cell viability count. And in water, cell viability count is 3.6 into 10 to 2 CFU per ml. And these tablets are taken for stability study, and is subjected to accelerated stability study condition as per ICH guidelines for duration of 180 days. And the process is ongoing. Then the conclusion. The result of my experiment work is formulating probiotic matrix tablet. It shows the good flow properties of probiotic powder and tablet mixtures. Was found to be in acceptable limits, and the cell viability count, hardness, thickness, disintegration test, and weight variation was performed and found to be compliant with the standards. And the HPMC and sodium alginate combination, they increase the acid tolerance of probiotic lactobacillus strains, and results encouraging as well as promising the possibility of formulation development of probiotic tablet with stable and effective application in antibiotic and antidiarrheal drug therapy. We use the probiotic because of the antibiotic medicine. They affect our GI microflora. So probiotic it improve the GI microflora. So we use probiotic with antibiotic and antidiarrheal drug therapy. So thank you.
Okay. One thing, yes. Ma'am, are you going to ask the question or should I? Tabassum, ma'am, Dr. Tabassum?
Madam, you will be first guest.
Okay, thank you. One thing I wanted to know, the probiotic you used, have you purchased it from somewhere or you have generated it yourself?
No, ma'am. I have purchased from market.
You have just from the market you have purchased the lactobacillus powder.
Yes, ma'am.
Okay. So how you have ensured there is no cross contamination?
No, ma'am.
Because when you are purchasing, when you are using in the formulation, while you are processing it, there might be cross contamination with secondary bacteria. Have you checked it?
No, ma'am.
And can you name any product in the market which is containing the lactobacillus? Have you studied the
Yes,
market?
ma'am.
What is available?
Sporlac powder.
Rolac powder. And is it present in combination? Is it present in the tablet form in any formulation?
No, ma'am. It is in a powder form.
Okay. So have you searched why they have not gone? Why the powders are better and why the tablets are not available? Because I think lactobacillus is also available in combination with the vitamins. Visilac, if you remember, there's a market product. Have you gone through what is the rationale? Why you want to have the probiotics in tablet form?
Because of patient complaints.
Patient compliance only. Okay. Another thing, question is, do you know what is symbiotics? Symbiotics. What is symbiotics? Probiotics you have said. What is symbiotics?
Probiotic and prebiotics. Yes, ma'am. Probiotic and prebiotic combination.
A combination of prebiotics and probiotics could have worked better.
Yes, ma'am.
So I will be very honest with you. How much work actually? How many days you have developed this formulation?
1 month.
1 month. Okay. Because it's a very short duration. I think you have just
Yes.
a lot of actually probiotics is not something you can play with uh, very easily. Okay,
Yes,
there are a
ma'am.
lot of factors associated with probiotics. One has to be very careful because these bacterias can, whenever you are they get the good environment, there is definitely even the curd you are having at home is having the secondary bacteria infection. So there is
Yes,
always
ma'am.
chances of contamination. Okay. Second thing is I find the studies, if it is in 1 month, you still need to improve upon.
Yes,
Okay,
ma'am.
especially probiotic related studies. I think microbial studies, cross contamination studies. Okay, all these studies need to be done
Yes,
more
ma'am.
thoroughly. And okay, it's a good idea, but it needs to be worked upon. Okay, lot of
Yes, ma'am.
background literature is needed. This is very raw. I find it very raw. I don't know. At M-Farm level, I still find it very raw. Okay. Thank you very much. It's a good idea, but just work upon it. Okay. Look for other alternative dosage form also. Like if you are talking about patient compliance, I think nowadays oral thin films is another area where you can look. Okay.
Okay, ma'am.
So, thank you very much for the presentation. It's a good idea, but I think it
Thank
still needs to be updated. Okay? Thank
Yes,
you very
ma'am.
much.
Thank you.
Okay, I think in continuation with what Garima madam said, there's a lot of work to be done in this project. Again, how did you select HPMC and sodium alginate? Have you done some kind of QPD approach in selection of excipients and optimizing their percentages, or you just randomly selected HPMC and sodium alginate? What is the basis for you to select these two polymers?
As per standard formulation, ma'am.
Which standard? From where did you get the standard formulation? Is it there in the pharmacopoeia?
आज दिन हमारा एक को नहीं थे
Is it there in pharmacopoeia?
आवाज ही नहीं
And how are you going to ascertain the purity of this microbial or lactobacilli strain? And how are you going to prove the last bullet of your conclusion that it is providing good antidiarrheal therapy? Have you done some proof of concept studies? Whether they are able to hear because there is no response.
MPH 002, are you there? Or shall we proceed? Because time is already done. We have 2 minutes for
Proceed.
Q&A.
We proceed.
MPH SRC 003, please keep your camera on.
Greetings to all. The topic for my presentation is Design, Synthesis and Biological Evaluation of PROTAC for its anti-cancer activity. Starting with the introduction, prostate cancer is the second most commonly diagnosed cancer in men after lung cancer. Prostate cancer primarily affects older men. The risk of developing prostate cancer increases with age. Majority of cases are diagnosed in men over 65 years old. The overexpression of androgen receptor plays a vital role in castration-resistant prostate cancer. Conventional therapy includes drugs which do not have potency to target undruggable proteins. They also show drug resistance problems. To overcome this, new targeted drug concept was developed, that is PROTAC, proteolysis targeting chimeras. The PROTAC includes heterobifunctional compounds having ligand for target protein and E3 ligase recognition moiety, both of which are connected by a linker. The rationale behind this work is enzalutamide, which is a second-line androgen receptor inhibitor, shows drug resistance due to mutation. To overcome this, PROTAC is designed. PROTAC is a complex which helps in degradation of undruggable protein through ubiquitination reaction. The major significance is that PROTAC do not need any particular binding site on protein, and it gets attached to anywhere on protein surface. PROTAC particularly binds to cancerous cells, which are overexpressive in nature. Hence, cancerous proteins will be degraded by PROTAC selectively, which in turn helps in treatment management of prostate cancer. The mechanism of action is as the PROTAC having target of interest binding domain and E3 ligase binding domain attaches itself to the target and E3 ligase simultaneously, bringing both in close proximity. E3 ligase then tag the target with the ubiquitin. The ubiquitin target is further subjected to degradation. After degradation, the free ubiquitin is further utilized for the next reaction. The experimental work is divided in 4 steps. First, drug design study, synthesis, characterization, and anti-cancer activity. The drug design study includes molecular docking and dynamics with the protein code KHS, and 7-KHS. The synthesis is carried out with the amidation reaction. The synthesized compound is characterized by TLC, IR, NMR, and mass. The anti-cancer activity is conducted for MCF7, LN-CAP, PC3, and DU145 cell lines. The PROTAC is complex of enzalutamide, which is AR receptor inhibitor, thalidomide, which is E3 ligase ligand, and NBOC 16-hexane diamine, which is a linker. The synthetic scheme is as shown as follows. The enzalutamide is converted to its acidic form with the acidification reaction, and the linker is attached, followed by the thalidomide, which is E3 ligase ligand attachment. The structure-based drug likeness property and ADME properties were calculated for PROTAC within reference to enzalutamide. The complex follows all the rules of Lipinski with some exceptions. However, these exceptions do not affect the bioavailability of PROTAC. These exceptions are due to the bulkier nature of PROTAC. The result and discussion for the studies conducted are as follows. Molecular docking study shows the binding score of -10.65 with protein code of 7-KHL, and with 7-KHS it is -12.1511. The molecular dynamics shows the stable conformation with the RMSD value of 1 to 3.5 angstrom. The characterization of designed PROTAC is done with the melting point, which shows PROTAC has 242 degrees Celsius melting point. The TLC shows that RF value is 0.8. The IR spectroscopy is done to elucidate the functional group, which majorly focuses on amide group, which shows the synthesis of PROTAC. Further characterization includes NMR spectroscopy for the structural elucidation. The splitting of peaks signifies the structure of PROTAC. The mass spectrometry confirms the molecular weight with the peak at 823. The anti-cancer activity is conducted with SRB assay having adriamycin as a positive. The nicotinamide assay is conducted for PC3 and DU145 cell lines, which are specific cell lines for prostate cancer. The results are as from SRB studies, it is found that PROTAC shows good anti-cancer activity with NCF cell lines, and GI50 was found to be below 10. Anti-cancer activity for PC3 and DU145 were carried with WST1 proliferation assay, which shows minimum inhibitory concentration is below 10 for DU145 and 2.19 for PC3 cell lines. The conclusion is as follows. Designed PROTAC may have potential to overcome drug resistance and have potential to degrade undruggable proteins. Molecular docking of PROTAC reveal excellent binding affinity with PDB code 7-KHL having dock score -10.65. Molecular dynamics study shows PROTAC complex is stable with PDB ID 7-KHL. ADME study shows that PROTAC follows Lipinski rule with some exceptions, however, which do not affect the bioavailability. Complex synthesized by amidation reaction confirms the characterization studies using melting point, TLC, IR, NMR, and mass. Design complex also shows good anti-cancer activity with MCF cell lines and PC3 and DU145 cell lines. The concept can be explored for other PROTAC drugs and can be evaluated further for its biological activity such as anti-cancer, anti-tubercular, and antiviral. These are the references, and thank you.
Garima, over to you.
Ma'am, please you take the lead.
Done. Okay. So can you just roll your conclusion slide? So if you see in your first bullet of the conclusion, you say that overcome drug resistance. Did you check the compounds that you synthesized in resistant cell lines?
No, ma'am, not yet. We have just checked it on prostate cancer cell lines, but the complex is event driven, so that's why it will be overcoming the resistance. That's our rationale.
So which are the resistant cell lines that you propose? So MCF7 is which
LN-CAP
cancer
is
cell line? MCF7 is which cancer cell line?
no, it is And MCF7 is used for it is a general cancer cell line. It is used for
No,
multiple
it is not a
cancer
general
cell
cancer
lines.
cell line.
We have also, it is
MCF7
for breast cancer also
it is a
and
breast cancer.
prostate. Uh, and the specific we have used it is PC3 and DE145 cell lines.
You've used that. And how are you going to
Yes.
overcome these Lipinski rule violations?
Actually, ma'am, we are also right now we are uh, targeting to synthesize small drug protac, which will be following the Lipinski rule. And also from the literature, we have noticed that molecular weight 900 to 1000 it is bioavailable for protac.
Can
So
you
there
go
are evidences.
can you go to the spectral data? The slide of the spectral data? Spectra, not this. Okay, can you tell me how did you do the MD studies?
Actually, we have not done it. We have it is like a third party. We uh, gave it for simulation studies to some other party because we didn't have that access at our end.
Okay, and what kind of after the TLC study, what kind of how does the LC? Did you do a purity study of this? This is TLC, right? Of the
Yeah,
synthesized
yes.
protac, did you do a HPLC study, peak purity study?
No, we haven't done.
Because it is very closely eluting. So if you see your reactant
Yeah.
and product, they are very
Hmm.
close to each other. So with this TLC, we cannot really say that it is pure. Can you go to the NMR? Mass and NMR data. Okay, so can you just explain this NMR?
Yeah, I can.
Don't read from the
See,
table of the spectra.
no. Actually, there is a peak which is the bond formation of clinker and thalidomide, which is not present in both our drugs and thalidomide. So that's one peak which confirms the structure, and it is like 1 to dioxypiperidine structure. It is
So
there
is the total
in
number of
NMR. It is.
protons, is the total number of signals matching with the total number of protons? Because मेरे को ऐसे दिख नहीं रहे थे.
I have to look into it.
And can you explain this doublet 7.63, 8.89 benzene ka? Which doublet is this? There are two aromatic rings. Two, no, in fact there are three aromatic rings. 1, 2 and 3. So what doublet is this?
Yeah. Actually, I can't say.
Huh?
Uh, Actually, I'm not able to see the graph properly, so I can't be able to
No. I'm just asking aromatic doublet, aromatic CH, hai na? Either 7.63 to 8.89.
Yeah.
When you say it's a doublet, you've got three aromatic rings. So can you just explain this splitting of doublet? Just have a relook at your spectral data and your characterization so that you can allocate the peaks properly. Okay?
Okay.
Okay. Okay, that's all from my side.
Just one question. What is the RMSD value for protein and ligand?
Like
RMSD
separately,
value?
ma'am, do you have to tell the separate RMSD value
Go to the next
of
slide.
the
The information is there in your MMG, this calculation. Go to that slide. Next, I think. Where you have shown the in silico, उसमें होगा.
Like the first present slide, it is backward.
No,
हाँ ये वाला. Yeah.
yes. RMSD.
Huh.
Okay.
For complex, it is input 3.5, and for protein and ligand, we have calculated it separately.
Okay. No, for the complex ligand, and this is 1 to 1 3.5.
Hmm. Actually, it is for both. That they have done for the 7 kHz and 7 kHl. With 7 kHl, it was 3.5, and for 7 kHz, it was 1.2.
It was around your value was, it was 3.5 or less than 3.5. Hello,
Actually, it was
your
just
RM is
3.5.
3.5. Bit of a good correlation. It should be, I think, less than 3.
Okay.
So you just check it. I am not a chemistry person,
No, no,
so you can
it
check
has to
with
be,
your
it
math.
has to be less, but I think they have outsourced it. They have got it done. So you need
Okay.
to just reconfirm with them.
Okay.
Okay, you just need to.
Actually,
Okay.
these values are for both the 7 kHz and 7 kHl proteins. That's why it have written it in range.
Okay. वो एक्चुअली आपकी ग्राफ से भी वही इंडिकेट हो रहा है. थोड़ा सा उसको रिविजिट करने की जरूरत है आपको, मेरे
Okay.
ख्याल से.
Yes.
Okay.
Okay.
As such, it is a very good work. You have done a lot of work, and I really appreciate that you are doing this kind of various studies and going through these work. It's a very good work. Okay. Thank you.
Okay.
Thank you. Judges, please make note. We will start with MPH SRC 006 now.
Hello everyone, this is ID number MPHSRC006. I'm here for the competition category under oral presentation in PG group. Here is my title is Use of in silico and green chemistry approach for designing and synthesis of novel pyranoparazole and pyranoparazole pyrimidine derivatives as promising antifungal agents under the fourth student registration congress. So first part of my presentation is introduction and problem area. So introduction is divided under the two parts, that is why fungal and conventional problems in the drug discovery. So why fungal is nothing but the increase in the resistance of antifungal drug discovery in worldwide day by day. There is a slower drug development, lower bioavailability and increased toxicity world. Conventional problem in the drug discovery are nothing but lengthy process, can take up to 15 years, use of more harmful chemicals, average cost that is high in the cost for chemicals and synthesis, and more synthetic step lead to the increase in the pollution. From this problem, I came across for the solution is showing with the presence of molecular hybridization. Molecular hybridization is used in the two types, that is in silico studies and green chemistry studies. In the in silico studies, ADMET and molecular docking is used. ADMET is nothing but adsorption, distribution, metabolism, toxicity study. And in the green chemistry, I used microwave assisted synthesis. These both type of synthesis or studies are showing less use of hazardous chemicals, pollution prevention, and time consumption. From this, I go through the literature survey for the pyranoparazole pyrimidine is showing better antifungal activity. In the substitution on the pyran ring, electron withdrawing cyanide group at the C3 position of a pyran ring improved antifungal activity. At the C2, NH2 shows increase in the activity, while NO2 linked via hetero ring is showing decrease in the activity. At the C4 position, aromatic ring increase in the activity, and OH group is also moderate the activity and effect on the MIC value. Ring fusion involves pyran pyrimidine. Pyranthiophane gives antifungal activity as compared to pyran and pyrrole fusion. Linked with the numerous heterocycle where triazole have shows increase in the toxicity rate, thus linked with the pyrazole thiazole preferred. I came across that when the pyridine and methylbensulfonamide is attached to the pyridine, it is showing the higher antifungal activity. I came across further two types of synthesis schemes in which pyridine is attached to the pyrimidine and methylbensulfonamide. From this literature survey, I came for the aim and objective, that is, aim of my study is to design and synthesis of novel pyranoparazole pyrimidine derivatives as promising antifungal agents. Key objective of my studies are literature survey, in silico studies, that is adamant docking, and synthesis of pyranoparazole and pyranoparazole pyrimidine NCs using green chemistry, and for the last antifungal activity. So from this literature survey, I came across for the designing libraries of NCs. NCs is nothing but new chemical entities. From the literature survey, I came across for the three types of series in which A1 to A16, B1 to B12, C1 to C11, these are my optimized pharmacophore. In the given table, I have given that what type of R1 group is attached to the given ring which I design in the NCs. These are my optimized pharmacophore and my design libraries. On these design libraries, I have come for the ADMET studies. ADMET studies are nothing but adsorption, distribution, metabolism, excretion, and toxicity study. This study I performed on the Schrodinger software by quick crop method. Lipinski rule of phi is showing the ADMET prediction. These are the ADMET result in which all compounds obey Lipinski rule of phi with the zero violation in my studies. After that result, I have gone for the further study on these all the compounds. The further studies are carried out, that is docking study. In silico method in docking study is showing protein and ligand interaction, from which I have came for the better interaction and better result from the PDBs. I have used three types of PDBs in my docking. These are the docking method, that is from the Schrodinger protein preparation and glide docking. These are the docking results in which my all compounds are compared with the standard drug, and docking score and gliding energy are compared with the docking elements. Further the docking score and docking energy, I have come for the synthesis studies. Synthesis having this three steps, that is scheme A, scheme B, and scheme C. So in the synthesis scheme series A, I involve that ethyl acetate, malonyl hydrogen hydrate, and aromatic aldehyde addition in which L-proline in 10 minutes of grinding. I found that A1 to A12 series, which is product called as dihydropyranopyrazole. In the series B, when the dihydropyranopyrazole is added with the toluene sulfonyl chloride with the presence of benzene and pyridine, I got the series B1 to B12. And for the last series, that is series C, I have used dihydropyranopyrazole with the addition of formic acid in the microwave condition for the 5 minutes. I have got the series C1 to 11 for the last product. I have used microwave here for the green chemistry. From this, the last is biological evaluation I gone through. This is nothing but antifungal activity. Antifungal evaluation of this synthesis NCs was carried out on the two fungal strains, that is Candida albicans and Aspergillus niger, by the agar-well diffusion method. Zone of inhibition and inhibitory concentration of NCs were determined by the standard drug. Biological activity and best MIC value are shown by the B1 and B3 and C3 and C4 compound with the same fungal strains. These are the biological evaluation and biological result with standard comparison. In the conclusion, I want to say that in silico studies and green chemistry are the best method in the preparation or in the paper of a research. In the in silico studies, NCs are developed by the in silico method, that is adamant and molecular docking. This can help to reduce the time and use of harmful chemicals that objectify towards the technical use in the development of NCs. In the green chemistry, synthesis of NCs were performed by green chemistry method, that is microirradiation, which can help in the atom economy, decreasing the ratio of waste material, time consumption, less hazardous, and pollution prevention method that are helpful for the environmental balance and safety. This research paper comes under the journal Recent Advance in Anti-Infective Drug Discovery under the publisher Bentham Publication. These are the conclusion. And these are the key references which I used in my research paper. Thank you.
Okay, so good amount of work. Did you do the docking work yourself?
Yes, ma'am.
Okay, so you have subscription of Schrodinger software, right?
Yes, ma'am. I have subscription. We have subscription of Schrodinger software, and on that software, I have used to do that docking and admin studies.
Admin studies. Okay. Can you go back to your evaluation data, your antifungal data? If you have a look at this data, now your best compound over here is B3, right?
Yes, ma'am.
Okay, now if you compare the same study, B3, and you see the MIC, there is a huge difference between this data. This is almost comparable, correct? Any reason why this must be happening?
No, ma'am, actually, I don't have a proper reason what is behind this. From talking about study, I have gone through this synthesis for these specific compounds. So I don't have specific reason for this.
Okay. Any reason why you selected three standards for this assay?
Yes, ma'am. In my reaction, I am having three types of rings. That is pyran ring, pyrazole ring, and pyrimidine ring. So that is why I have used the three types of PDBs in which these three rings are involved. So that's why I'm
How
using.
does it compare with amphotericin B?
What do you mean, ma'am? Sorry.
How does your series compare with amphotericin B? You have used scaffolds from here, these parts, but if you see the data, it is more comparable to amphotericin B, your results.
Yes, ma'am. Actually, by the MOA, ma'am, because mechanism of action is quite similar. That is for the antifungal or for the amphotericin B, ma'am.
Okay. Can you go to your synthesis slide?
Yes.
You have not mentioned your characterization in your presentation, right? So we assume that you have done that.
Yes, ma'am. We have done that.
What characterization studies you have done?
IR studies and NMR studies we have done in our college only from the another department.
Okay, and have you also done HPLC peak purity studies of your synthesized compounds?
No, ma'am. No, ma'am. PLC, IR and NMR studies I have gone through.
So what was the purity of your compound? Any idea? 75%, 90%? What purity did you obtain? Whatever series you have synthesized, what was the purity of those compounds?
Purity of compound. Yes, ma'am.
Okay. Maybe it will be a good idea to do a LC purity, because when you're doing NMR, your NMR compounds, when you send, they have to have a certain purity. You cannot send 50% pure compound, 70% pure compound.
Yes, ma'am. It is 70% pure compound.
How did you ascertain that it is 70% pure?
Compared with the results, ma'am, that what type of groups are present in the NMR.
But how did you ascertain 70% purity?
By TLC, ma'am, actually.
Okay, now in your this step, your pyrazole and sulfonyl chloride, this is at what temperature it is just mixing? Benzene, pyridine, do you have something over here? Are these the complete reaction conditions for your B series?
Yes, ma'am. Actually, for the first step, we are use L-proline and first compound 80.
No, so is there any temperature involved in this? Are they at room temperature? Are they at 30 degrees? Are they under air conditioned? What is it?
No, ma'am. Actually, I have used the water bath for this reaction. So
which reaction?
I'm making. No, first A series. A series. I have used the water bath. So that temperature I have measured for the 80 degrees Celsius.
So bottom line, when we represent schemes, they have to represent the synthetic conditions, right? So yes. I should know whether you're doing it under reflux, under heating, room temperature. So just make that change in wherever you're presenting it.
Yes,
okay.
Go back to
rationale. Go behind. the next slide. The next slide. Next one, next one. This, not this one. The next one. Where you have that table of your series. That one. This one, this one. Yeah. Hold on. How did you select these 12 compounds in a particular series? Like in series 1A, you have 16. Then you have 1 to 12. So what was the basis?
Yes, actually by various research paper, I have come across that A1 to A16 are the basic compound or the basic entity that is synthesizing various papers. But the difference between the aldehydes, so I came across through various research paper, and I get that what type of aldehydes are what type of substitution is having gives best results. So by this,
your fourth column is R1, right?
Yes.
Because I cannot see. I can see a photo over there. That is why. Your fourth column
that is R1. What, ma'am?
Your fourth column in this table, I'm not able to see. That is R1.
R1. Yes, ma'am.
So if you see, most of your compounds are having what type of functionalities? Electron withdrawing, donating.
Actually, ma'am, both types of electron withdrawing is also there and donating is also there. Because in various research papers I have gone through, I got that donating groups are also there and withdrawing groups are also there, which are having the higher activity.
So what do you conclude based on the activity of these compounds for R1?
By the synthesis, ma'am, actually, B1, B2, B3, B9, these are P series.
What is the conclusion? R1 electron withdrawing is good or electron donating is good? You've done the antifungal assay for this. So
donating
the results.
Donating electron withdrawing are showing more better results.
Okay, that's all from my side.
Thank you, ma'am.
Okay, just 2 questions from my side. One thing, what is your series 1? Can you come to the series 1? Series A reaction. Okay, is it a name reaction? Can you give me a name reaction? It is related to some name reaction. Is it a name reaction? Can you yeah, is it a name reaction? Your series A? Okay, and another thing is in your series A there is cyanide group, CN group.
Yes, ma'am.
How you are confirming that group? CN is present or not? How you are confirming? Have you done any study?
TLC studies I go for, ma'am. Actually.
Okay. You have not done any. CN group. How the CN cyanide group is there? How you are confirming that it is at that position?
Actually, ma'am, what I did for this TLC studies I have gone through, in the TLC, the various spots I have given. So in the I have gone through the comparison study. So first of all, I give for both reactant dots and then the product dot. So I have came across for that malon nitride, CN group is also present in the product spot.
Okay. Are you going in for the NMR also? IR group. Sorry, IR. Have you done the IR? IR I think must be available at your institute.
Yes, ma'am.
She has done NMR also. She's not presented it here. That's what she said.
Okay.
Actually,
IR, how you identify? How you identify CN group from IR? What is the characteristic?
Sorry, ma'am, I can't get you actually.
Madam has
Actually,
answered
what
the
is the
question she asked you.
correcting range?
Actually, ma'am,
Characteristic range for CN.
2300. So for the CN range is about 20 to 50. I mean,
Okay.
2000 to
Okay.
2000 to 52. I mean, by the ranges only.
Okay. Another thing is, Vita, you are using benzene. In your B series, you are using benzene. It is a highly carcinogenic solvent, I think. You may know benzene is a highly carcinogenic. So how can you say it is supporting green chemistry?
Actually, in the green chemistry, ma'am, I have presented the microwave condition. I mean, by on
Okay.
that basis, I was saying that the green chemistry word for the
But
third step.
still, a benzene in the use still. And how about the residue? Any residual is there? Residue solvent is there when you are using benzene? How you are checking it? When you are synthesizing the compound, is there any residual solvent remaining?
Uh, no, ma'am, actually, not really.
Okay, we just
Consider
confirm it
compound.
better, yeah, because it's
Yes,
okay. Thank you
yes,
very
ma'am. Yes,
much.
sure.
Thank you.
Thank you so much, ma'am.
Thank you. Now we would proceed with MPH SRC 004.
Greetings to everyone. I'm presenting research on mesoporous drug delivery of celecoxib, a promising approach for the treatment of arthritis. So, celecoxib is a choice of drug for treatment of arthritis. It is COX2 inhibitor and belongs to BCS class 2, which is having low solubility and high permeability, which leads to poor bioavailability. So, to increase its solubility and dissolution rate, we are using mesoporous silica, which is having the chemical stability and surface functionality. They are having the tunnable pore size and pore volume, and they are biocompatible. So aim of this studies and objectives are to formulate mesoporous drug delivery of celecoxib to improve its dissolution, solubility, reduce its adverse reactions and administration frequency, in vivo assessment for the enhancement of anti-arthritic activity. So the formulations were prepared by solvent evaporation method. 3 different grades of mesoporous silicas were used with 3 different ratios of formulation were prepared. Here the estimation of drug loading was done using this formula, and here out of 9 formulation, the highest drug loading was seen in mucilin and 244 FP, 1:2.5 ratio. In vitro dissolution process was used by FDA recommended dissolution method. The USP apparatus type 1 basket method was used. Tribasic sodium phosphate buffer was used, and sample quantity was used as 100 mg equivalent of pure drug. So analysis was carried out by UV at 250 nanometer. So in vitro drug release, here we can see the 3 different formulation graphs. So 1:2.5 ratio is showing the highest drug release at 60 minutes. If we compare the 3 different silicas, the maximum release was seen in mucilin greater than equal to Salad 244 FP, followed by HDP and pure drug. Moving further to choose the best formulation, Ben-Ferrani multiple comparison test was used, and it revealed that formulation mucilin 244 FP 1:2.5 ratios are showing significantly better results compared to the all 9 formulations. So further moving for BET analysis for drug loading, here we can see that 244 FP is having highest drug loading. So it's showing complete drug loading in the silica, and here mucilin is showing comparatively less drug loading. So here we can say that the formulations are showing significant increase in surface area, and which attribute to higher solubility and drug release. So moving further for solid state characterization and particle size estimation, compatibility studies by FTIR was carried out for pure drug and formulation, where all the major functional groups from the pure drug was shown in the formulation. So here we can see that there is no interaction between drug and carriers. So all the functional groups were well preserved. Particle size was found in nanometer range, and PDI value was less than 1. For the PXRD analysis, pure drug is showing highest intensity peaks, whereas Nucilin is showing less intensity peaks compared to pure drug. And in 244-FP, we can see that intensity of the peaks are drastically decreased. For DSC analysis, we can see the sharp endothermic peak at melting point, whereas formulations are showing decrease in the endothermic peak. So by PXRD and DSC, we can say that the crystalline nature of the drug has changed to amorphous nature, which is the reason for the solubility enhancement and dissolution rate. For same analysis, we can see here pure drug is in crystalline nature, which shows rod shape. In formulations, we can see that entrapment of drug in the nanopores of silica. So in vivo bone affinity was carried out by hydroxyapatite. Here the hydroxyapatite is found in human bones. So the 3 samples were analyzed for 6 hours. So Nucilin 244-FP is having increased binding with HA, and pure drug is showing less binding. Further moving to in vivo studies on arthritis, albino female wrister rats were used. The route of administration was oral. Duration of treatment was 28 days. And the grouping was done in 5 groups, where the 4 groups were injected with CFA to induce arthritis. 0.1 ml of CFA solution was used. Radiographical analysis after treatment period, we can see here the normal group is having normal bone structure, and control group is having swelling of the tissue, and there is bone erosion. For the standard and treated Nucilin treated group, there is reduction in swelling compared to control group. 244 is showing excellent reduction in swelling, and there is no bone erosion. Further moving, these are the images before the treatment, and these are the after treatment images. Here hematological parameters was counted, which was not much affected in 244-FP group. This 244-FP group is showing best result in the treatment period. So for the bone histopathology, for the normal group, here we can see normal structure and architecture of the bone showing the normal joint space. For the control group, here we can see reduction in the joint space, and the synovial membrane is thickened and has become hyperplastic. For the standard treated group and Nucilin group, we can see the bone marrow spaces and aplasis in normal condition. And at last 244-FP group is showing reduction in swelling, as well as the chondrocyte cells are under active proliferation, and there is cellular restoration seen. So 244-FP is giving the best results. So it was further analyzed for histopathology studies and safety assessment. So the liver is, these are the normal group animals. So for the liver histopathology, normal hepatocyte cells and normal central vein was observed. In spleen, we can see white and red pulp in normal condition. Whereas stomach, it shows the gastric mucosa and gastric pits in normal condition, and there is no ulcerative condition in stomach. So we can say that the mesoporous delivery of celecoxib by 244-FP has significant potential in treatment of arthritis with a safer drug delivery. Moving to the conclusion part, the formulation process was simple with high drug loading, and dissolution enhancement was seen in 1:2.5 ratio, and further it was analyzed by BET analysis. And solid state characterization for FTI, DSC, PXRD, particle size and SEM showed the positive result for 244-FP group. And moving further for bone mineral affinity, the formulation showed the highest affinity for bone mineral and compared to pure drug. At last, in vivo studies on arthritis with radiographical images helped in diagnosis and healing effect of the formulation. Hematological parameters were better control in treatment group of 244 FP. Histopathological reports of bone showed the significant reduction in the arthritis condition, and better control was achieved in 244 FP group. And safety assessment was studied on 244 FP group by liver, spleen, and stomach histopathology, which proved that the efficacy and safety of mesoporous delivery of celecoxib. Hence, it can be concluded that mesoporous delivery of celecoxib can be an optimistic and promising measure for the treatment of arthritis. And these are the references which I have used for my study. Thank you.
Hello. It was a good presentation
ma'am.
and a lot of work you have done for the project. It can be clearly seen with the your presentation that you
Yes,
have
ma'am.
put a lot of work in it. One thing I wanted to know, like you are using the mesoporous silica, I think. Hello. What is the
Hello.
solubility of the? Hello. Yes. You are using
Hello.
mesoporous silica. Hello.
Ma'am,
Can
I
you hear
can't
me?
hear you. Can you please repeat the question, ma'am?
Okay. Okay. Can you hear me now? Hello.
Ma'am, you're audible clearly.
Okay. Can you hear me now?
Hello.
004.
Hello. Yes. Your audio.
Yes,
Hello.
ma'am.
Can you hear me?
Yeah,
Can
ma'am.
you hear
Yeah,
me
ma'am.
now?
Now I can hear.
Okay. So I wanted to know whether you have used the mesoporous silica. Okay. So what is the
Yes, ma'am.
two things?
Yes,
What
ma'am.
was the solubility of silocoxib, pure drug? Hello. I think there is some problem. Tabassum ma'am, can you hear me? Dr. Tabassum?
I can hear. I think they are. सिद्धि जरा देखो क्या प्रॉब्लम है उनकी तरफ कॉल करके पूछो बिकॉज वो फ्रोजन दिख रहे हैं यहाँ से.
I think there is some problem with the connection. Can we come back later to that? We can move on.
Yeah,
Okay.
I think so.
As we have seen the presentation and everything else, I think
Yes,
we
we
should
can
move on.
ask them questions again. We proceed. Good idea, madam.
Okay.
We would start with MPH SRC 005. Please turn on the video. Hello everyone. Today I'm going to present my research topic entitled An Anti-Acne and Skincare Formulation from Fruit Paste. So proceeding towards the presentation. These are the contents of my slide. Next is the background. As we know that there are various skin problems upcoming now, and they are increasing at an alarming rate. So we are treating these skin problems by using various plant-based cosmetics, and the use of plant-based cosmetic is increasing nowadays. Plant-based cosmetics are used to cure various skin ailments, and these are used widely because they are natural. They are free from harmful and synthetic chemicals and widely accepted in the public domain. And the use of medicinal plants as the raw materials in the production of new drugs is increasing as they are capable of combating the drug resistance related to the microorganisms. And so the proof of this is that the global cosmetic markets is increasing at an alarming rate, and it is expected to increase at 4.2% during the forecast period till 2030. In 2017, the independent market studies, the global market value for the dried kiwi is 2.8 billion for 2 million metric tons, and it is growing at 4.2% in till 2025. Next.
This is the rationale behind selecting my topic, research topic. This is the problem statement. As we know that acne is a common skin condition affecting 9.4% of the total population and approximately 80% of the population between the ages 12 to 25 years. And acne treatment, there are various routes like the oral route and the topical route, the most commonly used. But the oral route has some disadvantages like the drug metabolism and low bioavailability. Then we have the topical treatments where we use the synthetic chemicals. So problems with the current treatment include the effectiveness, variability, the side effects, the antibiotic resistance, and the recurrence. Now, to overcome all these problems, we have natural chemicals. We have natural phytochemicals like the Actinidia deliciosa. Has been reported to contain various flavonoids and the phenolic substances, which are responsible for the antioxidant and anti-inflammatory conditions. So we have selected kiwi, that is the Actinidia deliciosa, as our main plant, the main fruit. So why we have selected this as it has various benefits, like we can see here the folate. It contains folate, flavonoids, enzymes, vitamin C, prebiotics, fiber, and vitamin E. So due to this wide variety of content in the kiwi, we have selected this. And the statistics of the kiwi is that India consumes about 1,500 metric tons of the dried kiwi. The total of the 13,000 produced in India, Arunachal Pradesh being the major producer of kiwi, and this rate is expected to increase by 2025. So here we can confirm that we can have huge amount of kiwi with us to prepare this type of gel. And as we are using here kiwi peel, so kiwi peel is used as we have seen here that India consumes 1,500 metric tons of kiwi. So those many number of peel waste is generated, we can get it from the industries. So this can be the major source, and we can manage this waste also by using this for production of our gel. And this is the novelty and the market value of and the kiwi gel, like the optimum utilization of the fruit waste for development of a cosmeceutical product, because we are using kiwi peel. No study for peel is reported till date. And for the skincare product, formulation has good antioxidant, SPF, and anti-acne activity. First account of kiwi peel being used as an anti-acne agent. It is cost effective, and prepared formulation shows better result than the market treatment. The salient features is that it is natural, produced from the fruit waste, and sustainable and efficacious. Next, the method we prepared. So the selection and authentication of the kiwi based on literature survey was done. Then procurement and drying of the kiwi waste peel from the fruit. After that, extraction was carried out by maceration technique for 24 hours. Then kiwi peel extract obtained after 24 hours. Here we can see in the next step. Then is the gel based preparation. Then the kiwi extract gel formulation was prepared. Then this formulation was evaluated for the parameters like the formulation parameter, formulation, phytochemical screening, HPTLC evaluation, antioxidant, anti-acne, and last is the sun protection factor. So these were the parameters evaluated. Next, these are the results of the evaluation. Like we can see here, this is the evaluation of the gel, where we can see that it was evaluated for its color, consistency, odor, pH. All these were visual observation. pH was done using the digital pH meter. Viscosity using the Brookfield viscometer, and we can see the viscosity is good and the spreadability is quite good. And next is the antimicrobial activity of the extract by using the direct bioautography method, which reveal the active bands as we can see here. And there are RF values. So these RF values on a constant basis denote that the compound is present, which shows antimicrobial activity. So this technique basically confirms the phytoconstituents having the antimicrobial activity. Here we can see the MIC values of the peel and the pulp. Next, which shows that 10 and 15 mg per ml respectively. Next is the antimicrobial activity of the formulation. Here we had a comparative study for standard which was used as the clindamycin herbal formulation. The marketed herbal formulation Shri Shri anti-acne herbal gel was used. The next, these were the two we prepared, 5% formulation and the 10% formulation. So the antimicrobial activity was tested against the microorganisms S. epidermis and S. aureus. Next, the sun protection factor was also studied, where we found that 10% formulation had better SPF than the 5% formulation. So this is the data which supports the results. This is the evaluation data for evaluation of the extracts. Here we can see the antioxidant activity of the pulp and the peel extract, where we can see the peel extract showed better antioxidant activity, which was performed using the DPPH assay method. And the standard here used was the ascorbic acid. Next is the zone of inhibition. Here we chosen 4 of them, like A denotes the standard, B is the pulp, C is the mixture, and D is the peel. So here we can see next is the HPTLC. So the D1 is the dried peel, as we can see in the figure denoted here. So dried peel, this confirms the activity of the peel, and this is the densitogram of the peel extract. Here these peaks show the confirmation. Next is the bioautography of the extract. So the appearance of white areas against the purple-red background on the chromatogram denotes the inhibition of the growth of bacteria due to the presence of the compounds that inhibit the growth. So this confirms the presence of the compounds that will inhibit the growth of microorganisms. And the constant RF values, as we have seen previously in the table, shows the, and also the clear presence of clear bands with the same RF value. It shows the confirmation that probably same compounds are responsible for the antimicrobial activity for the microorganisms. Next is the data for evaluation of the formulation. First is the drug diffusion study where percentage drug release was performed, where we can see the F1, that is the 5% formulation, and F2 is the 10% formulation. So we can see the 10% formulation had better results, better drug release than the 5%, and the study was done using the trans diffusion cell. Next is the antioxidant activity, and the standard was ascorbic acid. Here also we can see that the 10% formulation was more better than the 5% one in the antioxidant activity. This is the zone of inhibition for the formulation where we have done for S. epidermis and S. aureus with the 5% formulation and 10% formulation. So A here is the control, B denotes the 5% or 10% formulation, C is the marketed formulation, and D is the clindamycin. So these clearly denotes the confirmation of the 10% formulation as the better one. Next is the antimicrobial activity where we have comparative study as we have seen previously. So 10% formulation is as comparable to the standard. It is less as compared to standard, but it is more than the compared one, that is the herbal formulation that was marketed. So we can use this 10% formulation as our confirmatory one. So the conclusion arises here that kiwi peel extract was found to have the phenolic and the flavonoid content. These were better. Then 10% formulation was found to be better than the 5% in terms of the rheological and antimicrobial activities. And the formulation showed better antimicrobial activity than the marketed herbal formulation, but less than the synthetic one. And the developed Actinidia deliciosa peel extract gel showed good antioxidant and antimicrobial activities against the acne-causing microorganisms, that is the S. epidermis and S. aureus. Here we have tested these with these microorganisms as the study showed that S. aureus and S. epidermis were found in the acne patches that were tested. These were probably more in the concentration than the other microorganisms and were responsible for acne, and hence these can be a good alternative for the skincare products. And the kiwi peel extract thus was found to be suitable for the formulation as a skincare product. These are the references I referred. And
Hello. So it was a good presentation, but a few things I would like to ask because kiwi is nowadays very common, you can say, ingredient in lot of shampoos and other ingredients. So in the base, what you have used for the cream or gel, what you have prepared from kiwi extract, kiwi peels.
Preparation and kiwi peel extract is used for preparing
Yeah,
it.
in kiwi peel extract, you have used which base you have used for preparing the formulation?
Base here
Base,
is I
base,
used gel base is used.
gel. Which gel? Which
Gel base,
gel?
she's asking.
Polymer,
Gel
polymer.
base.
Carbopol was used for the gel preparation and kiwi.
How much percentage? You,
Yes,
you,
ma'am.
you don't have a slide of the composition. Can you put the composition of the gel?
Composition, yes. Composition. Can you show the slide for the composition?
No, actually, it is not included in the presentation. The
Okay.
evaluation is included.
But what is
Carbopol
the
1.
around? Okay. Your extract is in dry form or it was in some other form?
It is sticky in nature actually.
Sticky in nature. So, carbopol, with carbopol, you have directly, the extraction is done by what solvent you have used for extraction?
Methanol.
Solvent you have used? Yes, ethanol. Hello?
Methanol.
Methanol.
Yes.
Okay. So, methanol you have used. Okay, you have done the antimicrobial activity, and as you know, there are a lot of traditionally and in market also, we have lot of products like turmeric based
Yes.
products are there. So how your product is better than that?
Ma'am, as we know that kiwi peel contains a lot of flavonoids and polyphenols. It is actually a rich source of everything like phytochemicals, and it is better than turmeric. And it is reported that it has more phytochemicals than others,
Okay.
so it can be good
So you have
alternative.
you have gone through the literature where the comparison is there between the two.
Yes, ma'am. Actually, and it also contains vitamin C in more quantity and the flavonoids and phenolics than the others. So the comparative study is reported already. So we have used this one.
So
So
what
as
is the function?
other,
What will be the function of vitamin C here?
like it has, as we know that vitamin C antioxidant property it has. So it has all the activities like antioxidant, antimicrobial, antibacterial activities. So it can be a combination, and due to more content of the phytochemicals, it can be a good alternative to the current treatments present. Like even though there are turmeric and all other preparations, but this can cure it faster, the acne.
Okay. What will be the, you can say, have you tried it on any like model, animal model you have tried
No,
with
ma'am, not
acne?
yet.
Not
We have
yet.
not. Yes, we have performed
Because
the.
a lot of product, if you going for the if you think about the cosmeceutical, one thinks about the commercialization also. Okay,
Yes,
market and commercialization. So usually in the market we have polyherbal formulation instead of a single formulation, isn't
yes,
it? I think you must have gone through the literature.
yes, ma'am.
So, and for acne, actually, we have a treatment with the, you can say there are APIs available for the treatment of acne.
Yes,
So,
ma'am.
have you, because you have, as compared to standard, what standard you have told me, na? I don't remember. Yeah, the name of the API you are using for standard is
Clindamycin, ma'am.
clindamycin. Is it a regular treatment medication for acne?
Yes, ma'am. It depends actually, but clindamycin is mostly prescribed.
Prescribed. Okay. So as I was seeing from the graphs, there is a definitely a great difference between the activity of the two. If you see the antimicrobial activity with the standard, there is a
Yes, ma'am.
difference. The difference is very large. So is it possible to narrow down this difference by addition of
Uh,
other herbal ingredients or not? One
yes,
thing.
ma'am. It is
Secondly,
possible. Uh,
uh,
we are trying to incorporate those, but firstly, we just checked it for the individual kiwi peel. Like it contains
okay,
and it uh, and as we can see, the comparative study was for the marketed formulation. That was the herbal marketed formulation. So alone
okay.
kiwi peel was better than the herbal marketed formulation. And when we compare it to the standard, we are trying to incorporate other herbals also, like by seeing the compatibility and all.
And
So
what
further,
preservative you have used?
actually, parabens in small quantities were used as a preservatives, and we are trying this to shift towards the natural preservatives to overcome the effects of parabens.
Yes, definitely. Because most of the preparations we are looking for now are should be free from parabens. It is banned in many of the European countries. So, what we are looking for is a paraben-free solutions
At
or the
lab scale,
cosmetics.
we tried to prepare this, and we are trying to find the alternatives for it also, being it natural, so that it becomes totally natural and also it is a cost effective as we use kiwi peel.
Thank you. Thank you very much.
Okay, so is the on this slide only I have are the organisms that you have selected representative of acne?
Yes, ma'am. Actually, according to the study and literature survey, Asorius and Epidermis were found most in the patches. Like they were 92% of all the microorganisms in the acne patches that were studied. So most of the studies are reported for these two microorganisms.
What about P. acnes?
Yes, ma'am. P. acnes actually it is also responsible, but when acne progresses, the most of the condition is worsened because of SORES and epidermis. So we first tried on these, and we are trying to do the activity on the P. acnes also.
What? Why did you select 5% and 10% in the your formulation?
Check the means betterment. Which ones is the better? Like
No, no. So why did you stop at 10%? Because your graph shows 10% is almost double as active as 5.
yes, ma'am. So we were trying at increasing concentration, like which one shows better. So that was the one, and till
No,
10%
no.
we have
What
found.
happens when you have 15%, 20%? Did you try that?
No, ma'am. We are going to
Because
try
you
that
can
also.
get clindamycin jitna benefit. You've not tried, right?
Yes, ma'am. Like
And how
at lab extraction and all, it was not possible at that time. So we just tried it for 10% of that formulation we prepared. We are trying to
when you say
increase.
10%, this is 10% of your dried peel extract, right?
Yes, 10%
What is
of
the color
extract.
of the formulation?
It is brown in color actually.
Is it
The
appealing?
PVP. Yes, ma'am.
Is the color appealing as an anti-acne product?
No, on the first slide I have shown the picture of that formulation, so you can see it as it is brown.
So in the evaluation, when you say go to that same slide. I was just asking because you've seen the
Formulation was evaluated for its color, consistency, odor, pH. All these were visual observation. pH was done using the
viscosity.
pH measure to pH meter.
What are the
Viscosity
units
using
of this viscosity?
Yes, ma'am.
What is the unit of this? There are no units anywhere in your presentation. What is the unit of this viscosity? 7 बोले तो क्या?
Ma'am.
Viscosity, evaluation of gel. What do you mean when pH is characteristic? What do you mean viscosity is 7.1 to what?
Centipoise, ma'am.
Okay, and characteristic pH बोले तो?
Characteristic pH, ma'am, it is actually the value is shifted. The odor is characteristic and the pH is 7.12 and 6.63. It is the skin pH.
And this antimicrobial activity of formulation, what is this 26.76? क्या है ये?
These are zone of inhibition values.
What are the units?
Millimeters.
And is this standard that you have, is it matching with the reported value? Zone of inhibition value of clindamycin?
Yes, ma'am.
So out of, when you've already done the antimicrobial activity, what was the reason to do another antimicrobial activity of the extract?
Ma'am, firstly we confirm for the extract, like at what percent it is then we proceeded for the formulation and the confirmation.
Where is the data of the percent? In this, there is no data which shows that, and here you have done MIC, and here it is zone of inhibition. How are you going to draw parallels कि 5% is the right way to go? You understood? यहाँ पे तुमने नहीं, यहाँ पे तुमने MIC किया है ना? You said you use this bioautography to arrive at the concentration of the active. So what does this tell you? How have you converted 10 mg per ml to whatever 5%?
Actually, that peel extract MIC values are there for that, and based upon the conversion, we have prepared for 5% and 10% formulation.
How did you convert this MIC of 10 and 15 into 5% and 10%? Okay,
based upon the action.
No problem. Thank you.
Thank you.
Thank you. Ma'am, can we ask 004 for Q&A now?
Yes, ma'am.
Yeah, go ahead.
Yes, ma'am.
Okay.
Yes, ma'am. Yes.
Can you? Am I audible now?
Yes, ma'am. Yes, ma'am. Ma'am, because of my network issues, I couldn't.
Okay. Just I wanted to know, like celecoxib solubility. Have you checked?
Ma'am, as such, I have not checked the solubility, but we have done the this drug release, and then we have checked its solubility. So, drug release was the major point, ma'am.
Okay.
What is
So
the difference
So,
between
how
drug
can
release and solubility? Sorry, madam. Drug release and solubility में क्या अंतर होता है?
ma'am, solubility actually, ma'am, the drug is soluble in methanol, ethanol, and it is partially soluble in water. So, ma'am, as such, I have not checked.
Okay, that is the reported
Yeah.
in literature.
Yes, ma'am. Yes, ma'am. Reported in literature. I have done literature review, and then we have moved further for drug release dissolution tests.
because you have said ki after the incorporating it in the mesoporous silica, the dissolution has improved.
Yes, ma'am.
Can you give me some insight ki how what might be the reason for this? Is it
Yes, ma'am.
mesoporous?
Yes, ma'am. I'll tell. Ma'am, actually, this mesoporous silicas are having the tunnable pore size and pore volume. So when we load the drug in mesoporous silica, it get it get loaded in inside the pore and it gets absorbed on the pore. So when the drug is released, at first it is giving initial release, that is spring effect. Initial release is done, and later it is giving prolonged release, that drug it is inside the pores of silica, and at last third stage it will give parachute effect, like embedded drug will release. So it is giving the best drug release, ma'am, and it will increase the solubility of the drug.
What will be your final dosage form? How in what reach the patient?
Ma'am, it can be given as oral dose, like capsules, ma'am.
So okay, celecoxib you are going in for the oral solid dosage.
Yes, ma'am.
What about the solubility of the complex? You have not done, but I hope you tried because
Okay. Yes,
uh,
ma'am. Yes, ma'am. I will try.
okay, because the solubility once it is inside the and what is the stability in the acidic pH? Is there
What,
any
ma'am?
literature? Yeah, you are incorporating in mesoporous silica.
Yes,
What is
ma'am.
the stability of that in the
Ma'am,
your acidic environment?
ma'am, actually, I have done two dissolution tests. One at pH 12, that was US FDA method, and after that, after choosing the best formulations, I have carried out again dissolution at 6.8 pH, ma'am. So the drug release and dissolution rate was enhanced, ma'am.
No, so this
We have
formulation talking is capsule.
Yes, ma'am. Yes, ma'am.
Okay.
Yes, ma'am.
Beta, but when you are taking the capsule from the oral route, if you are giving the drug, it will
Yes, ma'am.
definitely come in contact with your stomach.
Yes, ma'am. Ma'am, for that we have done
So,
safety assessment, ma'am. Uh, Histopathology studies of stomach for 20 after 28 days of the treatment we have done, and as such we have not seen any ulcerative conditions or any morphological changes in stomach lining or gastric pits, ma'am.
In how many days study?
Ma'am, 28 days studies. Even
Generally,
we have done.
what is the treatment regimen of celecoxib?
Ma'am, as
It's like,
such, ma'am, it depends on the severity of ma'am arthritis,
because when you're
ma'am.
talking of arthritis, is it a chronic treatment or people give it for a month and then stop it? What is it?
Ma'am, it depends on the severity, ma'am. Like in how many days it will get reduced. So, ma'am, for induced arthritis rats, ma'am, I have done for 28 days studies. Then after that, I have done radiographical analysis to check the swelling and
bone erosion of the group of animals. And when it was showing the reduce in swelling and all, later we have gone for further bone histopathology studies, ma'am, in which it has shown the reduction in the bone swelling, and due to maybe because of mesoporous silicas, it is giving healing effect, and the cellular restoration was there, ma'am.
What is the metabolic fate of mesoporous silica?
Ma'am, metabolic hepatic metabolism, ma'am, like
Have you done LD50 of your placebo
Yes.
mesoporous?
no, ma'am.
Okay.
Yes.
Um, uh, your formulation is good if it is giving this effect, but I think still have to consider
What, ma'am?
why it is doing so. One thing I wanted to understand the mechanism actually, and whether
Yes, ma'am.
I have gone to the literature, is there any
Ma'am,
preparation available?
ma'am,
Hello?
actually, hello, ma'am, I can't hear you, ma'am, properly. Voice is breaking.
Okay. Can you hear me now?
Yes, ma'am.
So any transdermal or topical preparation is available for celecoxib?
Ma'am,
Because in
uh,
arthritis,
ma'am, as such, my research for the arthritis, there were marketed formulation of Celebrex. It was capsule only, ma'am, by Pfizer, and Velcoxib and Revisreba. These formulations I have found it, not any topical formulation, ma'am.
okay, what have you, because when you are going in for any formulation, you have to check the background. You have to find the logic behind it. Why it is not in the form like transdermal form, whether it is for what is the first pass bypass metabolism of celecoxib.
Ma'am,
Okay,
ma'am,
it's
liver. Yes, ma'am.
it's okay. You just
Yes.
go through a, I think you need the ADMA of the drug, as well
Yes,
as you
ma'am.
have to go through a go a bit deeply in the literature because you have done a lot of work.
Yes,
But
ma'am.
please find out the supporting logical reasons. Why
Yes,
is
ma'am.
it so? Is it
Yes,
so?
ma'am.
But why is it so? Okay.
Yes, ma'am.
So,
Yes, ma'am.
otherwise, you have done. I think you have done a lot of hard work from the whatever data I'm seeing. But I, I also
Ma'am, your voice is breaking, ma'am. Hello.
please find out the logical reasoning behind the things that are done. Okay, beta.
Okay, ma'am. Okay, ma'am.
Yes,
Thank
I
you, ma'am.
just I have a last comment. There is a paper which is there on entrapment of celecoxib into mesoporous silica particles in tablets. How is your work different from that?
Ma'am, actually, I have not prepared a tablet. I have only loaded the drug into the silicas, and then we fill
No, no.
the.
Have you come across this paper? There is this
Ma'am,
paper which is exactly what you have done.
no, ma'am.
So I recommend just go through the paper. It is in Journal of Drug Delivery Science and Technology.
Yes, ma'am. Sure, ma'am. Sure, ma'am. I will check.
Yeah. Okay.
Yeah.
Good work.
Thank you. Thank you, ma'am. Yeah.
Thank you. We will start with MPH SRC 007.
Good afternoon, everyone. I'll be presenting about the research work entitled Birinapant improves imiquimod-induced psoriasis in bulb mites. We start off with the introduction about heart disease. So psoriasis is an autoimmune inflammatory skin disease affecting 2 to 3% of the global population. It is characterized by skin lesions observed mainly on knees, elbows, scalp, nail, and lumbosacral region. The lesions typically show up as elevated, well-demarcated circular red to salmon color, and they are covered in silver scales. These plaques are rough and they are very dry and can crack and peel. Psoriasis has no cure. Even if we see positive results from a certain medicine, the disease is bound to relapse. At present, several anti-psoriatic agents are available, but they have certain side effects which limits their use. Thus, exploring new targets can provide additional therapeutic options. So the aim of our study is to evaluate the effect of birinapant in an animal model of psoriasis. And the objectives are to evaluate the anti-psoriatic activity of birinapant at different doses in imiquimod-induced psoriasis, and to evaluate the correlation of cellular inhibitor of apoptosis proteins, CIAP1 and 2 inhibition with pro-inflammatory cytokines involved in psoriasis. So the rationale. Apoptosis is a process important for regulating the skin homeostasis. And in psoriasis, we observe there is hyperproliferation of the keratinocytes. It might be in correlation that IAPs are overexpressed in psoriatic lesions, and birinapant, our drug, is an inhibitor of CIAP1 and 2. So inhibiting CIAP1 and 2 will result, can provide beneficial in psoriasis as the keratinocyte can then undergo normal apoptosis process. Additionally, birinapant has shown to inhibit the pro-inflammatory cytokines that are involved in autoimmune diseases. So we hypothesize that birinapant can help us improve psoriasis, like treat psoriasis. This is the study design. Our study model is of 29 days, wherein on day 0, hair of the back will be shaved, and then from day 1 onwards for consecutive 8 days, imiquimod cream will be applied onto the shaved skin of the mice. And then from day 9 till day 29, that is a total of 21 days, our treatment will be given. On day 30, animals will be euthanized and skin and spleen will be isolated for further analysis. The methodology. The experimental protocol was reviewed and approved by the Institutional Animal Ethics Committee. Biopsy mice with an initial body weight of 29 to 32 grams were used. After acclimatization of 1 week, animals were randomly divided into 7 different groups, each group consisting of 6 animals. So this is our grouping table. First group is of normal control, second of negative vehicle control, third of disease control, fourth will be our standard, wherein we'll administer cyclosporine 4 mg per kg per oral. And then groups 5, 6 and 7 are our treatment groups, wherein birinapant at doses 10, 20 and 50 mg per kg will be administered respectively. Moving on to the results, we evaluated different parameters like PASS scoring, back skin thickness, spleen length and mass. We'll start with the PASS scoring, that is psoriasis area severity index. Herein the data was collected throughout the study and then evaluated, and as we see that in cyclosporine and birinapant high dose, that is 50 mg per kg, the PASS scoring has significantly reduced at the end of our experiment, that is day 29. And in PASS scoring, we checked for erythema thickness and scaling. Moving on to the bag thickness. The bag thickness was measured using a digital vernier caliper, and at the end of our experiment, that is day 29, we see that there is a significant decrease in back skin thickness in birinapant high dose and cyclosporine group as compared to disease control. And same can be said for spleen length and width, wherein again birinapant high dose and cyclosporine significantly reduced the spleen length and mass as compared to the disease control. These are the histopathological studies. The black arrows indicate the epidermal layer of the mice, wherein we see the keratinocytes, and in imiquimod group, that is disease control, the keratinocyte have become hyperproliferated, that is epidermal hyperplasia can be observed, which is significantly decreased in our standard and all the treatment groups. Specifically, birinapant has shown a significant effect by reducing the epidermal hyperplasia. Biochemical the levels of NF-kappa B, TNF-alpha and interleukin-17 were evaluated using the ELISA kits, and all 3 levels, that is NF-kappa B, TNF-alpha and interleukin-17, were significantly reduced in birinapant high dose as compared to disease control. Again, the antioxidant level, the superoxide dismutase and catalase levels were evaluated, wherein they were found to be increased in birinapant and cyclosporine group as compared to disease control. So as our drug decreased the level of pro-inflammatory cytokines like TNF alpha, NF kappa B, interleukin 17, it also increased the antioxidant enzymes SOD and catalase. Also the other parameters, that is PSS score, back skin thickness, spleen length and mass were significantly reduced by our drug, that is birinapant, specifically at a dose of 50 mg per kg. Thus, we can conclude that birinapant has an appreciable anti-psoriatic activity at the specified doses. These are the references. Thank you.
Thank you for your presentation. So, what is the dose of birinapant and what is the treatment schedule for the same?
And the birinapant was administered in 3 doses, once daily, that is 10, 20, and 50 mg per kg.
Okay, 10, 20, and 50 mg per kg.
Kg. Yes, ma'am.
Per kg. So, what are the, you can say, what is how long the treatment should last? What is the treatment schedule? I wanted to know. For how many days one needs a treatment?
21 days, ma'am.
21 days.
Yes, ma'am.
Is there any associated recurrence?
Was there recurrence after the treatment? What are the chances of recurrence?
And we did not check for recurrence, as on our study ended on day 29, and on day 30th we euthanized the animals for isolating spleen and skin for analysis.
But you must have gone through the literature. And so it says, what are the chances of recurrence of the disease? What is the probability? There must be papers regarding that.
No, ma'am, I had searched for it, but I did not found specifically for birinapant.
No, for any treatment. It's just what are the chances of psoriasis to recur? Is there any literature available for any treatment?
There are chances. Yes, ma'am. All of the drugs that are being used for psoriasis, even if they proven to be effective, there is still high chances of disease relapse.
So, how you would say the birinapant is better than the other existing treatments in treatment of psoriasis?
Yes. So, firstly, like existing anti-psoriatic treatments have certain side effects. Others which have good effects, the other factors that play a role are costing and availability as well. So, newer targets need to be explored. So, that's why I had chosen birinapant because it is also being explored as an anti-cancer drug.
Are there any other models other than the one you used for induction of psoriasis?
Yes, ma'am, there are. The xenotransplantation model is the best model, but then imiquimod-induced psoriasis model is the most widely used because of its ease and convenience. And it mimics the human psoriatic conditions very well.
Okay.
What immunohistochemistry studies did you do? Did you do some immunohistochemistry studies?
Ma'am, I did histopath using H&E stain.
No. Did you do immunohistochemistry?
No, ma'am.
Okay. And what is the mechanism of action of this drug in cancer? You said they are exploring it as an anti-cancer also, right?
Yes, ma'am. Actually, the CIAPs, when TNF alpha binds onto its receptor, TNF receptor, in presence of CIAPs, it is responsible for triggering for like cell survival and proliferation. But when the CIAP is degraded by birinapant, the mechanism of TNF receptor shifts from cell survival to cell death by activation of caspases.
Okay, so it's an caspase activated.
Yes, apoptosis inducer. Yes.
Okay, thank you.
Thank you, ma'am.
Thank you. We would proceed with MPH SRC 008. Zero zero eight, please put your camera on. MPH SRC 008. Are you there? Last call for MPH.
Yes, I'm there.
Okay.
Yes, I'm there.
Okay, we will share this slide.
Good afternoon, everyone. Today I'm going to present my thesis presentation, which is entitled Assessment of New Treatment Approach in the Management of Epilepsy. First, we will start with the introduction. Epilepsy is a neurological disease and it is defined as imbalance between inhibitory neurotransmitter, which is GABA, and excitatory neurotransmitter, which is glutamate. It affects approx 1 to 2% people worldwide. There are a variety of epileptic anti-epileptic drugs like phenytoin, barbiturate, benzodiazepine, but despite of the availability of numerous anti-epileptic drugs, approx 30% epileptic patients are drug resistant. So there is a need to test new medication for the treatment of epilepsy. Rationale. As we know, glutamate is the primary neurotransmitter which is responsible for pathogenesis of epilepsy. My selected drugs are riluzole, which is marketed for ALS, is amyotrophic lateral sclerosis, and ceftriaxone, which is a third generation of cephalosporin. Riluzole has three mechanisms. It blocks voltage gate sodium channel, block glutamic acid release, and block excitatory receptor. Ceftriaxone increases the expression of GLUT1, which is glutamate transporter 1. It is also known as excitatory amino acid transporter EAT2, which is responsible for reuptake of glutamate in neurons and glial cells. So thereby it reduces the glutamate transmission and thus excitotoxicity. So we hypothesize that combination of riluzole and ceftriaxone gives synergistic effect in the treatment of epilepsy. For study, we used aim and objective to evaluate new treatment approach in the management of epilepsy. Objective to explore and testify anti-epileptic drug riluzole and ceftriaxone in the PTZ model, to compare the action of individual drugs and combination of drug to check the synergism. For studying, we use male Vistar rat. Then they divide into 8 groups before commencement of the study. Animals were acclimatized for 1 week. Then on alternative days, till 11th day, PTZ was injected in a dose of 35 mg per kg intraperitoneally. After the 5th dose, rats were fully kindled, as you can see here, which is on 9th day. Then re-challenge dose were given on 15th, 20th and 25th day to check the induction of disease. On 11th day onwards, test drugs were given regularly till 25th day. On 25th day, behavioral parameters were assessed, and on 26th day, animal was sacrificed and brain tissue were recollected, and then further evaluation were done. Grouping. First group is normal control. 0.9% saline were given. In disease group, only PTZ were given in the dose of 35 mg per kg intraperitoneally. In standard control, PTZ plus sodium phenytoin were given in the dose of 75 mg per kg. And vehicle control, PTZ plus 0.5% sodium CMC were given. In treatment group 1, PTZ plus drug 1 is which is riluzole, which is given in 5 mg per kg orally. In treatment group 2, PTZ plus ceftriaxone sodium in 100 mg per kg were given. In combination half dose, PTZ plus riluzole in 2.5 mg per kg plus ceftriaxone sodium in 100 mg per kg were given orally. In the last group, this combination of full dose that PTZ plus riluzole in 5 mg per kg orally were given plus ceftriaxone sodium of 200 mg per kg were given. Then various evaluation parameters were done. Behavioral parameters, seizure latency, which is the time of onset of seizure after PTZ injection, and seizure duration, locomotor activity using actophotometer were done. Rotarod performance for motor coordination were done. Then we did biochemical parameters, which is estimation of interleukin 6, GABA, glutamate, and antioxidant enzyme, which is catalase and SOD. Then results. In seizure latency increases significantly in combination of full dose and standard control compared with the disease control. Seizure duration, it decreases significantly in standard and combination of full dose as we compare with the disease control. They both are recorded in minutes. Locomotor activity and rotarod performance increases significantly in combination full dose and standard than disease group. Glutamate, which is responsible for excitotoxicity, decreases significantly in combination of full dose and standard group than disease group. GABA level were increased significantly in combination of full dose. Then catalase and superoxide dismutase antioxidant enzymes level increases significantly in combination of full dose and standard group than disease control. Interleukin 6, which is a pro-inflammatory cytokine, the level were decreases significantly in combination of full dose and standard group than the disease group. Then we did histopathological evaluation. Here we check the neuronal degeneration. So in normal control, there is no degeneration of neurons. In disease control, as you can see, there is a neural loss. And in treatment group, there is decrease in neural loss and combination of full dose has significant effect. Then conclusion. In this study, we evaluate the anti-epileptic effect of riluzole and ceftriaxone individually and in combination. Results showed that treatment with riluzole and ceftriaxone decreased the transmission of glutamate and increased the GABA transmission. It also showed that decreased duration of seizure and increased seizure latency, motor coordination, as well as locomotor activity. Thus, we can conclude that the combination of riluzole and ceftriaxone sodium have synergistic effect. Then these are my references. Thank you.
Sabasu ma'am, can you please take the lead in this or Meera?
Yeah.
Just
Okay. So, why did you select this PTZ model? You've got many more models, right?
Yes, ma'am, but it is the more prominent one. That's why.
What was your basis for selection?
Because ma'am, I selected this for chronic treatment. So our approach is to treat the epilepsy, not the preventive one. That's why I choose this model for 25 days.
Okay, and did you check the toxicity of the combination? Did you do a LD50 of the combination?
Yes, ma'am. I did that. Riluzole have oral LD50 value is more than 45 mg per kg in rats, and ceftriaxone has more than 10,000 mg per kg for rats orally.
Ceftriaxone is 10,000 mg per kg. This is a reported value, or
Yes.
this is what you have got?
I got this from safety data sheet.
Okay, no, did you do a LD50 of the combination? You have selected a combination, right?
Yeah, no, I did not do this. I didn't do.
Okay, is there a combination available in the market of any one of these with any
Yes,
other drug? Not these two. These
not
drugs.
in the market, not in, ma'am, not in the market, but it is done in preclinical studies. Riluzole done with the valproic acid.
Hmm. No, any in the in market, do you have any combination anti-epileptic treatment?
No, ma'am.
Okay. What is the dose of the these two drugs available in as individual products?
Ma'am, Riluzole has 5 mg per kg, and the other one is 200 mg per kg. मतलब the significant dose of both of drug is this.
Okay, so this is the effective dose.
Yes, ma'am.
Okay, and the term for the treatment term is same as what you have used in your model.
Yes, and it gave the significant effect when I used in combination.
But you've done only one model, no? That's what it's saying. So if both are known drugs, would it just think about it? Would it be useful also in comparing in the other model of convulsions? You can just have a search and maybe substantiate what you have observed with additional model data, because then you are sure, yes, this combination is working, because this combination is not reported, correct? You have hypothesis,
Yes.
so it will just prove your
Yeah.
hypothesis, or rather it will strengthen your hypothesis. So just read up and see if you can plan some more models, epileptic model ka a same combination.
Yes, sir. Yes, ma'am. Will do.
Okay, okay, thank you.
Thank you, ma'am.
Thank you. It was a good presentation, and you can say this is the kind of work because existing drugs we are using for a betterment. Okay, but a doubt question is there. Ki, have you gone through the side effects of both the drugs? What will
Yes,
happen?
ma'am.
Okay, so what are the major side effects? Are is there is a chance that the situation or the side effect will escalate in case we use the combination? Is there any chance of that? Have you gone through the literature?
Yes, ma'am. I did, and there is no chance to elevate the side effects when both are given in combination.
You have the support of
Side effects are
literature or
not.
yes, please continue.
Yes, ma'am.
The side effects. What are the major side effects?
Major side effect. Actually, ma'am, there is no major side effect as such. Riluzole has gastrointestinal gastrointestinal symptoms and or lethargy, and ceftriaxone has bloating, nausea, vomiting. That kind of side effects are there.
Okay. but lethargy is there. But it is indicating there are some sort of depressant effect of this drug on the brain, CNS, because both are acting on CNS system. Therefore, one has
Yes.
to be cautious.
Ma'am, it can actually, ma'am, in epilepsy, we want depressive like symptoms only, because in epilepsy there is a hyperactivation. So we want to reduce the glutamate level.
Yes. Uh, okay. I think, as Madam has suggested, you have to go in for the LD50 for the combination also, because sometimes a combination, the scenario is entirely different. So
Yes, ma'am.
I would suggest, once when you are thinking about the combination, and if it is showing a better effect, then you should going for the LD50 value also for the combination. Okay,
Yes,
that's
ma'am.
it from my end. Thank you.
Thank you, ma'am.
Thank you. We would start with MPH SRC 006. Zero zero 009, please on your video.
Hello everyone. My presentation topic for the fourth research student research congress is gastroretentive modified release tablet for Helicobacter pylori infection formulation and its evaluation. So my ID is MPH-SRC009. So the contents are introduction, background, rationale, aim and objective, method of formulation, method evaluation of formulation, result and conclusion. So background. Helicobacter pylori is a type of bacteria that can infect the stomach, and it also can damage the duodenum and tissue of the stomach, which is the first part of the small intestine. It can cause inflammation, redness, and soreness. The H. pylori includes gastric ulcer and resistance to the antibiotic due to the poor patient compliance. The rationale is a localized delivery of antibiotic in the stomach along with the proton pump inhibitor to the intestinal region for the effective treatment of H. pylori infection. The selection of dosage was no fixed combination of antibiotic and proton pump inhibitor was available in market, so we decided to make it. The aim is to formulate a tri-layer tablet consisting of maxofloxacin hydrochloride as a gastroretentive layer and pantoprazole sodium as a delayed and sustained release layer. The objective are evaluation of formulate formulation containing moxofloxacin hydrochloride and pantoprazole sodium in tri-layer tablet. The stability study of the formulation as per ICH Q1AR2 guidelines. The method of formulation. First, to prepare a first layer of maxofloxacin hydrochloride as a gastroretentive layer, the process is drug plus excipient, blending with geometric mixing, the powder blend is formed. After the powder blend is formed, the addition of binder to form the weight mass, and after the exclusion, the pellets are formed. Then the second layer of effervescent, the excipient is added, and with the geometric mixing and blending, the powder blend is formed. As the powder blend is formed, the binder is added and wet mass is formed. After the screening and drying, the lubrication is done and the granules are formed. After the formulation of granules for the pantoprazole sodium delayed and sustained release layer, the drug and excipients are added and the blending and geometric mixing is done. After the geometric mixing, the powder blend is formed and the binder adhesion addition is done to form the wet mass, and extrusion is done to formation of the pellets. The pellets are added to the stearic acid plus PEG 6000 at 60 degrees centigrade to form the polymeric matrix with the drug, and it is cooled up to 25 degrees centigrade, and screening and lubrication is done, and the formulation of granule are done, and it's the formation of third layer. The after the formation of three layer, the compression of tablet is done by rotary tablet compression machine. First layer of moxifloxacin hydrochloride, second layer of effervescent layer, and third layer of pentaprazole sodium. The tablet is formed. The evolution of developed formulation is done, and the weight variation test 1.681 gram. The hardness was found to be 6.88. The thickness was 7.40. The friability was 0.1%. The splitting time of tablet was 10 to 25 seconds. The lag time is 21 minutes. The floating time was 12 hours. Disintegration for delayed release, it is pantoprazole sodium 1.41, and assay was done. The moxifloxacin was found to be 102%, and pentaprazole sodium was found to be 98.72%. After the addition to the zero point 0.1 normal HCl, after 10 to 25 seconds, the gastroretentive layer goes upper to the upper side of the beaker, and the sustained release layer of pentaprazole sodium settle down to the bottom. The in vitro release profile. In the in vitro release profile, moxifloxacin hydrochloride and pentaprazole sodium was treated with 0.1 normal HCl, where after 2 hours, moxifloxacin hydrochloride shows 53.23%, and pentaprazole sodium show 8.59% respectively. As the study was continued, for moxifloxacin hydrochloride show 98% at the end of 7 hours, but the buffer was changed to the pentaprazole sodium was phosphate buffer 6.8 and show release up to 92.88% at 9 hours. The kinetic study was done. The correct correlation coefficient was done for moxifloxacin hydrochloride. The R square value was found to be 0.9823, and the correlation in vitro release profile for pentaprazole sodium was found to be Coesper-Pappes model 0.9856. The conclusion was gastroretentive layer ensure the prolonged content in the stomach, sustained release layer targets to the duodenum, and improve the treatment efficacy to the localized to the dosage form. The potential to reduce the dosage frequency was done. These are the some key references. Thank you.
Thank you for the presentation. One thing I wanted to know why you chose the moxifloxacin for the treatment of ulcers.
Ma'am, the moxifloxacin show the inhibition of topoisomerase of 2 and 4 of the bacterial DNA, and inhibits the transcription.
Inhibit the transcription for bacteria. H. pylori.
Yes, ma'am. Bacterial DNA. H. pylori bacterial DNA. Topoisomerase.
But is it specifically for halibacterial, Hali-pyroli? It is doing this the same?
Yes, ma'am.
Okay. Is it used in the treatment of the peptic ulcer?
In peptic ulcer, the pantoprazole is used. It
Okay.
inhibits the H plus and K plus pump inhibitors and leads to the inhibition of gastric ulcer.
Okay. But you are using in combination. Do you
Yes,
know
ma'am.
fraction moxifloxacin is having
What, ma'am?
interaction? How many drug interaction moxifloxacin is having?
Ma'am, due to the effervescent layer in between the tablet, the first layer of moxifloxacin, then the effervescent layer, then the pantoprazole sodium layer is there. Because of that, there is no interaction between pantoprazole and moxifloxacin.
But it is that actually moxifloxacin has the impact without anti-medications.
Ma'am,
Madam,
I
your
am not
voice is
able to
cracking.
hear.
490 medications have interaction with moxifloxacin.
Yes, ma'am.
Okay, and your one salt is it is a hydrochloride salt,
Yes.
and other is your sodium salt. Pantoprazole
On,
sodium is there,
yes, pantoprazole sodium.
sodium, and your this moxifloxacin hydrochloride is there.
Yes, ma'am.
Uh, uh, and effervescent you will release first. Effervescent layer is having outermost layer is
Effervescent.
effervescent.
No, ma'am.
Yes.
First layer is of norfloxacin, and in the norfloxacin, after the second layer is of effervescent layer, and third layer is of pantoprazole sodium. After the 24 second, it's in a acidic condition. 10 to 25 seconds, the effervescent layer remove, and the two layers are separated. Norfloxacin is gastroretentive, so it retains on the surface of the stomach of the acid, and the second layer of pantoprazole sodium settle down to the stomach and go to the duodenum, and where the action is shown of pantoprazole sodium, where the K plus and H plus pumps are there.
Have you done any in vitro studies on this? How much time it takes to
Ma'am,
separate the layers and how they are behaving? Have you done any photography or any studies regarding that?
no, ma'am.
Because you need whenever you are going for the gastroretentive, because your mechanism is seeing like two layers are getting separated. One is
Yes,
the and both are gastroretentive. One is settling down, other is floating, isn't it?
yes, yes, ma'am.
Okay. So, what is the floating time? What is the lag time?
Ma'am, I have mentioned into the slide.
Yeah,
Floating
please go back to that slide.
in evolution test. Two layer reiteration test.
Yeah,
Here
yes.
1.681
Go back.
gram. Like the hardness was found to be 6.88. The thickness.
Uh, just go back to the picture where you have shown the picture.
Ma'am, this picture.
Yes, yes, this one. This. Thank you. So, your sustained layer, from what I can judge, what is seen from my side is you are one of the this pink layer is for
That last one pink layer is for pantoprazole sodium.
pentaprozole
Last layer.
sodium.
It is settled down to the bottom, and the upper layer, which is a gastroretentive layer of moxifloxacin, in which we had added sodium bicarbonate, carbonate, and citric acid for its floating properties,
Float.
effervescent and floating properties.
And another thing I wanted to know, कि, moxifloxacin, can you take it empty stomach?
No, ma'am.
And pantoprazole Usually, the doctor recommends it in the morning, uh, empty stomach. So, how you are going to manage both things at the same time?
Can you repeat question, ma'am?
As such, doctors.
Hello.
Wait, man. कुछ lag आ रहा है.
Okay, ma'am.
Okay, can you hear me now?
Yes,
Yes,
ma'am.
madam.
What I'm trying to say, when doctors recommend pantoprazole sodium on empty stomach, morning, first thing in
Yes,
the
ma'am.
morning, okay?
Yes,
But
ma'am. Yes, ma'am.
your moxifloxacin hydrochloride you have to take after food,
Yes,
according
ma'am.
to you. Yeah. Then how you are going to manage both?
Ma'am, that pantoprazole sodium is unstable in gastric condition, so we have made it it's alkaline in nature by using the light magnesium oxide.
Okay. Munira, please. Hello. Can you get me?
Yes, ma'am.
Okay.
Yes, ma'am.
Because okay. So what is your? Just go to the slide with the release, the drug, indicating the release of the drug. Dissolution profile.
Ma'am, this slide.
Yes, yes. Dissolution profile.
What, ma'am?
Dissolution profile you have done for
Ma'am,
pentaprozole. Dissolution you have done media you used. What is that cycle and the media you used for the dissolution profile?
ma'am, can you repeat, please? It's your voice is breaking.
What is your dissolution profile and the time period
Yes.
for dissolution? What is your, you
हाँ.
can say, the dissolution profile के लिए आपने क्या क्या किया बेटा? आपने कौन से मीडिया यूज किए, कितने घंटे के लिए डिसोल्यूशन लिया, क्या
Okay,
मीडिया
okay,
चेंज
okay,
किया,
okay.
मुझे बताइए.
Yes, ma'am. Yes, ma'am. Yes, ma'am. First we have taken 0.1 normal HCl for the gastric purpose, and in which the benzofloxacin show 53% release in 2 hours, and pantoprazole sodium show 8.59% release in 2 hours. But due to the settling down of the pantoprazole into the duodenum, it's changed the pH up to the 6.8 phosphate buffer pH we have taken, and the second media was taken of phosphate buffer, and the release of pantoprazole shown there, and it showed the release for up to 92% at 9 hours for pantoprazole sodium.
Actually, I wanted to know the reason. I wanted to know the logic behind using 6.8 when you are preparing a gastro-retentive dosage form.
Hello.
Hello, Munira.
Yes, ma'am.
You continue with the questions. I think there is some network problem from my end. Siddhi, can you hear me?
Summa,
Hello?
you are muted.
Can you hear me?
Yes, Garima ma'am, you are
Yes, we
audible.
can hear you, madam.
Okay, so I think there is some problem with the connection. My question was why you are using 6.8 pH for dissolution study when your formulation is gastroretentive. In the stomach, it
Ma'am,
is
gastroretentive is for moxofloxacin hydrochloride. Ma'am, I want to clear that moxofloxacin, it's for moxofloxacin gastroretentive, and after the settling down of the pantoprazole sodium, it's going to the duodenum, which pH is 6.8. So we are using phosphate buffer for duodenum.
okay. Okay, thank you. Thank you.
What is the method of analysis that you have used for the release studies?
UV spectrometer, ma'am.
Wavelength?
Wavelength for amoxicillin was 292 and pantoprazole sodium was 280 in the zero 0.1 normal HCl, and for only pantoprazole in 6.8 buffer it was 284, and in distilled water it was distilled water I haven't done that. It's R square value I have done for it's 0.99
R
for
squared?
both
No.
the
Why is
pantoprazole as well as
the wavelength different?
where, ma'am?
In your media? In 0.1 normal HCl and in 6.8 phosphate
Ma'am,
buffer,
wavelength are same.
why is your wavelength different?
In
Okay, I think we should proceed.
ma'am, same.
Then why it is same?
Thank you, ma'am.
Proceed next.
Okay, ma'am. We would like to call upon MPH SRC 010. I request MPH SRC 010.
Hello and good day to everyone. My code for 4th Research Congress is 010. The topic, For my oral presentation is enhancing oral drug delivery formulation and evaluation of rapid disintegrating tablets for improved combination therapy of olmesartan and hydrochlorothiazide in hypertension. As per FDA, oral disintegrating tablet, that is ODT, is defined as a solid dosage containing medicinal substance which disintegrates rapidly, usually within a matter of seconds when placed upon the tongue. With this, I want to explain the rationale for selection of disease, drug, and formulation. Hypertension is transitory or sustained elevation of systemic arterial blood pressure to a level likely to induce cardiovascular damage. Hypertension is defined as a systolic blood pressure above 140 mm of mercury or a diastolic blood pressure above 90 mm of mercury. Hypertension itself is not a disease, but it increases the risk of cardiovascular health. Approximately 214 million blood pressure patients are expected in the country by 2030. The 6th Joint National Committee on Detection, Evaluation and Treatment of High Blood Pressure recommends combination therapy, that is, fixed dose combination, as an initial treatment for hypertension. FDC therapy with two different classes of antihypertensive agents will achieve goal pressure in more than 70% population. The combination of olmesartan and hydrochlorothiazide has been shown to be an effective and well-tolerated treatment option that provides blood pressure lowering efficacy and improves elevation of blood pressure in hypertensive patients. Olmesartan is an angiotensin 2 receptor antagonist which blocks the binding of angiotensin 2 on AT1 receptor and prevents vasoconstriction and fluid retention, resulting in antihypertensive activity. Whereas hydrochlorothiazide, a thiazide diuretic, reduces the reabsorption of electrolytes from the renal tubule, resulting in increased excretion of water and electrolytes, which include sodium, potassium, magnesium, chloride. Olmesartan hydrochlorothiazide in combination reduces the systolic and diastolic blood pressure more effectively than either component as monotherapy. The formulation of selection is ODT, that is orally disintegrating tablets, which are also called as mouth dissolving tablets or melt in mouth tablets. The disintegration time for ODTs generally ranges from about several seconds to about a minute. The reason for selection of ODT is for dysphagic population, that is the people who have difficulty in swallowing, which include the geriatric, pediatric, and disease condition patient. Since such a tablet can disintegrate only in small amount of water in the oral cavity, it is easy to take by a patient. For example, it can be taken anywhere at any time by anyone who do not have easy access to water and has no risk of choking. These are some of the key literatures which I have used during my journey for the formulation and evaluation of FDC as an oral disintegrating tablet for hypertension. The current marketed formulation available for fixed dose combination of olmesartan and hydrochlorothiazide are Benicap, Allmark, and Olmesast, where Allmark H of Intas Pharmaceuticals has been used for comparative study with the optimized formulation. The marketed ODT formulations are Belzan, which is a Valsartan, an antihypertensive agent, and Maxaltan Deprexia as anti. Hello and good day to everyone.
SRC 010, your presentation is over or there was any glitch?
There was a glitch. It ended in between only.
Yeah. It just went off, something wrong at our end.
Yes.
Comparative study with the optimized formulation. The marketed ODT formulations are Belzan, which is a Valsartan, an antihypertensive agent, and Maxaltan Deprexia as anti-migraine and antipsychotic agent. Thus, the market analysis shows that there is no marketed ODT preparation as an FDC for antihypertensive effect, which results in selection of olmesartan and hydrochlorothiazide as a drug of choice. The aim of my research is to formulate and evaluate oral disintegrating tablet for administration of fixed dose combination containing olmesartan and hydrochlorothiazide for the treatment of hypertension.
whereas the objectives are to conduct the pre-formulation and drug excipient compatibility studies, to formulate and evaluate oral disintegrating tablet of fixed dose combination, to compare in vitro release profile of test with a marketed film-coated tablet of fixed dose combination containing olmesartan and hydrochlorothiazide, that is the Intas H pharmaceuticals tablet. The pre-formulation study showed that the appearance of both the drugs are white crystalline powders with a sharp melting point. The solvents used for solubility studies are water, pH 1.2 and pH 6.8 buffers. All the excipients and drugs were run for the compatibility studies, which shows no change in visual appearance at different temperature and conditions. The drugs individually, in combination, and in combination with excipients were observed for FTIR and DSA for a period of 4, 8, and 12 weeks under stress condition, which shows no interaction with excipients even under stress condition after 12 weeks. Oral disintegrating tablets of olmesartan hydrochlorothiazide and combination were formulated using various super disintegrants by direct compression method. The super disintegrants like cross-caramelose sodium, cross-povidone, SHD, which have great impact on the rate of disintegration and dissolution of tablet, mannitol as diluent, MCC as binder, sucralose and sodium as saccharine, as sweetener, vanillin as a flavoring agent. The proposed methodology for the formulation is direct compression because of its advantage in shorter process and cost saving. All excipients were sifted from 40 mesh strain and mixed in polybag by geometric ratio and blended for 15 minutes. Finally, the blend were lubricated with talc and magnesium stearate using as a glidant and compressed using 8 mm and 10 mm flat punches in Rimac compression machine. The developed formulations were evaluated for hardness, friability, weight variation, disintegration, drug content. Cross-caramelose sodium, the overall result indicated that the cross-caramelose sodium had a higher edge of acceptability, of which in vitro integration were within the prescribed limit and comply with the criteria for ODT. Thus, F4 and F11 are the optimized batch for 200 mg and 400 mg tablets. The in vitro dissolution study shows the percentage release of dissolution media in dissolution multimedia, that is pH 1.2 and pH 6.8, with 90% release within 5 to 15 minutes. ODT developed for combination using super disintegrant like cross-caramelose shows good disintegration and dissolution profile. Formulation F4 and F11, which has been considered for optimization by performing repetitive analysis, were compared with the marketed formulation Allmark H20 Intas. A comparison of optimized formulation shows that the olmesartan and hydrochlorothiazide were effective and suitable than conventional film-coated tablet of all the parameters. Thus, the stability shows that no significant changes in physical properties, which are tablet hardness and disintegration time, no significant reduction of percent drug content of stability sample. With this, we can arrive to the conclusion that the ODT were found to have better drug release as compared to marketed film-coated tablet formulation. Conclusion: the developed ODT for olmesartan and hydrochlorothiazide to enhance patient compliance and therapy. Optimized formulation demonstrated stability in accelerated studies over 3 months. Further investigation, including in vivo bioavailability and pharmacokinetics, are recommended as a comprehensive assessment of performance. These are my references. Thank you so much for your attention.
What are the prescribed, what are the pharmacopoeial standards for ODT?
As per Indian Pharmacopoeia, it should.
Can you go to that table where you have the parameters? Optimization वाला जो table है. हाँ, ये वाला. This is the composition. Sorry, this is the composition. Okay, this, yeah.
Yeah, ma'am. As per Pharmacopoeia, the disintegration time should be less than 1 minute for the ODT tablets.
Correct. No, what is the basis for you to select F4? If you see F3, there is not a significant difference but it's got a better content uniformity. So any particular reason why F4 over F3?
Yes, ma'am. Disintegration time was the main reason for my selection of this.
2 seconds.
Yeah, ma'am. But it was giving a comparatively better, so that's why I have selected the F4 as
F7
optimized.
is 19 seconds, which is more than F3.
Yes, ma'am, but it is in twice the dose. Like you can see the weight uniformity, it is 200 and 400 mg.
So what? So you shortlisted one for 200 and one for 400. That's what.
Yes, ma'am. The one which I have shortlisted for the 200, I have done 400 for that particular optimized batch only.
Okay. What is the market share of ODT products? How well it is acceptable to the population?
Ma'am, like it is better for the patients, like they can take it.
Put your conclusion slide. Conclusion.
Yeah.
So your first this thing is enhance patient compliance and therapy. That's your first conclusion, right? So what is the patient accept? Do you have any data on patient acceptability of ODT versus conventional tablets?
Yes, ma'am. Like hypertension is a case during the migraines or in such condition, and while people are traveling, they might have issues. So they can take the ODT tablet without the use of water, and it is for immediate use. So and also for the geriatric and pediatric population, like there is a case of dysphagia, difficulty in swallowing. So for such crowd of population, ODT is a better dosage form as compared to the normal film coated tablet where there is a difficulty in swallowing.
No. This is your hypothesis, no? Is there any data which shows that the market share of ODT products is 25% more than the conventional tablets? That is what I was asking. This is
no, right now. Yeah. Right now I don't have the market data, but I can surely check for it. But the main target audience were the geriatric patient and the dysphagic patient, so that we can have a better compliance in such categories.
Okay, that's all from my side.
Garima ma'am.
Madam.
Can you continue me? Please.
Yes, ma'am.
Please take the data also.
Next.
Yes, ma'am. We would proceed with MPH SRC 11.
Yes, ma'am.
SRC 10, we are done with the presentation. Thank you.
No, ma'am. I just wanted to mention that for SRC code 11, I am the co-author, and the presenting author, there was a death in his family, so he is not present right now.
Okay.
Yeah, so I am going to defend for him.
Hello and warm greetings to one another. My code is 000 and my title for oral presentation is site-specific delivery of resveratrol to colon cancer cells, uracil-based nanoparticles functionalized with ligands in delayed release capsule. Colon cancer. Colon cancer, also known as colorectal cancer, starts in the colon or rectum and which are part of the lower digestive tract. Treatment involves surgery, radiation therapy, chemotherapy, and targeted therapy. Minimal cost of chemotherapy or radiotherapy per cycle is around 65,000 rupees, and that for colon cancer surgery costs around 3.5 to 5 lakh rupees. Here we can see the number of new cases in 2020, including both the sexes and all the ages. Colon cancer contributes about 10%, and number of deaths also colon cancer contributes about 9.4%. aim to formulate and evaluate ligand functional nanoparticles of resveratrol for colon cancer. Objectives to prepare, design, and develop pH-sensitive nanoparticles for site-specific and selective delivery to colon cancer cells of resveratrol functionalized with ligands filled in a delayed release capsule. To evaluate and characterize the developed formulation for physiochemical parameters, drug release, and stability. Background and rationale. Resveratrol. It's an anti-inflammatory agent. Inhibits oxidative stress in colon cancer. Decreases colon cancer cell survival, easily available, low cost as compared to existing chemotherapeutic agents. While ligand-based strategy, CD44 are highly overexpressed in colon cancer cells. That is why, by selecting this strategy, we can highly selective and specific action of the drug, improve drug payloads, decrease side effects, enhance therapeutic action, decrease cost of therapy, improve quality of life for cancer patients. Going towards the background and rationale for ligand-targeted polymeric nanoparticles. By choosing this approach, we can enhance the solubility, decrease the overall dose, improve stability, enhance uptake, and targetability. Rationale for using combination of the polymer. Nudragine RS100 is cationic in nature. In conjugation of negatively, which helps us in conjugation of negatively charged hyaluronic acid. S100 will ensure a colon target. Combination of both will result into pH as well as time-dependent nanoparticles. Here we can see if the nanoparticles are time-dependent, therefore they get released in the pH 1.2 of the stomach. But if the nanoparticles are pH-dependent, they reach up to small intestine and get released. But when the nanoparticles are pH as well as time-dependent, here we can see nanoparticles avoid complete drug release and deliver drug to the inflamed colon. Here are some examples of clinical trials and patents with respect to the colon cancer. Reformulation studies, including solubility studies, PSC and FTIR were performed. And by here, we can interpret that resveratrol was soluble in ethanol, acetone, and phosphate for pH 1.2, 6.8, and 7.4, including 1% of phenylating. AMD, analytical method, was developed and validated by you. Here we can see UV spectrum of resveratrol was developed in methanol for pH 1.2, 6.8, and 7.4. And here we found out that lambda max was found to be 305 nanometer. Calibration curve was also plotted in methanol, phosphate buffer pH 1.2, 6.8, and 7.4. Also, analytical methods were validated for linearity, correlation coefficient, slope, intercept, LOT, LOK, precision, robustness, and accuracy. Formulation and development. Organic phase containing polymer and drug and surfactant was added dropouts into the aqueous phase containing PVA. This leads to formation of polymeric nanoparticles. Now, after the formation of the polymeric nanoparticles, we have to do conjugation of the nanoparticles by HN. By addition of nanoparticle dropouts into HA solution. After the nanoparticles are conjugated, we have to lyophilize the nanoparticles and thereby fill in a delayed release gap. Graphical representation of conjugation process. Here we can see PVA in water and also RS100 and S100 polymeric mixture and resveratrol in ethanol. Nanoparticles are formed by solvent emulsification method. At the surface of the nanoparticle, we can see RS100 and S100 polymer molecules. And HA being anionic in nature, it is attracted towards the positive RS100 polymer molecules. And by ionic interaction, conjugation is happening. Preliminary batches of unconjugated nanoparticles and conjugated nanoparticles. These were prepared without the drug and by changing the ratio of polymer and surfactants. And after the unconjugated nanoparticles were formed, they were selected based on the particle size and observation, that is, they are turbid or clear. And for conjugation, hyaluronic acid was used in the concentration of 0.1%, 0.25%, 0.5%, and 1%. FTIR spectra of conjugated nanoparticles. The black one is the FTIR spectrum of the conjugated nanoparticles with HA. And here we can observe that the nanoparticles are perfectly conjugated with HA. Optimization of resveratrol nanoparticles. Herein the surfactant and drug to polymer concentration ratio was varied and the batches were screened for particle size, zeta potential, PDI, and intrapendent force. The particle size and zeta potential of the batches 5, 6, and 9 are as follows. In vitro release study. The in vitro release study was performed at temperature 37 degrees Celsius RPM and almost 22 ml. The study was firstly performed for 2 hours in 0.1 normal HCl, for 2 to 6 hours in phosphate buffer 6.8, and further in phosphate buffer 7.4. All the 3 mediums include 1% phenate membrane, high media dialysis membrane, 50,000 Dalton. And this was the release study. Herein we can see all the 3 batches are not showing not more than 10% release in 4 hours and 20% and more than 20% release after 6 to 7 hours. And the 100% release is reaching in 14 hours. These are the 10 images of the formed nanoparticles. Spray drying and lyophilization of resveratrol nanoparticles comparatively yield analysis. Here, the lyophilization was done with the help of two processes. Here, the spray drying and lyophilization of the resveratrol nanoparticles are done. Here are the variables of the two processes: freezing temperature, freezing time, shelf temperature, and pressure. And the process parameter for spray drying, that is inlet temperature, outlet temperature, aspirator speed, and feed pump rate. By now, comparison of the lyophilization and spray drying, we get yield in lyophilization is 95 to 100%, but time required is 16 hours, and the texture is flaky. But in the case of spray drying, yield is 63%, time required is 2 hours, and the texture here obtained is smooth and granular. Formulation of the powder. Now, the spray dried powder has to be filled into the capsule. Herein, equivalent amount of resveratrol is taken, equivalent to 20 mg of resveratrol is taken. Spray dried powder equivalent to 20 milligram of resveratrol is taken, that is around 320 milligram. Aerosol 200 and magnesium stearate were weighed 5 and then they were filled into the delayed release capsule. Formulation of the capsule. Resveratrol nanoparticles by solvent emulsification have been formed. Then they have sprayed that into nanoparticles, and then each capsule containing 20 milligram of resveratrol was loaded. Evaluation of the formulated powder. Same images. Herein we can see the spray dried nanoparticles are near to circular in shape. Powder analysis was done, including bulk density, tab density, angle of rainbow, and etc. In vitro dissolution study of the final batch, that is the resveratrol nanoparticles from the capsule. Herein we can see that till 8 hours, not more than 10% of drug is released from the capsule, and after the 8 hours, there is significant release and increase in the release of the resveratrol nanoparticles from the capsule. We can estimate that our capsule is reaching the colon within 8 hours, and after reaching the colon, there is release of the resveratrol nanoparticles from the capsule. Compared to in vitro dissolution study of resveratrol free drug and resveratrol nanoparticles, herein we are able to see that the resveratrol free drug is not been able to release after the interval of 16 hours, whereas the resveratrol nanoparticles are released after 6 to 7 hours, that is reaching to the colon, and there is significant increase in the release of the resveratrol nanoparticles from the delayed release capsule. Release kinetics of resveratrol show that it follows zero order kinetics. Stability studies were also performed for all the batches, and we can interpret from the 1-1 data that the physical appearance was unchanged. Assay was 98.5 to 96%. Capsule integrity was unchanged, and after the 3 months, there is physical appearance change, like slight discoloration is observed. Assay 92.5 to 89%, and capsule integrity is slightly sticky. This may occur because of the poor storage condition, or because of not proper poor storage condition, or because of not proper storage. Conclusion: hyaluronic acid conjugated resveratrol loaded nanoparticles were successfully prepared and converted into a capsule dosage form. Nanoparticles solubility and stability enhancement from conventional drug delivery, polymer combination of Ubragit S100 and RS100 prevented premature and burst release of the drug. Hyaluronic acid conjugation, site-specific and site-selective action. Thank you.
Can you go to the last slide? Conclusion. Okay, how did you ensure the last bullet, site-specific and site-selective action? Did you do any studies to conclude like this?
No, ma'am. We have not done any in vivo studies, but based on the in vitro studies and based on the literature data, like hyaluronic acid has a site-specific affinity for the CD4 cells, which are overexpressed in colon cancer. The data has been
CD4 cells or CD44?
CD4 cells, ma'am.
CD4 cells are also expressed on macrophages
Yes, ma'am. But during colorectal cancer, like in colon cancer, they are overexpressed.
Why I'm asking, CD4 is a very common expression, right? The CD4 cells are expressed by many cells. So how is the formulation going to be site specific? You have your lymphocyte population which is expressing CD4,
Yes.
right? So how is the formulation going to decide ये वाला CD4 अच्छा है, ये वाला नहीं जाना है? Think about it, no problem.
Yeah.
Okay, and again, what is the mechanism of drug release in your conjugated resveratrol loaded nanoparticles?
It is zero order release, ma'am. The kinetics is zero order.
Is the conjugation physical, and it just what happens? How does it come out of the nanoparticle?
Ma'am, drug releases from the capsule, like the drug released from the capsule.
जो zero order बोल रहे हो वो तुम्हारा kinetic नहीं पूछ रहे हैं. What is the mechanism of drug release?
It was not a burst release. It gives a constant release throughout the period, so that it reaches the colon.
So the first conclusion, hyaluronic acid conjugated resveratrol. तो ये physical bonding है, chemical bonding है. तो जो भी bonding है वो release कैसा होता है उसमें से? That is what I'm asking. Is it just a physical admixture? What it is? That is what I was asking.
It was because of the physical process, like the conjugation process which we have done to conjugate the hyaluronic acid with the resveratrol. It was a physical electrostatic process.
How did you confirm that it is conjugated?
Based on the release data, ma'am, which we have shown as a conjugated release and unconjugated release distribution.
नहीं वो release पहले you did conjugation. So how did you ensure कि I have conjugated? Release you did later, right? So in that when you did this thing in your methodology, how did you ensure that you have conjugated HA to your resveratrol? So what is it? HA2 drug, HA2 resveratrol, HA resveratrol drug. What is it?
HA2 drug, like a drug was entrapped in the nanoparticles, and nanoparticles were conjugated with hyaluronic acid.
So so nanoparticle and hyaluronic acid conjugation क्या physical है कि chemical है?
Physical है, ma'am.
Physical है. Okay, go to the previous stability data. What do you think would be the proposed shelf life of this product that you have developed?
It may be around. It may go to 6 months, as per the data which I have got as a 3 month.
No, but it 3 months में ही it is showing at room temperature discoloration, stickiness. And what about the assay? Is it within the standards? The assay is 92.5%.
Yes, ma'am. The standard which were there is 89 to 110%. So it is within the limit at the room temperature. It was in exaggerated conditions. It goes beyond the.
This standard is for what? Which Indian pharmacopoeia?
No, ma'am. It is a herbal drug. Resveratrol is not mentioned in the Indian pharmacopoeia. The draft which is available.
No, I understood that. But when you say the standards are this percent to this percent, तो ये कहाँ से मिला information?
Ma'am, based on some of the literatures which are there, resveratrol पे जिस पे study हो गई, based on that data, I have concluded this statement.
So based on the literature you have concluded, the standards are those which are pharmacopoeia.
Okay.
Is resveratrol official in the Indian pharmacopoeia?
No, ma'am.
Just revisit on the stability and even the storage conditions, because I don't know, you will probably have to put a note on the storage conditions.
Okay.
The shelf life journal literature. What is the shelf?
Sorry, ma'am.
Adam, your voice is cracking.
What is the shelf life of your herbal part? Herbal component.
Resveratrol, ma'am.
Yes.
my resveratrol, it was around
Like regarding, there must be some literature regarding the same, no?
yes, ma'am. Hello.
Yes. Hello.
Can hello? Can you
So,
repeat? You ask
so
shelf life for resveratrol.
Yes, yes.
Okay, so ma'am, based on the literature data, the shelf life which I got to know it is around 1 years, 1 to 1.5 years. Shelf life for resveratrol.
okay. Okay. Thank you.
Any further questions now?
No, no, just visit the literature back because some Google search about resveratrol stability said that it is stable for 3 months at 4 degrees centigrade, which actually your data what you have got makes sense excepting assay. So
Okay,
your
ma'am, I'll
assay
look
has
into
gone
it.
down from 98.5 to 94.8 even at 5 degrees. Just
5 degrees
revisit, maybe
Celsius.
just do a recheck on this.
Okay, ma'am, thank you. Sure. Thank you.
Okay, that's all from my side.
Can we proceed?
Yes.
Yes, please.
Now I would call upon to MPH SRC 02.
Solubility and bioavailability enhancement of tadalafil, a comparative study of hydrophilic and lipophilic systems. Let's start with the background and rationale. Tadalafil is a potent, reversible, competitive inhibitor of phosphodiesterase 5, an enzyme that inactivates CGMP. Inhibition of PDE5 present in the smooth muscles of the penis leads to increase in the intracellular level of CGMP, subsequently resulting in penile erection. Tadalafil is also used to treat men who have signs and symptoms of benign prostatic hyperplasia or BPH. BPH is caused by an enlarged prostate. Men with BPH usually have difficulty in urinating and need to get up at night to urinate. Tadalafil will make these symptoms less severe and reduce the chance that prostate surgery will be needed. Tadalafil is also used in both men and women to treat the symptoms of pulmonary arterial hypertension to improve your ability to exercise. Moving on towards methods for enhancing the solubility and bioavailability. This study aims to enhance the solubility and bioavailability of poorly water-soluble tadalafil, hydrophilic beta-cyclodextrin inclusion complexes, and lipophilic self-emulsifying drug delivery systems were investigated and compared for their effectiveness. To capitalize on the rapid onset of action associated with orally disintegrating tablets, beta-CD tadalafil complexes and sets were further formulated as ODT. Starting with beta-cyclodextrin inclusion complexes. They are prepared by three methods: kneading, homogenization, and dry blending. These complexes are further converted and compacted into orally disintegrating tablets. Here we can see the disintegration time and the manufacturing difficulties faced during the preparation of different ODTs. The disintegration time is significantly less for tablets where beta-CD is made using the homogenization method. Also, no compression issues were observed. Moving towards the drug release study, here as well, we can see that the beta-CD complexes containing the tablets prepared by homogenization have better release rates. Moving on to the other enhancement method called as SEDS. SEDS or self-emulsifying drug delivery system is a lipophilic system that consists of an oil, surfactant, and a co-surfactant. Based on the self-emulsification time and percent transmittance, and also considering the IIG limits, here from the pseudo-ternary diagram, we can conclude that the 1:1 ratio of surfactant and co-surfactant was considered to be optimum for the SEDS formulation. The ideal S-mix to oil ratio was also identified. Avicel was used as an adsorbent for converting liquid sets into solid sets. Three different formulas of solid sets were prepared and compacted into ODTs. Here we can see that formula 3 shows less disintegration time. With the help of drug release studies, it was found that sets had better release rate compared to the available marketed tablets. Moving towards the in vivo studies, if we take a look at the in vivo studies done in Vista rats, it clearly shows that SEDS has higher AUC and Cmax and also achieve Tmax in lesser time. Moving towards the conclusion, dissolution studies demonstrated that superior drug release from both systems compared to the marketed product, with sets exhibiting the best performance. The lipophilic set systems outperform the hydrophilic beta-CD inclusion complex in augmenting the tadalafil bioavailability. In Vista rats, and in vivo kinetics revealed that sets resulted in higher AUC, Cmax, and faster Tmax, indicating enhanced bioavailability compared to both beta-CD complexes and the marketed products. These are the references. And thank you.
The benefit that you have seen for SEDS, is it a generalized benefit? Can I apply the same benefit across drug categories? Because you said it's better than the beta CD complex also. So are generally SEDS better for bioavailability via enhanced resolution?
Ma'am, most of the research studies that have been done using sets or any self-emulsifying like SMEDs or SNEDs, it's mostly done on BCS class 2 type of drugs in the literature.
Yeah, so this is in the context of that class of drugs only. But you did it for tadalafil, right? So can I just say it says it offers a superior bioavailability versus the need drug for class 2 drugs? Okay, how many any marketed products available which are based on SEDS technology?
Ma'am, marketed cyclosporin SMEDs have been present in the market.
And what kind of claim they carry? Superior bioavailability.
They just carry like better bioavailability in general. Hello.
Yeah, you are audible. What are the limitations of SEDS as a technology?
Limitations.
You could say there are no limitations. That's all right, but whatever it is.
I'll say like low drug loading capacity would be one limitation.
So what is the drug loading capacity?
In this case, for year, it's around
in your case.
Here we have prepared 10 mg. 10 mg is the dose of the drug.
So, but did you do a ceiling dose study, or you just stopped at 10 mg? Did you increase and see whether it was able to load or not, or did you stop at 10?
We did 10 like so as to compare with the marketed ones.
No, I understand. But when you do an optimization study, did you try to increase the loading just to see whether it is able to take it? Can you go to your bioavailability wala slide? Yeah, okay. Yeah, this one only.
Yes, ma'am.
So, did you also have a beta CD complex? You did it here in the laboratory. You did it in the laboratory, the beta CD, or you picked up the data?
No, ma'am. The beta CD complexes were converted into orally disintegrating tablets. Both were converted. The liquid sets were converted.
You did that. Sets ODT and BCD to ODT.
Yeah. So in Vista rats, sets ODT gave better AUC and faster Tmax and higher Cmax.
Okay, so what is this pure drug and what is this marketed drug?
Marketed drug is the marketed the available ones of.
No, is it drug or is it formulation? What is it?
Of marketed formulation.
Okay. Because I was wondering what is pure drug and what is it's a
Marketed
formulation.
drug is the formulation. Pure drug is the pure tadalafil.
Okay.
Okay. Just few questions from my side. What disintegration method you have used for the determination of
Sorry,
what
ma'am?
disintegration method? Which disintegration method you have used? Official disintegration method?
Yes, ma'am. 900 ml purified water at like 37 degrees Celsius.
But in the ODT, you are thinking about the mouth conditions, okay? You don't have 900 ml of fluid in your mouth. So how you are to justify your DT time? Or the movement is that rigorous as official method. Can you get my question?
Ma'am, could you please repeat that? Your voice is cracking a bit.
I wanted to ask the disintegration time method you have used. How you are going to correlate it with the in vivo conditions? ODT is orally disintegrating tablet.
Yes.
And your official method has 900 ml of liquid. Sorry, water.
Yeah,
And your it is
USP.
According to USP and IP both. So have you tested with the modified disintegration test?
Ma'am, in the like for disintegration, no. We have tested the in vitro diso in 0.5% SLS, which mimics the salivary conditions. But
SLS?
Not for disintegration.
Okay, how 0.5% SLS mimics salivary conditions?
We found it in the USPC official protocol.
So what is the pH of that solution? SLS? What is the pH of that SLS solution?
pH. Ma'am, I'm not aware of the accurate number, but it should be somewhere around 7. I'll let you know.
Okay.
Okay. Thank you.
Thank you.
Thank you. We would proceed with MPH SRC 030. Please turn the camera on.
Warm greetings, everyone. My title is Colon Targeted Bioadhesive Pellets of Curcumin and Cyclosporine for Improved Management of Inflammatory Bowel Disease. Ulcerative colitis is an inflammatory bowel disease that causes inflammation and ulcer in the colon and rectum. Cause of ulcerative colitis is unknown, but it can be due to stress, diet, and immune malfunction. Conventional dosage forms face several challenges, which are a lack of specificity and not guaranteed rate of drug release, and the reduced drug availability at site of action, and also systemic adverse effects. To overcome these limitations, we have designed bioadhesive pellets. Advantages of these pellets are as follows. They have a targeted drug release, slower transit through colon, and less adverse effects, improved therapeutic efficiency by localizing the drugs to the target site, and they also facilitate intimate contact of the formulation to affected site, and they can also modify the tissue permeability, which in turn will increase the contact time in the target region. Cyclosporine is a immunosuppressant agent, and curcumin is an anti-inflammatory agent. When given in combination, the dose of cyclosporine can be reduced, which will in turn cause less incidences of nephrotoxicity. And and since we are making bioadhesive pellets, there is a high localization of the drug at the target site. Curcumin inhibit the T cell proliferation induced by PHA, PMA, and CD28, whereas cyclosporine only inhibit the T cell proliferation induced by PHA. Therefore, curcumin can overcome the lacking of cyclosporine in blocking PMA and CD28. Hence, they act in a hence they give synergistic effect. Methods and materials. These are the list of the materials used, which are curcumin and cyclosporine as the active agents, carbocol 940 and hydroxypropyl cellulose as the bioadhesive agent, Pivdegid S100 as the pH-sensitive polymer, and AVCell PH as a diluent. Out of many batches taken, these 5 batches were having good properties. Out of these 5, also batch B3 showed optimal properties. 4% curcumin and 2% cyclosporine were used in the pellet core as API. NaCl was used as an anti-sticking agent, and the water was used as a binder. For making the bioadhesive pellets, first of all the excipients were sifted through 40 mesh separately and then mixed and blended for 10 minutes. The wet mass was obtained using water as the binder, and then this wet mass was fed into a 1-screw axial extruder, and the obtained extrudate was placed into a sterilizer, which was operating at 1,500 RPM. The pellets obtained were dried, and then the pellets with optimum size of around 1 to 1.2 nanometers, 1 to 1.2 millimeters, were then chosen for coating. The coating solution made up of 2% beta volume Pivdegid S100 in S100 solution in the solvent of IPA and acetone in the ratio of 1:1. Talc, triethyl acetate, and the sensed yellow color was there in the coating solution. For coating, fluidized bed dryer was used. Coating was done up to 5%, 10%, and 20% weight gain in pellets. Formulation batch 3 exhibited the best sphericity and highest yield compared with other batches, and also in the bioadhesion test and swelling index. Formulation batch 3 showed the best sphericity and highest yield compared with other batches, and batch 3 also showed maximum bioadhesion and swelling index, which can be seen in the given tables. So for bioadhesion testing, the ex vivo method was used, whereas the force required to displace the bioadhesive pellets from the red colon skin, red colon surface, was measured as the force of adhesion. The API content for curcumin and cyclosporine was found to be was found to be complying with the USP limits. SAM analysis showed a good spherical shaped pellet with smooth and intact surfaces. In vitro release study was an important part of this project because we want maximum amount of drug to be released at pH 7.4, which is rectum and colon. So to mimic the journey of the pellet from stomach to large intestine, we used different pH media for different interval of time. Like we used 1.2 pH media for first 2 hours, and then 6.8 pH media for next 4 hours, and then 7.4 pH for pH buffer until 24 hours to mimic the conditions of colon and rectum. The release profile shows that the most amount of drug was released after 6 hours, which means the drug was released after it passed through small intestine and went into the large intestine. That is a rectum and colon, and both the drugs followed zero order release kinetics. At the end of 24 hours, 76% cyclosporine and 71% curcumin was released. To confirm the efficacy of the prepared pellets, in vivo studies were done. Animals were evaluated for the change in their body weight and also the histological examination of the red colon. Animals were divided into different groups. So for change in body weight, we can very well see the the test formulation group. The change in body weight of the test formulation group is much better than all the other uncontrolled groups. Histopathological studies were done to confirm the absence of inflammation at the epithelial layer. So we can see that test formulation control at 10x and 40x shows no signs of inflammation. In the conclusion, the performed studies shows that optimized type and concentration of polymer improve the sphericity and impart uniform size distribution. It was observed that carbopol 940 in combination with hydroxypropyl cellulose in the ratio of 1:1 shows maximum bioadhesion as compared to other ratio of the polymers. Further, in vivo studies helped to confirm the efficacy of the optimized formulation for IBD management. Therefore, bioadhesive pellets containing cyclosporine and curcumin in combination can be used for the management of inflammatory bowel diseases. These are the key references. Thank you.
Okay, can you go to the conclusion slide? Which polymer is in published literature, which polymer has shown the best bioadhesion properties?
Different polymers used for bioadhesion are carbopol. Then also pectin is used, and xanthan gum, etc.
That is there. Which has the best? You've selected some. There must be a reported that there are 50 polymers which have got bioadhesion. Out of them, which is reported to be the best? Is carbopol that one, or any other polymer?
Then the best one, like which I have seen in the literature, these are xanthan gums or carbopol are the most used.
Are the most bioadhesive polymers?
Mostly used in different research.
Okay, go to your release profile.
HPMC is also used like in various literatures.
Yeah, this one. So if you see over here, drug content, is this formulation official? You say hence it complies with the USP pharmacopoeial limit of 90 to 110%.
Yes, ma'am. The limit is for the API in normal tablets, like in the oral dosage tablets of cyclosporine and curcumin, and not for bioadhesive tablets. tablets. Like it is not official as such. The combination is not official.
So this drug content that you have reported and the limit, is it of the same thing? Is it same?
Yes, ma'am. The content is for the tablets which we have prepared, and the limit is for the oral tablets which are available in the market, which are not bioadhesive, basically. Normal oral tablets of cyclosporine and curcumin.
You have curcumin and cyclosporine tablets official in the USP. Do you have curcumin and cyclosporine tablet official in the USP?
Ma'am, not as combination, no.
अरे तो तुमने ही लिखा है ना, hence it complies with the limit. That is why I'm asking.
Yes, ma'am, it complies with the individual limits of both the drugs, right?
So it is official in the USP. That is what I'm asking.
Yes, ma'am. Since like the limit was given for 1900.
Okay, okay. ठीक है. 1 second. Yes, madam. Go ahead.
Have you done the stability study of this preparation?
Yes, ma'am. I have done. Due to time constraint, I was not able to put in this PPT, but I have done.
But do you have some insight into it? Can you share?
Ma'am, can I share my PPT or should I?
No, just explain. It's okay. It's okay. You don't have PPT.
So ma'am, stability studies were done like for according to the Q1A R2 guidelines. So for like they showed good stability with the combination showed stability over for a period of 6 months.
So you have curcumin, you have purchased the curcumin from the market, curcumin, or you have isolated it from the source?
No, ma'am. It was a gift sample from I guess Autochemy. Autochemy gave us a gift sample.
So curcumin is having anti-inflammatory, but how you decided the dose? What should be, how much amount is to be put into the tablet and how much it is effective? Have you tried different combination of curcumin and cyclosporine?
Then basically like for the management of IBD, curcumin is used like in many papers they have used 2% to 4% concentration of curcumin. So this combination is novel, curcumin plus cyclosporine, but they have been used individually with this concentration 2% to 4% individually. So that's why we have used.
Okay. Yes, just I wanted to know, because you have given in your objective that it will lower down the dose of the cyclosporine.
Cyclosporine.
Yes. So from what data I have seen, there is no proof of that. It is lowering the requirement of cyclosporine.
When we are using 4% cyclosporine, which is like if we give 200 mg of pellets in capsule, so like only 8 milligram of drug is going into patient. So that is a quite drop in the dose of cyclosporine as compared to other oral formulations or IV formulations.
Have you tried with the regular dose, and when you reduced the dose with cyclosporine, the result are same?
No, ma'am. They've tried in the range of 1 to 4% only.
Thank you very much.
Thank you.
Thank you. Here we come to the end of post-graduation oral presentation program. It is indeed an honor and a privilege for me on behalf of the organization team to extend my heartfelt gratitude on this momentous occasion of Student Research Congress 2023. First and foremost, our sincere appreciation goes to our distinguished judges, Dr. Garima ma'am and Dr. Tabassum ma'am. Your presence has not only added prestige to this event, but has also been a source of inspiration for all of us. We are truly grateful for your valuable time, commitment, and for sharing your immense knowledge with us. Your insightful evaluation will undoubtedly shape the future of research endeavors present here. Thank you to our participants for your enthusiasm and commitment to advancing research. Your contributions have made this event richer, more diverse, and intellectually rewarding. Last but not the least, a token of appreciation and gratitude to our esteemed judges, Dr. Garima ma'am and Dr. Tabassum ma'am. We present this small gesture as a symbol of our immense respect and appreciation for your valuable contribution to the success of Student Research Congress 2023. I request the honorable judges to share few words.
Garima madam, over to you. अच्छा मेरे को
Thank you very much. It's a pleasure to be part of this Congress. Actually, I'm very much impressed with some of the works that has been done. And I can see the students have put on a lot of efforts doing their research work. And it is always a pleasure. Actually, Munira knows best because contacting and having an interaction with students and having the newer current point of view, how the things are going on is always a pleasure for all of us. So thank you very much for Dr. Muni, especially Dr. Munira, for giving me this opportunity because I think we share the passion for the research. So thank you once again. Thank you very much, and thank you all the participants, and having and best wishes for continuing with the good work. Only one suggestion to all of you: please try to look for the best and try to make your products commercially viable, because it is all, and solve the problems. Which are solving the problems is a major need. Another thing is whenever you are using a combination of drugs, please check your IPs, because right now you have a very distinct you have to give a very strong supporting evidence that it is viable and it is relevant. If you want to get it approved, any combination you are going to get it approved. So once again, thanks everyone. Thanks Dr. Munira as well as Tabassum ma'am for supporting us, and hope we will get more opportunity to interact with you. And again, once again, thank you. It's a very well organized, very well coordinated event. Thank you very much.
Thank you, Dr. Garima. It was pleasure seeing you online. It's been a long time we have not met. So thank you. I know you spent almost whole day. I really appreciate your time. Thank you very much. Over to you, Tabassum madam.
Basically, I would just reinforce what Garima madam just mentioned. It was a nice collection of projects, presented also well, and some of the comments which Garima madam made, I made, see that you ponder over it, and if you can integrate into your research, it will strengthen it further. But otherwise, I think we had a good battery of projects which were shared today. And they were also diverse, majorly driven by formulation development. We had medchem projects, we had pharmacology projects exploring nice combination in epilepsy. So that was nice. I think we had a good blend of projects that were presented today. So well done to all the participants.
Thank you, ma'am, for your words of wisdom. The much anticipated results will be revealed tomorrow during the valedictory function, scheduled to begin at 12 noon. We understand your eagerness to discover your achievements, and we extend an invitation to join us in the celebratory event where we will acknowledge and applaud the outstanding efforts showcased by and throughout the Congress. To make it convenient for you, the link of access have been mailed to you. Please check that you have the necessary details. We are eagerly anticipated virtual gathering with you tomorrow. Thank you once again for your active participations. We would start with IIC program from 4. Thank you so much.
So do we have to leave the meeting?
Yes, ma'am.
If you wish to listen to IIC, you can stay back.
No, I thought we were supposed to discuss the results.
Talk to Garima madam and discuss.
Yeah. Okay. Chalo. I'm leaving the meeting. Thank you.
So this is the same link for IIC? I mean to say.
Yes, ma'am. Yes,
Innovation. Okay.
Yes, ma'am.
Okay. Innovative ideas.
Yes, ma'am.
Okay. Okay. Hello, Pooja. Good evening.
Good evening. How are you, ma'am?
Thanks. I'm good. Thanks for your time. I know you're busy, super busy.
Yeah, no problem.
But your insightful observation and comments would definitely help into innovative ideas, because it's ultimately what we want to see is that innovation aspect. And I'm sure your keen observation and comments will definitely help students for their future programs and future projects.
Yeah, sure.
Good.
So who's starting with first?
Uh, so my team will give all details. They will introduce you. Yeah. So here you have.
Yes, ma'am, we'll begin soon.
All right, yeah.
Good afternoon, everyone. I, Rachna Patil, welcome you all to the second half of 4th Student Research Congress themed Innovations for Better Health, organized by SVKM's Dr. Bhanuben Nanavati College of Pharmacy, co-hosted by University of Mumbai, industry partner ACG Worldwide, and SciTech Center. In the first half, we have gone through postgraduate oral presentations, and now we move on to Innovative Idea Challenge IIC. Innovation is to introduce creative, problem solving, and application based ideas for efficient progress. This session will hopefully be an inspirational and learning experience for everyone. Now, I present you all with the judges of IIC. First up, we have Miss Pooja Shah ma'am. Miss Pooja Shah ma'am is Distinguished Panel Member of Institute Innovation Council IIC of SVKM's Dr. Bhanuben Nanavati College of Pharmacy for academic year 2022-23, where she mentors the college on innovation and entrepreneurship excellence. Ma'am is a registered patent and trademark attorney for India and is practicing IP for almost a decade now. She is an experienced IPR attorney with a history of working in law firms and corporates and is immensely skilled in patent prosecution, patent drafting, infringement analysis, FTO, legal research, legal writing, patent law and prior art search, trademark filings and prosecution, design filings and prosecution, copyright protections, and contract drafting, filing suits in the Bombay High Court related to IPR cases. After completing her graduation from BNCP in 2013, she went to pursue LLB from Jitendra Chauhan College of Law and Diploma in Patent Law and Practice from Institute of Intellectual Property Studies, NMIMS. After gaining her good experience in the field of IPR, she set her foot in the journey of entrepreneurship and ventured with her own law firm, Artemis Law Associates, in December 2020. Ma'am was featured in Woman Entrepreneur magazine August 2021 issue and has also conducted workshops in various educational institutes about IPR awareness. A very warm welcome, ma'am. Thank you for gracing us with your presence.
Thank you so much. Thank you for your kind words and nice introduction.
Thank you, ma'am.
Next up, we have Dr. Sujata Savarkar ma'am, Professor and Head of Pharmaceutics Department at SVKM's Dr. Bhanuben Nanavati College of Pharmacy. Ma'am was associated with pharmaceutical companies in India at managerial positions in the field of research and development, formulation development of conventional and novel dosage forms for regulated and domestic markets. Ma'am has been executive member of Control Release Society India chapter since 2014 and secretary of CRS IC for 2023-25. Ma'am has received research project grants worth about 1.5 crore rupees from industry and government funding agencies. She has presented about 50 research papers in national and international conferences like CRS India chapter and AAPS. Ma'am has been awarded with IDMA Award for Reviewer Research and Industry Outcome by SVKM's Dr. Bhanuben Nanavati College of Pharmacy in 2018. Ma'am has received travel grant from CRS and AICTE for attending and presenting papers. Extending a warm welcome, ma'am. Thank you for gracing us with your presence.
Let me express my heartfelt thanks to both of you. Pooja being into corporate, her own practice, IPR practice, in spite of her busy schedule, she is with us. And it shows that her dedication to prepare the next generation for innovative thinking. And I'm sure students, those who are presenting today, innovative ideas, Pooja madam herself has her own IPR firm, and she would be giving more insightful, valuable observations and comments on your project, which is going to remain with you lifetime. At the same time, the new project, when you work on, you will be definitely be able to incorporate all those suggestions and observation. So it's a kind of a lifelong learning. Thanks to Dr. Sujata madam, in spite of being lot of academic and other activities, she is going to judge all the innovative ideas, challenge along with Dr. Ms. Pooja Shah. And Sujata madam herself has patents granted. So I'm sure her keen observation will help all the participants to improve upon, or it would be a kind of a lifelong learning for all the participants. Thank you very much, both of you.
Madam, for inviting
Thank you,
us.
ma'am.
Thank you so much for your kind introduction, Rachna. Thank you. In fact, we look forward for a lot of interaction, new ideas coming from you. And first day, it will be specifically since we get to see a couple of ideas generated, it will be a good learning experience even for us as well. So thank you for having us over here. Thank you.
And also, I would like to just, just give a brief introduction about what aspects that I am looking here per se. Whenever you explain your innovation, I would like to understand from two perspectives: what is novel about the invention and what is inventive about it. Novel means with respect to whole world, how is it novel? If it is somewhere published already, what modifications have you made so as to make it inventive? So on whenever you're presenting any idea, I request all of them to please throw some light on the novelty and the inventive step aspect, so that we can discuss more about it and kind of ascertain whether it is patentable or not. If it is not right now, what modifications you can do it, so that in future if you want to apply for a patent, you can do that.
Yes, ma'am. Thank you for your suggestions. Now we'll begin with the presentations. Before we begin with the session, I would like to request all the participants to make note of the guidelines for IIC. Presenters are requested to rename themselves as per the allotted codes if not already done. Presenters cannot reveal their names or any affiliations. Each presenter is requested to stay online, switch on the camera when their code is called out. Pre-recorded video will be presented on the screen. Only one author is allowed to answer for the Q&A session. Hope all the participants have thoroughly reviewed the instructions. Also, I request the judges to confirm that they have received their scorecard.
Yes, I do have.
Thank you, ma'am. Now, I would like to call upon IIC 001. Your video will be shared shortly. IIC 001, are you there?
What we are seeing is SRC 002.
Okay, ma'am. There was a mistake. ISC SRC 001 will be presenting at the end. ISC SRC 002 will be presenting at the beginning. ISC SRC 002, are you present?
Yeah, I'm present, and my team members will be joining.
Yes, please switch on the camera.
Hello, our participation number is IICSRC002 and our idea is making a prebiotic using agricultural food waste for promoting growth of Faecalibacterium prosnitzi in patients with inflammatory GI diseases. Starting with the graphical abstract, we will be extracting inulin from food waste to make a prebiotic that supports the growth of Faecalibacterium prosnitzi, which is already present in our gut microbiota, to treat inflammatory bowel disease and improve the overall gut health. Background. So Faecalibacterium prosnitzi is a bacteria that is an abundant and important bacteria that is already present in our gut microbiota, and it ferments dietary fiber to produce butyrate and other short-chain fatty acids which have an anti-inflammatory activity. Keeping this activity in mind, it can be used to treat inflammatory bowel disease, a disorder characterized by chronic inflammation of the GI tract, with Crohn's disease and ulcerative colitis being the principal types. Now, the delicate symbiotic host-gut microbiota relationship appears to be lost in IBD, which can be restored using the prebiotic. India wastes 30 to 40% of the vegetable and fruit produce every year, and this waste contains prebiotic compounds like inulin, fructose, glucose, and acetic acid, which can be used as a substrate for the growth of F. prosnitzi. Another benefit of recycling food and agricultural waste is it offers various environmental, economic, and social benefits, such as improving the soil quality, reducing the emission of greenhouse gases, conservation of water, landfill reduction, and it reduces the overall pollution as well.
Hi, so I'll be talking about the current market size for the probiotic and prebiotic market in India. So the Indian probiotics market is expected to exhibit a compound annual growth rate (CAGR) of 20.1% during 2023 to 2028. This shows that there's a very big scope for our proposed product or idea to be sold in the Indian market. The prebiotic market as well was valued at rupees 21.5 billion in the year 2021. This goes to show how actually big this market is and how there's a great opportunity for people who are developing probiotics and prebiotics such as our idea to get our products in the market. Factors affecting this market, this is growing prevalence of infections. People are getting aware about benefits of using pre and probiotics. People are being aware about their gut health. There's a lot of social media awareness spread amongst the youth, especially. Availability of probiotics through online and offline distribution channels as well. The market is expected to grow again due to consumer awareness in the aspect of health, as I mentioned. Recent trends have also caused the rising consumption of pre and probiotics. So there's a really big trend going on currently about the awareness for gut health for people. Social media and other sources have spread the benefit of good gut health, and this pushes the masses towards buying such products. So we are targeting our product to be sold as a powder or a tablet. And existing inulin prebiotics in India are priced at an average of rupees 3.16 rupees per gram. So our product is targeted to be lesser than the average cost, and it would be environmentally sustainable as well, which could again be a good selling point.
So after all of that, we come to the methods. So first is to extract the inulin from the agricultural waste. So here we've taken apple waste, which is first ground into a fine size, and then it undergoes an inulin extraction method, which is hot water extraction. So the product of this extraction is filtered twice. This is to separate the liquid that contains the inulin and the solid waste. So after this, the filtrate is centrifuged to to separate the aniline from compounds and other proteins. And after that, the layer which contains the aniline is further purified by HPLC, and its pH can be adjusted and it can be checked with the pH meter. So after the extraction of aniline, how effective is it to promote the growth of F progeny? So here F progeny is normally grown in a YCFAG broth, which is yeast extract, acetone, fatty acids, and glucose. So now here, instead of glucose, we would use aniline as a source of carbohydrate. So that becomes a YCFA agar, and the concentration of aniline would be changed to see which is the best concentration of aniline that promotes the maximum growth of F progeny. And the F progeny, since it's an anaerobic bacteria, it would be grown under in an anaerobic chamber, and every 2 hours for 24 hours, the CFU per ml is checked. So now F progeny, as it grows, it produces metabolites like SCFAs, which are short chain fatty acids. So that can be quantified in the YCFAI agar itself. So the agar is sampled over time, and then it is internal standards are added to that sample, and HCl is added, and then that is mixed and incubated. After that, diethyl ether is added, and it is centrifuged. So now, because SCFAs are soluble in the diethyl ether, they are extracted in the organic phase, and after that NaOH is added, and then it is centrifuged again. So now, because of the NaOH, the SCFAs move to the aqueous phase, from the organic to the aqueous phase, and then this SCFAs, which are now in the aqueous phase, can be used to run in the HPLC, and that way SCFAs are quantified. Now, apart from that, what is the effect of aniline on the growth of intestinal cell lines? This can be checked by a Hoxband system, which is human oxygen and bacteria anaerobic system, wherein bacteria and intestinal cell lines are grown at the same time. So here it is basically a falcon tube in which there is YCFAI 1% semi-solid agar, which is inoculated with F progeny. And on top of that, an animal medium, an animal growth media is added, which is DMEM. And on top of that, the CaCO2 cells, which are intestinal cell lines, is added on top of that. And then all of that is incubated for 18 hours at 37 degrees Celsius. So after that, how do we analyze the results from our methods? Is first of all is to quantify the aniline that is extracted from the apple waste that we have collected, and that can be done by HPLC. After that, to check which concentration of aniline promotes the highest growth of F progeny has to be checked. So for that, a line graph can be plotted of CFU per ml over time for different concentrations of aniline, and the differences in this CFU per ml can be checked by ANOVA. Apart from that, the amount of SCFA that is produced by F progeny is also quantified, and it can be plotted over time. And so that way we can choose which concentration of aniline promotes the highest, promotes maximum growth of F progeny and maximum production of SCFAs. Then to check the effect of aniline, the extracted aniline, on the intestinal cell lines, two things can be done. First is MTT assay, which is to check the cell viability of the CaCO2 cells. And apart from that, an ELISA can be done to check the expression of anti-inflammatory markers. This becomes important because we are aiming to, that this prebiotic helps alleviate inflammation in IBD patients. So checking the anti-inflammatory expression in CaCO2 cell lines after the administration of aniline is an important criteria. So to conclude, apple waste, it contains aniline, which can be extracted by hot water extraction method. And this aniline can be used to promote the growth of F progeny. And this F progeny produces various metabolites like SCFAs that can help promote anti-inflammation in the intestinal cells. Here are our references, and thank you.
Thank you. I request judges to begin with Q&A session.
So I have a question to begin with. Any of you can answer actually. So coming to the cost perspective, you said that currently 3 point something rupees per gram or ml is the cost. How is your product priced? What will be the costing of the whole process and extraction and the final product? I hope my question is clear.
Hello.
ISSC 002, please switch on your camera and respond. ISC 002, are you there?
Sorry, my network is dropping. I'm not able to switch on my camera. Will that be fine?
Yes, it will be fine.
Yeah, so I took an analysis of some of the current products, 10 popular gut health products on social media sites as well as Amazon. And I got an average of rupees 3.16 per gram. But in our case, since we are planning to use food waste, as Harsh mentioned, it was the apple waste, onion waste. So our projections would be around rupees 2 per gram to start with, and annually accounting for inflation, it could go up to rupees 2.50 per gram. And we would plan to sell it as, or like according to our idea, it would be capsules or a powder, which would be around 100 grams as a serving or like in a packet. And yeah, so per tablespoon would be the serving size. And that would account to around rupees 10 per serving for a person, which I think is pretty good compared to the other products on the market.
So I think the other products out in the market, I think Yakult is one of them. And I've read the label of Yakult. It has a lot of sugar. So the currently existing. This is from the innovation perspective I'm talking. So currently, whatever product that are existing has a lot of sugar, and I think as a health conscious person, I will not have something which is having so much of sugar, which brings down my immunity. If you must have read the research about sugar, it brings down the immunity. So how are you drinking Yakult or any other probiotic or prebiotic to increase your immunity or gut health?
Exactly. I think most people, since are not aware of the amount of sugars and the harm this does, that's why we were targeting our innovative product to be had in the form of a tablet or like a powder, which would eliminate the requirement of adding sugar to it to make it taste good.
So
because the purpose of sugar for Yakult as a drink is to make it appealing to kids or even adults, and they add the sugar so that it tastes good. But if like the serving is in the form of a capsule, say, or a powder, then the need for adding sugar would be completely eliminated.
Yeah, so I think this is one kind of innovation that I see, it can be out booming in the market because whatever existing prebiotics or probiotics are there, it is actually more harmful than what they're claiming to be. So I think your product, if it is commercialized in the right sense, it could bring some value.
Thank you.
Over to Sujata ma'am, actually, I'm done with my questions.
Okay. Have you done any literature search for this, or have you done these experiments in your lab? Or it is just a concept that you are presenting?
So we haven't performed the experiments. It is just a concept.
But I see a couple of papers published in this particular domain where they already have used apple pomace to make prebiotic. Okay.
Yes, they have extracted inulin in a lot of papers. Yes.
Correct, because I'm just seeing in front of me one of the paper which states that apple pomace has been used for prebiotic potential. One paper of food hydrocolloids.
Yeah. Yes.
So in this regard, how new is your technology?
So for us, we are specifically extracting inulin from the apple pomace, and so that would help to achieve the growth of a specific microorganism, that is F. prosnitzi. So our purpose for that is specifically to alleviate inflammation in IBD patients. So along with that, apart from inulin, we can also extract acetic acid and other fructooligosaccharides, which also help promote the growth of this microorganism. So the papers that are already there, they are more of a proof of concept of how a prebiotic can be extracted. But what we are showing is more of an application approach of how it can help to alleviate a specific illness, like IBD.
Can you go to the manufacturing process, please? The slide showing manufacturing process where you have mentioned. Okay. So here, how are you going to achieve purification by HPLC?
So the HPLC would be more of an analytical technique of to check out the percentage purity. We won't exactly separate it with HPLC.
So then what would be the final form of your product?
The final product would be a solid.
So how are you going to convert that into a solid dosage form?
Okay, so after the fourth step, that is filtration, we would centrifuge out the inulin from the filtrate. So inulin would come out as a solid.
And say inulin is quite unstable. So how are you going to stabilize that?
Okay, so inulin can be stabilized with a lot of salts. For ammonium salts can be used to stabilize it, and also we can associate with other. Since it's a capsule, we can add other compounds which would stabilize it.
What would be your marketed pack?
Sorry, marketed?
Pack. How are you going to project it into market?
Okay. Maitri, do you want to answer?
Yeah. Could you repeat your question? I'm sorry, my network dropped.
So my question was, how are you going to present it to the market? What will be your pack?
So as I mentioned already, it would either be capsules or powders, and we would plan to have really attractive packaging to, appeal it to the kids especially. And our competition mainly would be products like Yakult. So I think our selling point would be, first of all, the cost, which would be lesser, and the amount of sugar. Like there will be perpetually zero sugar. These could be our USPs, which would help us crack through the market. And I feel like it would be, a really good selling point, even with big players like Yakult in the market.
So that case, if it is your target population is kids, what about palatability?
Yeah. So since it would be in the form of a capsule, the palatability should not be an issue, because it would just be one capsule or half a tablespoon of a powder to have per day.
Thank you.
Yeah.
Thank you. We'll proceed with IIC SRC 001. Judges, please make note we are presenting video of IIC SRC 001. IIC SRC 001, please switch on your video. IIC SRC 001, are you there?
Yes, I'm here.
Okay.
Currently working from some lovely professional university. I have submitted my abstract in the section of innovative ideas. My ideal topic is bridging the healthcare gap by introducing Umid Ken in rural areas. So in this PPT, we will talk about the what is
Can you be a little more louder, please?
introduction part and how it is going to implement of Umid and what is that gap
Ma'am, it's a pre-recorded video.
Okay.
Hello, ma'am.
I want to fill from my ID.
Yes.
I can explain idea here if it is not audible there.
No, it is fine. Wish, I request the judges to make the call whether he can explain it right now or the video recording should be played.
Okay, video recording is okay. No problem.
Okay, ma'am. Thank you.
Yeah, between the rural healthcare and the professionals, or which facility I'm going to provide through the ideas. In the outline, in this deputy, we will discuss some introduction part, implementation of Omid Kendra, and what is the benefit of Omid Kendra and future plans and its sustainability. So let's start. First, before moving ahead, we need to ask one question. What is one key challenge faced by the rural communities in accessing healthcare? As we know, more than Indian population, like more than 60 or 70% Indian population are living in rural areas where healthcare facilities are not as much as good as in urban areas. If I talk about myself, because I belong to village or some rural areas, so for me to access a doctor which are a good doctor, I mean to say, like having a good experience or good knowledge about the healthcare or about the diagnostic purpose. So if I want to meet a doctor, I have to travel 60 to 70 kilometer on a single way, and I have to return other 60 to 70 kilometer. Means I mean to say, if I want to visit to a doctor to for any further disease, I will waste my whole day. First, I will waste my money. It is not a waste, but I mean to say it will waste my time in the term of my days or in the term of my money also. So what happens for the rural healthcare in India? What is the scenario right now? Majority of the Indian population which are residing in rural areas, which we know, but still, there is a lack of accessibility to educated healthcare professionals in these areas. Why it is happening? There are two major reasons. First, doctors don't want to come because they want to prefer to reside or live in a cities or urban areas. They don't want to waste their time. They don't want to waste their time on traveling. So if I already told you my house is like 60 to 70 kilometers far from having a good doctor, so if doctor will come on a daily basis, it is like he will waste, he or she may be waste his 2 to 3 hours on a traveling basis. So this is a reason. And second one, in rural areas, there is a no any kind of infrastructure where doctor can practice. And in terms of infrastructure, they don't have medical support. There is no such kind of diagnostic machines or diagnostic healthcare support is available. So they prefer to the in cities. So I want to fill through this gap through my ideas. In introduction part, we will discuss about the overview of the healthcare gap as we have already discussed. Challenges faced by the rural population in accessing healthcare. We have completed what is the importance of bridging the healthcare, introduction to Umid Kendra and its objective, and the Umid Kendra model. Okay, so now move forward. What is Umid Kendra? As I told you so many times, Umid Kendra, but what is actually it is? So basically, that as defined as Umid Kendra is a proposed solution to bridge the healthcare gaps in rural areas. It utilizes technology to connect patients with doctors and aims to provide quality healthcare in these undeserved areas. So basically, I want to clear here, we are not going to do so many stuff. We will simply provide doctors to the patient without having the physical presence of doctor in the rural areas. We will provide healthcare support to the patient through the video conferencing, like doctors, whether he is in cities or some other areas he is living. There is no need to doctor to come to my clinic. He will provide assistance through the video conferencing to the patient. I want to open a meet center where I will provide these facilities. I will provide a desktop or I will provide a TV section where I will install these facilities. Even I will hire the paramedical staff like pharmacists, like nurses. So whatever the doctor will give the suggestion through video conferencing, we will provide that facility. Suppose doctor has prescribed some medicine, then I am the person who will organize the medicine facilities there. So whatever the doctor will prescribe the medicine, there will be the qualified pharmacist who will dispense the medicine to the patient. And if a doctor prescribe any injection, any IVs, any other thing, then there are like paramedical staff, nurses will be there who will provide the support to the patient in these problems. So what is the model of Umid Kendra? So Umid Kendra is just a simple model. Under the Umid Kendra model, doctor which are dedicated doctor will take 1 to 2 hours on a daily basis through video consultation with patient. On-site pharmacists provide prescribed medicine and paramedical staff are available for emergency services. Like in any situation, doctor prescribe any kind of physiotherapy facility. We will hire a physiotherapist. It is not difficult to hire paramedical staff as much as difficult for the doctors. So it is our model how we are going to implement these Umid Kendra. First, we will utilize the technology. We will set up a Wi-Fi or a strong internet connection there and utilize our technological spec so that there will be smooth work should conduct between the doctor and the patient. Video conferencing for the remote consultation with the doctors. Okay. Provision of prescribed medicine through the local pharmacy. As I told, we will have a piece of qualified pharmacists which will dispense the medicine there. And in any further case, we need to have paramedical staff. We will have paramedical staff like nurses, like physiotherapists, or some other medical staff. So, what is the benefit of Omicron? As I told you, the gap. So, these are the benefits. First benefit is improved accessibility to healthcare for the rural population. Rural population now can access to a doctor which are qualified doctor. Let me tell you one incident. Some incidents taken place in my village. What some people has taken the medicine from the medical store and he has suffered a severe allergic response after taking the medical because his body is not adapted to that medicine and a severe allergic response has taken place and in that case we have to take that person to the city or shifted because the medicine which were given by the medical store or medical staff, they are not a well pharmacist or a good pharmacist. They even don't know the disease condition and they have given whatever they know, like 2 to 3 medicine, and they will give only that kind of medicine. So first, we need to improve our accessibility to healthcare for the rural population. Second, reduction in travel time and cost for the patient. It is so easy, right? We can reduce the travel time in this way and cost for the patient. Cost for the patient in the such way that doctor whom they are providing through video consultation, there may be the chance that they will charge less or we will provide that platform where the doctor will charge less. So third one is timely medical assistance during emergencies. So suppose any kind of emergency has taken place in that areas and the time there is no transportation available during the night. So we can provide emergency services through by just by taking the consent from the doctor or the advice from the doctor, we can further go through any kind of emergency services. And the fourth point, which is enhanced trust and patient-provider relationship through the physical visit. It is not like that only the doctor will provide the video consultation. Uh, we need to improve the relationship or belief, trust between the doctors and the patient. So we will call the doctors to physically visit at least 1 day in a week so that the trust can enhance because what is happening the patient will think that they are not the good doctor, they are just even patient can think they are not doctors, suppose they are some other person or just they are manipulating us, so we will call the doctor physically so trust can enhance. Let's move next PPT. What is our future plan at sustainability? First of all, our future plan, we want to expand the reach of Umidkan to more rural areas. More and more rural areas, we will establish this Umidkan, collaborating with more doctors and healthcare professionals, continuous training and development of staff for quality care. Whatever the staff we will hire, we will provide them a few months training, and then after training, we will provide the quality of healthcare support to the patient. The fourth point is ensuring long-term sustainability of Umid Care through partnership and funding opportunities. Whatever the funding we will receive, we will expand our future plans. So that's all from my side. Thank you so much.
Session is open for question answer round.
Pooja madam?
Yeah. So my question is, so who will be doing these payments? Basically, directly from the patient, it will be going to the doctors, or it will be like some kind of application on which you first pay the payment and then you get a consultation, or how is it gonna work? How is it gonna function?
Ma'am, is it like a physical existence having without the doctor is there? Suppose as the patient is visiting in a hospital physically, so patient will be there, but doctor will be through providing through the video consultancy. Apart from doctor, we will provide every facility there related to medical support, even in physiotherapies or some other. We will provide that kind of services there. But what is happening now in our areas, doctor don't want to prefer to come there because of they don't want to waste their time on the traveling basis. So it is like, and for us also, if we want to go to meet the doctor, it will take our whole day. It is like if we are providing there every services except the doctors, and doctors will be provided through the video consultation because it is easier to hire the paramedical staff rather than a doctor.
So the payments will be done to the hospital directly.
Yeah, and we will make tie-up with the doctor. Whatever the payment is required, the doctor we will pay there, but it is to us payment.
So what do you get out of it?
What, ma'am?
What do you benefit out of it?
Ma'am, my benefit is there. It is not only on a whatever the margin is there on a medicine or other medical facilities which were normally running in the hospitals. That benefit we will get there, and we will provide whatever the benefit we are getting to the doctors also. Like simply, this is a simple basis hospital which are working in urban cities. We want to implement this in the rural cities, having without the presence of the physical doctor.
No, this model of yours, business model, I want to ask you if you are rolling it out in the market, what do you get out of it?
Like it is the similar, ma'am. Just tell you, like whatever the thing is implementing in the hospitals, we will get the profit from that one also, like from medicine, by selling the medicine, and even by giving the IVs or some medical services to the patient, like laboratory facilities or others. We will make a margin to that one also.
Okay, so I understood. Okay, from those things you can get your commercial value. Also, have you thought of any? I mean, have you heard about this model earlier? Have you taken inspiration from somewhere, or is it your own
Yeah, it is.
creation of this idea?
Right now, I didn't had any model which is working on this model. I realized this problem to my areas. Then I proposed this idea. I don't know. I have not searched thoroughly, but I didn't had any ideas which is working in this way.
Okay, over to you, Sujata ma'am.
Okay, so then what it would be like? Would it be like an app or what? What are you proposing to do? Because I'm
Ma'am,
still, yeah.
it is not necessary to have app, because now we know that there are so many apps available. People uh, don't want to install so many apps in their phone. It is basically, we will provide that facility there. It is a simple video call, ma'am. Whatever the patient will visit in the hospital, in the whatever we have named that center, Umeed Kendra, the patient will be visit there, and we will provide to the doctor through the video consultation. It is a simple way. Even the doctor doesn't need any kind of a special facilities or to install any app or to work in their any special platform. They need only phone to start consulting anywhere. They will just prescribe. They will just prescribe the medicine, send us to the WhatsApp or anywhere. We will dispense the medicine. We have the pharmacy or every setup there.
That case, because this concept of video consultation is already there. Yeah.
Ma'am, but
There is
yeah, you are right. It is already there, but it is
already
not
there.
having.
So, what is the novelty in your idea? The only noble cause that you're doing is you are catering to the rural area, which is good. But in that case, what is the novelty in your concept?
Ma'am, whatever the other which are providing through the video consultancy, but at the same time they are not providing the services like providing the medical medicine. I know some of the apps which are providing the video consultation and they are sending medicine after 2 to 3 days later. But whatever the doctor will prescribe the medicine, we will provide the medicine at the same time. During even the emergency condition, we will check the BP of the patient. We will check the and inform the doctor the BP is this one, or blood pressure or sugar level is this much, and according to that we can provide even the emergency services there. This is not only having the video consultation or the medicine will be delivered after 3 to 4 days.
So you mean to say it's like a real-time thing that you are doing?
Yes, ma'am.
Okay.
In that case, what would be your cost of any medicament?
Ma'am,
Or what would be your cost? Let's say if you take a disease, I'm not talking about a major disease, of course, but if you, let's say, a person suffering from influenza, simple disease like influenza, or any simple infectious disease, what would be the cost to the patient?
like ma'am, first of all, we want to reduce the cost of the consultation. Even the doctor is providing through the video consultation. We will make a tie-up that in such a way they can reduce at least 100 or 200 in the
Can you
consultation.
tell me the rough cost that you estimate out of this service that you are giving?
Yes, ma'am. We have not concluded any survey or something, but whatever the price is going on a
But when
market
you are
basis.
proposing this business model, you must have worked on some unit cost, right? So what cost that you're proposing?
For cost, it is not like uh, we are charging something extra. Whatever this medicine is available in cities or town, we will provide on a same basis MRP, or giving by the 10% or 20% discount. We will provide on the same basis whatever the thing is happening in the urban cities. We
Get my question at all. Okay.
okay, ma'am.
Okay, thank you.
Thank you.
Thank you. IIC SRC 003. Are you present?
Yes, ma'am. We are present.
Yes, we'll begin with your video.
Raising the water quality standard for the rural India. Our abstract number is IIC SRC 003. The content includes introduction, rational, methodology, result, and discussion conclusion with references. So here we are going to challenge the problem statements where we already know that industrial wastewater contains heavy metal and harsh chemicals like acids, soaps, and solvents. These heavy metals are we are going to remove from those water bodies by using this plant, which is pistia statiotes and econic acids, which are well known to cause the eutrophication. So our approach is basically that this two plants, which is Pistia and Econia roots, basically the roots which can chelate the toxic heavy metal impurities as well as certainly new agricultural impurities like neonicotinoids. The phytoconstituent which we have extracted in our lab are known to be alkaloids, flavonoids, phenols, and tannins. These are used in the chelation process where they form an insoluble complex and then they get precipitates down and leave the supernatant water. So this process is known as a phytoremediation, where we use the green plants and microbes to concentrate or break down the pollutants. Our rationale includes to prove the phytoremediation abilities of both plants and quantify the rate of absorption as per the time in the given biomass. Here we extract, isolate, identify, and structurally elucidate by molecular docking, which are the phytoconstituent responsible to chelate to work out which phytoconstituents serve as the best candidate for this complexation of the arsenic and lead, to find the ways to encourage repurposing of use of pistia and ecornia, to prepare an industrial device that can take out lead and arsenic impurities from the industrial effluent and reach wastewater before it is released into the water bodies. So, how is the project relevant to the healthcare? As we all know that rural India is mostly having a lack of access of the medical benefits as well as the education. They are lacking behind. So we all know that the industrial wastewater is mostly drained into the river, and these industries are located mostly in the rural areas. So here the people face more problem related to the water which comes from the river, which can also affect their medical condition, where obviously the heavy metals which can cause these organs like skin, lungs, brain, kidney, various diseases are leading to these medical conditions. So our methodology is, this is the AR9 purifier, which is of chambers included. The AR9 stands for Aqua ISO, and 9 denotes for the total number of components required to make it. This was the experiment which we performed. This is the prototype we have made our lab, which is used to purify the drinking water in the central chamber. The central chamber, which is CIR, includes activated charcoal, ionized resin, and the root extract or the powder of those two plants which we have extracted. So the result which we got was that the water passing through the system and were found to be of the neutral pH. But the limitations which we came across was that the entire root powder was used, and the separation of elements that do not contribute to the heavy metal chelation were not separated properly. So the sediment filters get used up quickly, which can also lead to the clogging. Possibilities of the leakage across the compartment of the central chamber is also leading to the limitations of our AR9 filter. Then we have collected the plants in the tubs containing fixed amount of water. So these plants were collected in the water of 2.5 liter and 100 ml of the stock solution of already containing the lead were introduced in them. So we have performed the experiment on the lead, not on the arsenic. On the arsenic, we are still in the process to give the results. So basically, this experiment is performed on the lead. Every tub had two plants, and 10 ml of the water samples were withdrawn in the tub after time period of 2 hours, 4 hours, and 24 hours. They were analyzed in the UV visible spectroscopy, and the results which we got was progressive depletion of the heavy metal from the water was observed. These are the result for 2 hour, 4 hour, 24 hour and 24 hour at the 210 and nanometer. So basically the lead has the spectrum absorbance at the peak wavelength of 210. This is the note that as we got the same result at 210, so we switched it to 210.5 nanometer. Then the next step was we have collected the aerial part as well as the non-aerial part. The aerial part basically consists of the leaves chopped. They were chopped, triturated, and then put to the centrifugation. Same for the roots. And both showed the high concentration of the phytoconstituents as well as the chemicals. Upon increasing the dilution for the UV readings and computing with respect to the weight distribution and percent composition of the aerial tissue, and this is the result and discussion. This we performed at 4 hour, 8 hour, 24 hours for both statiotes and crassips. Coming to the conclusion part. So basically here we are going to make this water purifier, which is AR9. This purifier is supposed to collect all the heavy metals from the industrial water before it is drained out into the river, so that the river pollution can be minimized to some extent. And it is mostly focused into the rural areas where people can mostly afford it. So this phytoconstituent of both the hydrophytes were selected by us, which have not been fully characterized and structurally illustrated. We are still in the process, and also we are supposed to enhance the extracted chemical detection by AI detectors as well. We are still in the process to do it using the molecular docking to decode exactly which phytoconstituents serve as a chemoattractant to chelate out our target impurities, which is mostly arsenic and lead. We can come up with the sophisticated device to treat this industrial effluent. The phytoconstituent can be immobilized such that the water can enter and exit the pores of the mechanical entrapment media. This use of phytoconstituent can be regenerated by passing over a buffer from the top of the entrapment such that the heavy metals leaves the phytoconstituent and selectively bind to the ligand in the buffer. So this AR9 purifier is supposed to combat all these heavy metals so that the pollution can be minimized. These are the references. Thank you.
So I have a question. What is the quantity of the plant parts that you've taken? So you have said about the aerial plant parts that you have taken, right, for the experimentation.
Yes, ma'am. So here we have performed the analysis of this aerial, non-aerial, and as well as the whole plant. 1/1000 concentration we have used to do the UV absorbance. Like we have got the lead acetate extract as well through this.
No, what is the quantity I'm asking? What quantity of plant parts have you used?
1 leaf, ma'am, for aerial. Yes.
Just one sufficient.
Yes, ma'am. For the UV absorbance part, we have done for 1 leaf. Rest for the non-aerial, we have used the roots.
Okay. And why have you chosen this particular plants only? Why not? Because I believe there are so many plants which are used as bioabsorbent. They are used for cleaning water, for absorption of lead, arsenic. Why have you chosen this specific plant parts? Have you read some literature about it, or have you seen some paper publication about it? What is it?
Yes, ma'am. We have gone through the various other plants as well. But after going through the literature survey, we found out that Ecornia pistia and this other plant, which is the Pistia stratiotes, these both plants have the high absorbance quality of the heavy metals, mostly lead, arsenic, and other chemical which are available in the industrial wastewater, which is soap as well. So yes, we found out through the literature that this both plant has the high absorbance efficiency.
So, can I say that it was already known in the art, the absorption properties of lead and arsenic by this
Yes,
plant?
ma'am. Yes, ma'am. We have gone through the literature survey and we already know that these plants do absorb a lot of heavy metals. So we went through this AR9 purifier with these two plants.
So, what is the innovation here? I mean, what is the inventive step here?
Oh, ma'am, this is actually the green entrepreneurship we are going.
Also given to alkalizers and other which can be absorbed in them.
Okay, thank you. Thank you. That's it from my side.
Yeah, thanks.
Thank you, everyone. IIC SRC 004. Are you present? IIC SRC 004. Are you present? You are not audible.
Am I audible?
Yes, yes. Now you're audible.
Okay, thank you.
We are playing your video.
Greetings everyone. The topic of my research is nanocomposite of emtricitabine in hydrophobically modified pullulone, an attempt to reduce the associated hepatotoxicity. Coming for background and rationale, emtricitabine is a cytidine analog which belongs to BCS class 1, showing high solubility and high permeability, which will eventually lead to severe hepatotoxicity and lactic acidosis. The polymer I am using is pullulone, which is biodegradable in nature and which is used for the incorporation of the drug into nanocomposite. As we can see here, in the hepatocyte, conventional form of emtricitabine will eventually lead to micro and microvesicular steatosis. Our aim is to replace the conventional form of emtricitabine by nanocomposite of the drug for the reduction of the toxicity. Then aim and objective. The research aimed to attenuate the side effect of the emtricitabine by modifying its solubility in biocompatible nanocomposite using hydrophobically modified pullulone. Coming for the objective, initially chemical synthesis of biocompatible modified pullulone is done, followed by the entrapment of the drug into biocompatible platform to reduce its toxicity. And non-clinical safety assessment is done through in vivo biochemical analysis and histopathological study, followed by in vitro MTT assay on hepatic G2 cell lines. For the synthesis of pullulone, that is modified pullulone, cholesterol and succinate anhydride is used as a primary reagents. And at the end of the reaction, cholesterol succinate pullulone is done, where cholesterol succinate is incorporated into pullulone. For the characterization of the modified pullulone, FTIR and NMR spectrum is done, where pure pullulone and CHSP is compared. CN peak, C double bond O peak, COC peak were observed significantly in the CHSP group, where confirms the presence of the hydrophobic room in the modified pullulone. To ensure the results obtained from the FTIR, NMR spectroscopy was done, where pure pullulone showed 15 protons, as nature of all the protons were same. Singlet peak was observed. Coming for the CHSP, triplet peak was observed, as 180 protons present in the CHSP were not in the same nature. Triplet peak was observed, confirming the presence of the hydrophobic group of the CHSP. Preparation of the nanocomposite of emtricitabine. Step 1, solution method was employed for the formulation of the nanocomposite of the drug. Step 2, thin film hydration technique was employed for the incorporation of the polymeric nanocomposites of the drug into the liposomes here. This is the formulation table. As you can see here, 1:1, 1:2, and 1:3. Three ratios are taken of the drug and polymer, which was further evaluated for the particle size analysis and entrapment efficiency. All the formulation shows the particle size in the nanometer range, and PDI value was less than 1. And the entrapment efficiency, F2S and F3S, shows the better entrapment efficiency. For the surface morphology analysis, TEM study was done where particles of all the three formulation were in spherical in nature and freely scattered without any aggregates, confirming the stability of the formulation. And for the study of drug release pattern, in vitro drug diffusion study and ex vivo diffusion study was done. For in vitro diffusion study, dialysis membrane was employed, and for ex vivo radiodinum membrane was used. As you can see in the graph, F2S and F3S are showing like controlled drug release for 8 hours, and if after 5 hours in vitro there is a overlapping of the drug release pattern, which is also depicted in the ex vivo study where F2S and F3S are showing a overlapping drug release pattern. Release kinetic of F2S, here Higuchi and Cosmer-Pepper plot was plotted, where it clearly followed the Cosmer-Pepper release kinetics with the non-Fickian transport mechanism. For the toxicity analysis, albino blister rats, that is female, 200 to 250 gram weight was grouped for the study. Duration of the test was 28 days. Oral administration of the drug with nanocomposite loaded liposomes was done. After 28 days, animals were subjected for biochemical analysis and histopathology study, where blood samples were withdrawn using retroorbital route. These blood samples were subjected for the biochemical analysis using SGPT, SCOT, and ALP kits, and rats were sacrificed for collection of the liver sample, which was further subjected for the histopathology study. This is the table that represents the grouping of the animals into 5 groups. And coming for the biochemical analysis, as we can see here, all the 3 formulation falls in the normal range of the enzymes, but the pure drug clearly depicts the toxicity of all the 3 enzymes above the normal range, where F2S shows the better efficacy compared to F1S and F2S formulation. For histopathology study, saline treated group shows normal hepatocytes and normal central vein cells. For emtricitabine treated liver, lactic acidosis with inflammation, portal fibrosis, and steatosis with inflammation of severity 3+ was observed, whereas F1S treated, this was not expected, as fatty evacuation with a mild 2+ severity was observed, but F2S treated and F3S treated liver cells shows the near normal cells and normal cells of the portal vein and bile duct with no toxicity observed. Evaluating the biochemical histopathological study, F2S was found to be the promising formulation and was subjected to the further cytotoxicity study. This is the procedure that is employed for the MTT assay. Hepatic G2 cell lines were incubated for 28 hours, which was further treated with the pure drug, F2S, and doxorubicin. After the change of the culture media, MTT reagent, that is in the concentration of the 10 microliter, was added for the further incubation of 3 to 4 hours. And again, exchange of the media was done with addition of the DMSO of concentration 100 microliter, which was further incubated for the 10 to 15 minutes in room temperature, which was used for the reading the absorbance at spectrophotometry at 590 nanometer. This is the graph which shows the cell viability study depicting the percentage of cell viability of pure drug and F2S. As the concentration increases, we can clearly see the significant changes of the percentage of cell viability in the pure drug and F2S. As the concentration increases, the percentage cell viability of the pure drug decreases significantly compared to F2S. So to confirm the results from the graph, these pictures were taken in the concentration 40 microgram per liter to evaluate the results obtained from the graph. For coming for the summary, characterization of the polymer was done using ATR, FTIR method and NMR method, which confirms the presence of ester. For pre-formulation study, it confirms the drug and modified polymer were compatible and stable. Evaluation of the formulations, presence of surfactant improve the entrapment efficiency. High proportion of the modified polymer in the formulation could control the release of the drug, and in vitro ex vivo release confirm the sustenance of the drug release for 8 hours. Coming for the toxicity analysis, F2S found to be the best in the range of the SGOT, SGPT, and ALP enzymes. Compared to F1, F3S and F2S shows the normal hepatic cell lines with no toxicity. But to confirm the best formulation, F2S shows the better cell viability in hepatic G2 cells compared to the pure drug. So I can conclude that emtricitabine nanocomposites in modified pull-on could be the safest mode of delivery of the drug, overcoming the hepatotoxicity with an easy reproducible process. By this, the aim was achieved, that is a safe delivery of the emtricitabine by loading it into the biocompatible nanocomposite using hydrophobically modified pull-on. These are some of the references that I have referred for the project. Thank you.
Over to you, Pujant.
Yeah. So also I've heard about curcumin being used as a potent antioxidant, for similar thing, like it also reduces the hepatotoxicity. So can you have you done some studies on other drugs also? Because I have myself heard about how curcumin loaded pullulan acetate nanoparticles could solve the physicochemical defects of curcumin, like solubility, and it has been effective used for hepatotoxicity and for liver damage basically. So have you done some studies on other drugs also, or have you taken like this particular BCS class 1? And also my second question is, you mentioned somewhere about BCS class 1 drugs, right?
Yes, ma'am.
So why have you not taken class 2 or class 3 in this matter? Like, what is the advantage of taking BCS class 1 per se?
Yes, ma'am. For the first question, I am going to answer that, ma'am. I have not studied for the curcumin or for the turmeric, ma'am, because our main aim in the study is this drug is showing some toxicity, which cannot be prescribed for the liver disease patients. So we are trying to modify the release of the drug so that it will not cause the toxicity what it is causing in its conventional form. So we are modifying the drug, so there is no need of addition of any other component which make the formulation aspect a little complex, or it will need extra studies or other complexity for the addition of any other excipients. But what my aim is here, I am modifying the drug here so that it is capable of reducing the toxicity at my formulation what I have done. For coming for the second question, I have not taken BCS class 2 because this drug is having the potential to treat the HIV in a better way. It is a recently innovated drug which came in the market very recently, but because of its toxicity, it is not given individually, ma'am. It is given in the combination with tenofovir because of its main problem is it is having toxicity. Eva, that is because of it belongs to class 1, ma'am. It is having its high solubility, which will eventually leads to the toxicity. Here, my aim, I have achieved this aim by modifying or by controlling the solubility of the drug, so that it will not cause the toxicity or what toxic level it was causing in the liver when it was treated for the HIV patients.
Okay. Yeah, that's it for me.
Okay, you just mentioned that you have modified the solubility of your drug. So, can you present the solubility data?
Ma'am, I have not modified the solubility, ma'am. I have controlled the release of the drug by using the polymer. The polymer I am using is a pullulon polymer, which is a biodegradable polymer. I have modified my polymer into hydrophobic form, and for that hydrophobic polymer, I have incorporated my drug. So this hydrophobic nature of the polymer will reduce the release of the drug in the liver, which was the main reason for the causing for the toxicity which was causing for the in the liver. Because of achieving the modification in the polymer, I am controlling the release of the drug compared to the conventional drug.
Can you show the release pattern of your formulation?
Yes, ma'am.
The objective was to make a polymer which can control the hepatotoxicity, right?
Yes, ma'am. Yes, ma'am. Ma'am, my objective was like polymer I am using, even that is highly hydrophilic form. So I am modifying the polymer to the hydrophobic nature so that it will incorporate the hydrophilic drugs. When delivered to the liver, it will slowly release the drug in the pattern that I like. I control the release of the drug. Like what I have achieved is after reaching the liver, this is a pro drug, ma'am. It needs the conversion for the active drug for the activity, for the further activity for treatment of the HIV. Because of this nature,
have you made a pro drug, or you have made a polymer which is encapsulating the your drug? Because these are two different things, madam. Pro drug is different, and a polymer that you are converting. Can you just go a little for this to the previous slide?
ma'am, I have used the pro drug to incorporate it into a hydrophobic polymer. It is a pro drug. I am not doing a pro. Sorry, ma'am.
Which pro drug have you used?
Ma'am, entricitabine, ma'am.
So, entricitabine is not a pro drug, no? Help me understand.
It is a pro drug, ma'am. Emtricitabine, it is a pro drug. It needs an activation in the liver, which is a major cause for the like liver toxicity, that is lactic acidosis and hepatotoxicity, what it is causing. Because like so too much of the conventional, when we give the drug in the conventional form, like dose dumping happens or the accumulation of the drug happens in the liver because of this conversion, that is phase 1 transformation of the drug. Like there is a need of conversion of the drug to its active form. So to control the release of the drug in that phase, I have incorporated the drug into hydrophobic form of the polymer that I am using.
Got it. So, can I say that this can be as a technology, platform technology to evade hepatotoxicity of other drugs also?
Yes, ma'am, we can say like that. It depends upon the nature of the drug also, ma'am. Like here I am taking BCS class 1, and and main problem is this drug is having its main accumulation property in the liver. That is the factor that is promoting us for controlling its release in the liver.
And I also understand that you're converting it into a liposome. Can you go to the manufacturing process?
Yes, ma'am. Little backwards.
Yeah.
Previous slide, please.
What was the necessity of, so you had drug with polymer, you're agitating, what was the necessity of converting it into liposomes?
Ma'am, here I was ensuring that the drug, that nanocomposite what I have formulated, it is entrapped in a form like as we know, liposome is a hydrophobic form. So that I am taking it to another extent, so that I am not taking any chance here, so that nanocomposites can be broken down in the liver and cause toxicity. This is a step to make sure that even the protective layer, that is lipid layer in the liposome, is acting to control the release of the drug.
But don't you think your lipid layer in the liver would be acted upon by the enzymes?
Yes, ma'am, it will act upon enzymes. That's why only after like to release the drug, it has to cross two barriers, ma'am. Here, one is the lipid layer of the liposomes. Another one is the lipid layer of the polymer what we have incorporated into the polymer.
But first,
so both.
Yeah. Go ahead. Sorry. Go ahead.
Both the factors will it is an advantage for us for so that drug is not immediately released from the formulation that I have formulated, which is the main problem for the causage of the toxicity.
In that case, because we very well know that there are not much marketed products of liposomes, right?
Yes, ma'am. Yes, ma'am.
Apart from amplisome and doxorubicin, because per se, the manufacturing technology or the liposomes are very unstable.
Yes, ma'am. That is true, ma'am.
So then what made
Um,
you choose liposomes as a platform?
like, ma'am, next we can take the formulation to the next step, ma'am. There is no problem in that. After formulation into the liposomes, we can freeze dry it so that the product what we get from the freeze drying we can incorporate into any other dosage form like tablet or capsules in the conventional form.
Cost of the product. Because if you're adding
Sorry.
so many steps, what would be the cost of your product?
Yes, ma'am. Like I know it will be like a little cost, more cost, but this approach will give us by giving only the drug, we can reduce the toxicity that is associated with the drug.
Okay, thank you.
Yes, ma'am. Thank you, ma'am.
And what about the percentage efficacy of the drug?
Ma'am, that study we have not done yet, ma'am. My aim was of the study was to reduce the toxicity. For the further studies, we can take up to the treating of the HIV treated animals. And for the like to do that study, we need special approval from the different agencies to like to develop diseased animals in the lab. So we have not done.
Can you not do the same studies by simulation method instead of using animals?
Yes, ma'am. We can do that, ma'am. But because of the time limited factor, I have completed till this step.
Okay.
Secondly, one more suggestion. Whenever you are presenting animal data in any of the technical forums, your ethical approval is must. You need to mention the protocol number.
Okay, ma'am. Yes, ma'am. I'll consider that next time, ma'am. Sorry for the inconvenience.
Thank you, ISC SRC 004. Now we proceed with ISC SRC 005.
Hello everyone. I will be speaking about my research proposal, which is a novel drug dosage form, scopolamine gummies, under the category of Innovative Idea Challenge. These are the table of contents that I will be covering in this presentation. So, introduction about my research proposal. So, my drug, the API that I'll be using is scopolamine, which is a tropane alkaloid, also known as devil's drug, devil's breath, and hyoscine in the chemical name. It is one of the most prescribed drugs for motion sickness all over the world because of its anticholinergic activity. My formulation is a gummy, which would be chewed and it is very convenient, it is patient friendly, it is very easy to administer, it attracts the geriatric and the pediatric category of patients, and the route of administration would be via the buccal mucosa. As you masticate it, the drug will permeate through the the skin lining and would get into the blood stream and causes anti-emetic activity. Background of my drug. Scopolamine is used as all over the world. It is so 1/3 of the people are prone to motion sickness, and around 80% have medium to high motion sickness, especially even the pregnant women and the children. There is nausea and vomiting, and it's also used post-operatively. The current marketed formulations in the market are in the forms of tablets and transdermal patches, which have a comparatively a slower onset of action. There are various disadvantages of transdermal patches, such as skin irritation, hypersensitivity reaction, the pH compatibility issue, the skin diseases, and sometimes it could be inconvenient for the patient, or for the pediatric category, they would probably just scrap it off. And there are pharmacological effects such as probably blurred vision and a very slow onset of action. So gummies would be comparatively faster as through the buccal mucosal drug delivery system. The mechanism of the drug, as I said, that it has anticholinergic properties. It blocks the M1 receptor. It blocks the acetylcholine from sitting into the receptor, and it avoids the impulses to travel through the body, and it causes anti-emetic activity.
rationale that why we choose gummy as our formulation. Uh, it has a greater scope. It is cost effective. It is very readily available. It could be customized in various shapes, forms, colors. It would be very patient friendly. It would be naturally derived. I'll be covering my ingredients. It could be customized. It would improve one's lifestyle, and mainly, when it comes to oral drug delivery system, it would minimize the first pass metabolism. The key ingredient in our gummy is agar-agar over gelatin, because agar-agar is naturally derived. It has better consistency, and it is the perfect substitute. There are advantages such as it settles at a higher temperature compared to gelatin. It stays more stable. It is a vegetable origin, so it could be used for all vegans. It is clear, transparent. It has greater water binding capacity. It has a greater synergy with the sugar and the hydrocolloids. It's better tolerable. It has high water binding capacity, greater. The gel strength is better. There is fast. It completely solubilizes to the close to the boiling point, and the storage life is extended. This is the methodology where we took all the dry ingredients, we blended it with the water, dispersed it properly, we boiled it, and then we added the colorant, we added the sweetening agent, we even added beta-cyclodextrin as a complexation agent to mask the bitter taste. We added into our molds and we allowed it to cool down. These are the ingredients that we use. These are the batches, the picture on your left, on the right, are the batches that we performed. We started with gelatin, and then because of the test, the stability test, the appearance of agar was better and more chewy and better consistency. We used agar water, honey as a sweetening agent, scopolamine, the drug. We use beta-cyclodextrin for taste masking, and we use beetroot powder as a natural coloring agent, but various others can also be used. You could also add pectin and starch for better coagulation, and it would lower the settling time, and the gel should have the tendency to hold the drug. So you could play with various excipients as per need. The result of our gummy was it can cause faster onset of action, more convenient, reduce side effects, reduces the first pass metabolism, reduces the bitter taste, increase in the bioavailability, and greater absorption and mastication of the drug increases the absorption, and it causes faster onset of action. The data analysis, we have two graphs here. The first graph says the gummy is highly emerging into the market. By 2026 and 2024, there'll be a greater increase. And this is the map of scopolamine, how it's taking all over the world. It is greatly used in the Southeast countries. The organoleptic properties of the drug were performed. The Bloom test was performed, which is used to determine the strength of the gel, the ability to form a gel structure. So lesser the Bloom means it is softer, more elastic in texture, and the chewiness is desired. We got low to medium Bloom, which was decently okay, and the stability of the gummy, as you can see on the picture on the left, that was the gelatin one, which melted at the room temperature, and we used agar-agar, which lasted more than 6 months. We conducted the melting point, which was 9 degree, and the pKa was 7.5 to 7.8. Solubilization, it was very moderately soluble in water. We use solubilization techniques such as beta-cyclodextrin. The log P value is lower, which is in lipophilic in nature, easy to permeate through the buccal lining. It belongs to BCS class 1. Conclusion of a project that it is convenient to use the gummy around 30 minutes before a travel, considering the half-life. It is faster via the buccal route. Packaging, it could be airtight to avoid the moisture interruption. And agar is a preferred formulation for the gummy because of its stability. These are the references that we went through for our research proposal and the literature survey that we performed. So here I come to the end of my presentation. Thank you.
Yeah. So I have a question. You said that scopolamine is used for motion sickness, right?
Right.
But as you know, scopolamine also is used and abused for its hallucinogenic properties. I don't know if you must have done the literature for this or not, but scopolamine do have dangerous side effects as well, and it is abused a lot in some countries because it gives you hallucination effect. Also, the question is like you said about it reduces the side effects. So now the side effects can also have like increased heart rate, drowsiness, dry mouth, skin redness. So how have you tackled all this? And if you're providing in the form of gummies, don't you think it will lead to more abuse? Something which is like readily available, something which is even the kids might kind of tend to overeat or maybe not check the dose. How do you tackle that? Because of course the scopamine overdose can lead to respiratory paralysis, coma and death as well.
So the dose we've incorporated is 0.4 milligram, and our packaging will be very limited in amount. So there will be no drug abuse when it comes to the kids. And for the dose for hallucination is comparatively on the higher side. And to tackle the side effects, because this is a buccal delivery, the drug will directly permeate into the bloodstream, so it will be minimized compared to the other. The like exposure will be comparatively lesser.
But it is a buccal thing, so obviously there will be like the first barrier will be anyways crossed.
So because the formulation is a gummy, it is already high in moisture content because we'll be because the gummy tends to absorb moisture. So the dry mouth part will be tackled through this. Means comparatively, if it's sublingual, the gummy is more preferred because it's a matrix form. So it will be released very in a controlled manner.
Okay. So if I get some more questions in the meantime, I'll ask. Over to you, Sujata ma'am.
Okay, thank you, madam. Now tell me, clarify whether it is a buccal product or it is a sublingual product. Because what I understand,
Our product is a buccal.
So you mean to say that the gummy will adhere to the inner lining of the cheek?
So it will require a lot of mastication, a lot of chewing for the.
So then it is not buccal. What do you understand by buccal? Buccal means something which adheres to your inner buccal mucosa, your inner lining of the cheek. Here it is not adhering. You're chewing like a gum. I don't know whether help me understand. Your gummy product is something like what you get of Mepro. Mahabaleshwar, you know, na? Mahabaleshwar mein wo Mepro ka fruit. That is the gummy, right?
Yeah.
So then it is not buccal. You're simply chewing it and swallowing it.
But because of the mastication, we want the tongue will be adhered towards the cheek, so it will get
You can't control that, right? You can't control it. Can go sublingual also.
How will you control that it will adhere to the cheek? No. Just mention that it is gummy, which will be chewed by the kids and swallowed. Correct, na? Okay. That is what you want.
Correct.
Okay. Now you mentioned that your scopolamine, the drug is BCS class 1.
Right.
So then what is the necessity of complexing it with beta cyclodextrin when your drug has exceptionally high solubility?
Because scopolamine, the API is bitter in taste, so it requires taste masking.
So BCD is the only option to mask the taste?
It is preferred because it forms when the drug is there, the beta cyclodextrin forms a complex. It's like a carrier which will put the drug because it is hydrophilic on the outside and hydrophobic towards the inside. And the other property of cyclodextrin is also that it forms a film with the salivary mucus. So the contact with the taste receptors reduce.
And then one thing is there, your cyclodextrin is an oligosaccharide. Externally, it is hydrophilic in nature, and internal core of beta cyclodextrin is hydrophobic. And your drug itself is hydrophilic. How is it is going to take up your molecule?
As it forms the film also along with the buccal mucus, it reduces the contact with the taste receptors.
Have you done any stability study of your gummies?
No, it's a hypothesis. We've not conducted a lot of studies. We just made the base formulation.
And what is the novelty in your idea?
So scopolamine gummies do not exist as of now. There are transdermal patches, and there are disadvantages of transdermal patches. And to target the pediatric section, we chose this, and because it compared to the transdermal, this has a faster absorption, a faster onset of action. So this is our novelty.
Actually, I would just like to interrupt.
Yeah.
So regarding your claim about gummies, I think there might be some scopolamine gummies. I just saw it online. There is a website, Way of Leaf, in which these scopolamine gummies are being sold. So I think somewhere out in the market it is already there. Of course, it may not be there in India or maybe a particular region, but use of scopolamine is already known. The dosage form is already known. Only this is like a very less known dosage form that you're talking about. So I think even if there is a novelty to some extent, it may not be inventive. So what is your say on this? Like, what is your take on it?
So when we did our literature survey, we did not really come across this product. There were other API with gummies such as diphenhydramine and other antihistamines, but as per our literature survey, we did not really come across scopolamine. So we took up as our hypothesis.
Secondly, what Madam said is correct, that there is a possibility of abuse, right?
Right.
When the mother is not at home, a child might find the jar, child might find the product. Since you are making it as a gummy, hopefully you will make it as a palatable. So, how would you control the abuse or overuse of your gummy?
Correct. Because in a free time, generally we also tend to kind of like mentors, okay, like you have 1, 2, 3, whenever you're free, just you kind of pop it, and there is no control over it. Since it is easily available, freely available, there can be chances of abuse. And as I said, the hallucination effect that a lot of youngsters might be looking for, they might kind of abuse this product. So have you thought about this?
So our label of our product will contain that the gummy is it has to be taken in a limited number. It cannot be just taken any time. It has to have a duration. And secondly, the packet size, we will not have a whole package of gummies. It will be the sachet or the container will hardly have a 1 or 2 gummy.
So then how it will be cost effective? If you're just giving me 1 or 2 gummies, how it will be cost effective?
So, as per our tentative plan of cost, we plan to have 1 gummy around 50 rupees.
If it is 1 gummy around 50 rupees, why should I buy your product?
I rather buy Avomine for motion sickness,
Absolutely.
which is like a strip.
The transdermal patch is comparatively more expensive.
Okay, thank you. Well, any questions? Questions to you? Any questions from your side?
No.
Thank you.
Thank you.
Thank you, IIC SRC 005. Next, IIC SRC 006. Are you there?
Yes, ma'am.
Okay, switch on your video, please.
Am I visible, ma'am?
No.
Ma'am, I think due to some network issue, I'm unable to.
Okay, no issue. We'll
May I
start with your.
proceed? Yeah, okay.
Greetings of the day, sir and ma'am. First of all, I would like to thank you all for providing me this opportunity to present in front of all of you my innovative idea and also realizing its potential. Starting with the title of my presentation, it is the herbal mucoadhesive nanoparticles, a novel drug delivery system to deal with ototoxicity. HAPCON to beat ototoxicity. These are the contents of my presentation. Starting with the introduction, we will move towards about what is ototoxicity, then what are the current challenges dealing with the treatment of ototoxicity, then our innovative idea, and finally the conclusion. Starting with the introduction, even in this world of advanced and high technology, and also the various treatments available for cancer, there is the high rate of mortality of the cancer patients due to delayed diagnosis, high recurrence rate, and the various life-threatening side effects due to chemotherapy or other treatments. And one such major and life-threatening side effect is ototoxicity, which can be caused commonly due to the platinum-based drugs such as cisplatin or the radiation therapy and other supporting drugs like gentamicin and lutetiuretics. So ototoxicity is nothing but the poisoning of ears or the inner ear, which may lead to the permanent loss of hearing ability. And the most challenging part of treating this condition is its hard to access location and the tissue barriers present in the inner ear. So to provide an effective treatment and with minimum to no side effects, we came up with an innovative idea of introducing kiwi seed oil, which will be a major source of omega-3, that is our drug, using hyaluronic acid-based mucoadhesive nanoparticles as a drug delivery system. Now, what is ototoxicity? Ototoxicity is a very commonly reported adverse drug reaction, which is about 90 to 93% of patients are experiencing who are receiving these drugs as a part of their treatment, especially cisplatin, which is an anti-neoplastic drug and which is commonly used in the treatment of various types of tumors or cancers. And apart from this drug, there are over 200 medications which can be ototoxic or are poisonous to the ears, and yet there are no FDA-approved drugs available to treat this condition. How these drugs create ototoxicity? They may enter the scala media of the cochlea in the inner ear, and they may potentially cause permanent damage to the hair cells or the cochlear cells, and which may result in apoptosis and other conditions resulting in permanent loss of hearing and vestibular dysfunction. And this can profoundly impact the life of patients, and they may also be seen as a burden to the society. This is the mechanism of one of the ototoxic drugs, that is the cisplatin causing ototoxicity. The cisplatin may enter into the cochlear cells or the hair cells and may cause ototoxicity or the cell death in various ways, one of which is the mitochondrial damage, and this mitochondrial damage may lead to the calcium influx resulting in the ROS production, that is reactive oxygen species production, and also increasing the oxidative stress. The other ways are the activation of TRPV1 or the NOX3 enzymes, and this may also eventually result in the mitochondrial damage or in the increase of the reactive oxygen species. This reactive oxygen species may lead to lipid peroxidation, which may lead to inflammation and other conditions, and also in the increase of reactive nitrate species, leading to the increase of the oxidative stress, and this all ways will eventually lead to the cell death of the hair cells, which may be permanent. Now, what are the current challenges dealing in the treatment of ototoxicity? First of all, the anatomical location of the inner ear is very hard to access. As you can see from this figure, it is very deeply located. Then there are various tissue barriers such as blood labyrinth barrier present in the inner ear. And this barrier is not only physical but also a biochemical barrier which present where efflux pumps are present. Then due to the presence of this blood labyrinth barrier, there is a low blood flow rates or the less perfusion rate in the inner ear. The current strategies of drug delivery are highly invasive methods, and they may also necessitate the use of surgical procedures. And these methods are such as intratympanic, transtympanic, and intracochlear injections or other implants. And these highly invasive methods can cause permanent perforations to the tympanic membrane or the other sensitive parts of the inner ear, and may also drastically reduce the patient compliance. So why we choose mucoadhesive nanoparticles? First of all, mucoadhesion, as the name suggests, it is the attachment of a drug delivery system to a biological lining or a membrane. So as the round winter membrane is present on the cochlea of the inner ear, these mucoadhesive agents are meant to attach to this membrane, and then they can provide a path to the drug into the cochlea. Then nanotechnology, we know that from the past decade, it has been successfully applied in the various medical fields, foremost in the cancer therapy. So any nanoparticle is capable of diffusing this round window membrane, and it offers the possibility of introducing the drugs into the cochlea without the need of any surgical trauma or any surgical trauma to the sensitive ear. So complying with this concept, we idealize the use of hyaluronic acid as a polymer infused with the kiwi seed oil as a major herbal source of the omega-3 PUFA. This is the overall representation of our mucoadhesive nanoparticles. This is the outer layer of hyaluronic acid, and it will be filled with the kiwi seed oil or our drug. Now, why hyaluronic acid? Hyaluronic acid as a polymer has various advantages, such as it is biocompatible. Therefore, we don't need to worry about whether it will be irritating to the inner ear. Then second, it is a very tissue-specific targeting. It shows tissue-specific targeting so that it can target to only the drugs which we need to target, and it may not cause any harm to the other specific cells. The third advantage is that it has enhanced mucoadhesion properties than other polymers such as silica. And fourth is it is biodegradable. That means it is biofriendly. Now, omega-3 rich kiwi seed oil. Kiwi seed oil has a very interestingly nutritional fatty acid profile. It is a rich source of vitamin C, E, potassium, magnesium, but also a very remarkably stable source of omega-3 and omega-6. Omega-3, it contains up to 60 to 62% of omega-3 fatty acid. Now, omega-3 has a very interesting properties. It is antioxidant in nature. It is anti-inflammatory. It has been linked with the improvement of the blood flow to the cochlea, and this can eventually lead to the production of the cochlear hair cells. Now, what are the benefits to the society? We all know that, first of all, it is going to help the health sector of the society. It is going to definitely benefit the health sector of the society. Secondly, it will definitely benefit to the agricultural and the herbal industry, as it will increase in the production. It will increase the demand of the kiwi fruit and the kiwi seed oil, and this will also a benefit to provide a cheaper alternative to the society, as we all know that how expensive the therapies and treatments are for cancer and also for the treatments of ototoxicity. And this will overall lead to the economic increase of the society, or it will benefit the economic or national economy of the society. Coming to the conclusion of my presentation. So on the basis of this evidence, we came up with this innovative idea that it is possible to treat ototoxicity by introducing this hyaluronic acid-based mucoadhesive nanoparticles, that is a HAPCON, which is infused with the omega-3 rich in kiwi seed oil. And this can increase the capacity of these cells to resist the treatment-induced or oxidative insult. So we all know that the otological field, that is the new drug delivery system, has so far been demonstrated to improve the solubility and viability of the therapeutics in the cochlea. And these herbal nanomedical approaches will broaden the way for a more efficient and more specific treatments for hearing-related diseases and vestibular disorders. What are the future perspectives? It may need the formulation and designing. It may include the in vitro studies, including the cell line and bioassays, the in vivo studies, such as the animal models, and also various ex vivo studies. These are the references of my presentation. Thank you.
Yeah, so I have a question. So, what are the currently known herbal, therapies for ototoxicity, that you are aware of right now?
Ma'am, currently I have not found many of the herbal sources which are used in the treatment of ototoxicity. Only the steroids, various steroids have been used, and also there is a study based on the repurposing of various drugs. But I have not found any herbal source.
Have you done some patent or non-patent literature search for that?
No, ma'am.
See, I'm trying to say is if it is really breakthrough, which I find it very interesting, since it is herbal alternative used for ototoxicity, I think you should, if it is patentable, you should go ahead for patenting of the same.
Okay, ma'am. Okay.
Provided you do the animal studies or stage 1 clinical studies. So basically, if you show that there is some kind of data which is supporting your claims, then you can go ahead for patenting of the product or the formulation.
Yes, ma'am. Thank you.
Okay. Can you go? So what I understand that you'll be infusing the kiwi seed oil into hyaluronic acid, right?
Yes, ma'am.
Can you go to that particular slide number 8? Okay. So then how are you going to incorporate that oil in hyaluronic acid?
Ma'am, I'm planning to infuse the kiwi seed oil in the hyaluronic acid with the method called nano precipitation.
Because your oil is lipophilic. Hyaluronic acid is highly hydrophilic in nature.
Yes, ma'am, but hyaluronic acid it can be used with oils also. So we have came up with this concept to use kiwi seed oil. Ma'am, there are various techniques in the preparation of nanoparticles. So apart from nano precipitation, there are emulsion evaporation technique or solvent evaporation technique. So with the help of that also we can infuse kiwi seed oil in hyaluronic acid polymer using.
But evaporation, because ultimately your oil will be in the liquid form. So then
Yes,
how do you ensure the entrapment of oil into the HA? What is your polymer that is going to hold both hyaluronic acid as well as your kiwi seed oil?
Ma'am, I have to study this concept further, but this was just a base hypothesis which I presented.
And why kiwi seed oil?
Ma'am, kiwi seed oil because we are majorly focusing on omega-3 and we wanted to, omega-3 is generally we get in the form of, fish oil or any animal source, but we wanted to go with some herbal source of omega-3. And what we found from the literature survey that kiwi seed oil is the majorly it majorly contains omega-3 than other oils that we found. So that is up to
But
60 to
omega-3
63%.
omega-3 oils you can get from ample plant substitutes. Because kiwi is not a cheaper substitute, certainly not. So then, in that
Yes,
case,
ma'am.
how your product would be affordable by common people.
Ma'am, omega-3 can be available from other herbal sources, but it contains very less amount of omega-3. So ultimately, we will need more amount of oil to extract omega-3 from them. So that is why
How
we
much
chose
oil
kiwi.
would be incorporated? What will be your dose or what will be your label claim?
We have not evaluated that much, but the ratio of my nanoparticle can be around 1:2 ratio. One for
And
hyaluronic
what
acid
kind
and
of dosage form you would be giving to the patient?
can you please repeat, ma'am?
What kind of dosage form you would be giving to the patient?
We are planning to go with the gel formation of these nanoparticles.
Also, there is avocado oil that's been used commonly, in the art for autotoxicity. I think you can explore that as well. And because that also has shown significant anti-inflammatory activities and used for autotoxicity.
Okay, ma'am. I will go through it.
Thank you, ISSRC 006. Ma'am, just a minute. We're moving to ISSRC 007. Ma'am, is the video visible? Hello, ma'am. Is the video visible?
Yes.
Okay.
I don't think the audio is playing.
We can't hear.
Could we restart the video with audio? Now, within. Good afternoon, everyone present.
No, we can't hear anything. I insist you can just explain the PPTs orally.
And we shall be presenting our innovative idea. Our innovative idea is to take.
The volume is too low actually.
You're not audible.
Ma'am, just a minute. Ma'am, sorry for the delay. There is a technical issue.
Good afternoon, everyone present. We are team number 007 and we shall be presenting our innovative idea. Our innovative idea is to take an already common superfood and improve it, fortify it to target a term called hidden hunger. Now, what's hidden hunger? So, Indians are known to have carbohydrate and fat rich diets, and that in turn causes something known as protein energy malnutrition. So there's basically an imbalance between the amount of protein that an individual is consuming. Now, within this, there are also a lot of micronutrients that are missing within our diets. Micronutrients such as B12 are lacking in vegetarian diets, and insufficient consumption of fruits, vegetables, and dairy in general contribute to micronutrient deficiency such as calcium deficiency, magnesium deficiency, etc. Anemia, one of the most common deficiency, which is caused due to a deficiency of iron, is extremely widespread in India. Vitamin A deficiency is also something that is very prevalent in India, as well as vitamin D deficiency. Even though we think that India gets a lot of sunlight, it's the fact that people do not go out and they do not interact within the sun is that they do not get any vitamin D. Here we have a few statistics. So starting from the top left, it's the prevalence of vitamin A deficiency. So as you can see, India on average has about a 20 to 25% depending on the age group. Below that, we have vitamin B12 deficiency, which also is about an average of 20% among all the age groups. And if we look at the prevalence of anemia, which is the lack of iron, that varies between 60 and 30%, depending on the sex and the age group. And that information is according to the NFHS. So now that it's not as if there's no solutions to this problem, there are vitamin and mineral pills that can be taken, as well as powdered supplements that do contain the amount of the micronutrient required. However, there are a lot of limitations to this. Firstly, you could just be forgetful and not consume it. Secondly, is a term called pill burden, which is basically just keep taking pills and it gets sort of tiring. Third is a taste or digestive issue. People sometimes don't like consuming powdered supplements due to its chemical taste. There could be side effects. These things are usually expensive and they aren't available to a wide section of society due to their costs. Also, a lot of times pills and supplements cannot be provided to younger children and doctors don't really prescribe it. So let's move on with our innovative approach.
Okay, so this is our innovative approach. We have sort of a way, a more lifestyle compatible way that will be suitable for all ages. It will also be highly nutritious and most importantly, it will be tasty. And this is in the form of a yogurt. Plus, it will also have a taste of an indigenous fruit rather than the ones that are already available in the commercial market. Okay, so why did we think of yogurt? Firstly, it's a high acceptance as a food item. It's protein rich. It has probiotic ability that will help us regulate the digestive system. It is high in calcium and phosphorus. It also has smaller amounts of zinc, potassium, and magnesium that will aid in the development. And it also has good source of vitamin D, vitamin B12, and riboflavin. The reason for using jamun as a fruit to flavor the yogurt is firstly, jamun is a well-recognized fruit in India. It has a unique taste and also it's familiar. It's loaded with vitamins like A and C and also iron. It has a presence of anthocyanins that has anti-inflammatory properties. It's rich in phosphorus and potassium. Jamun is known for in the treatment of diabetes. It treats bladder issues. It treats anemia. There are a lot of advantages to using jamun, which is why we thought of using jamun. Along with jamun, there are other potential fruit candidates that can be used in this product. We think of jackfruit, which is rich in vitamin B6, vitamin C, potassium, magnesium, and antioxidants. Another fruit is custard apple, which is again rich in vitamin A, B6, vitamin C, and potassium. And another fruit that is not very well known, but it is still indigenous to India, which is mangosteen. It's rich in vitamin C, copper, magnesium, and other antioxidants. The future prospects for this particular product is it will promote the growth of indigenous fruits in India. We have already seen that people tend to consume more exotic fruits these days. The indigenous fruits, the local fruits that are in the market, there's not much light that's shed onto them. So this particular product creates awareness to promote the growth of indigenous fruits in India. And also, we have seen flavors of yogurt in the market. They're usually the common ones that we always see, like strawberry, blueberry, banana, etc. But having a unique element, which is indigenous fruits of India, like jamun, custard apple, jackfruit, this will help our products stand out. The other future prospect is economic support to Indian farmers. This is in alignment with the campaign called Vocal for Local, and it also creates a lot of export opportunities for this product.
So the idea of flavored yogurt is not something unique in thought. However, it has never been tried with indigenous fruits. There are multiple brands who do sell flavored yogurt, and that would sort of give you a thought that flavored yogurt has penetrated the Indian market. However, research suggests that this is actually still a very new section of the market. Flavored yogurt is not that common within Indian households. This product can be pushed as a preventative form of medication, having to never reach pills, supplements, or intravenous medication forms of medication. It can also be consumed by all age ranges since it is extremely healthy. It is extremely easy to consume. There's no need for any biting or swallowing. And also, yogurt in itself is extremely common in households. It is extremely healthy. And the fact that we are adding more benefits on top of it already makes it a very, very appealing product for households and hopefully even doctors to recommend. These are our references. Thank you.
Yeah, so I have a question for both of you. What is the innovation here? I would just like to understand that first.
Okay, so the innovation is not exactly flavored yogurt, as I've already mentioned in the video. It is an idea that already exists. The innovation is that, first of all, it's a product that not only can impact health, it can also impact social sectors and can also impact economic sectors, as we mentioned in our last slide. We do believe that this type of product, including indigenous fruits, can have a good impact on rural areas if we work closely with a lot of farmers. Secondly, the product on its own is an extremely strong base product. You can consume it obviously with a spoon, but then you can add it to shakes, you can add it to lassis, you can make froyos. Third, we hope that it will be a good contender, a strong contender to ensure that the younger generation develops better eating habits. Obviously, they're extremely picky, so providing them with good nutritious food while making it appealing and something that they want to consume is also something important. Third, indigenous fruits are comparable or lower in glycemic index when you compare to a lot of exotic fruits that these yogurt companies use, such as mango, raspberry, blueberry. It contains really high levels of vitamins, so we are not artificially fortifying any of this.
So why would I not consume the fruit directly, considering it's indigenous and more reaping benefits? Why can't I just have it raw fruit? I mean, why do I need to infuse in yogurt? And secondly, the part of infusing in yogurt, of course, you must have heard of this brand Epigamia. They are already using it in smoothies and flavored yogurts and sugar-free yogurts, and so many things are already there in the market. So how is your innovation, or how is your product
Okay,
different?
so with regards to Epigamia, oh sorry, somebody else speaking.
No.
Okay. With regards to Epigamia, yes, they are a big company. However, relatively speaking, the flavored yogurt market is extremely small. If you still look at it, there are a lot of players. However, they don't really have that big of a grasp on the market. Also, there are a lot of competitors that are interested in coming in. The South Korean brands are interested in coming into India. So, you said you would consume the fruit directly. So that's another thing. The fruits that we've mentioned here, such as jamun, mangosteen, nobody really consumes them anymore. They're sort of on the back burner. They're only consumed by people who really know of them, or like by people who have easy access to them, such as people in rural areas. So our idea is to take that and bring it towards an urban setting.
I think everybody uses jamun. I think even at my place there is jamun easily available, and my parents do get it. I mean, and if I tell my parents that okay, I am insisting of having a jamun yogurt, they'll say no, please have jamun. We'll get it directly for you. So, what is the driving force for me to indulge in a yogurt, then directly the fruit?
Okay, so in yogurt, you don't really have to go out of your way to consume it. You can add it to a meal. You can add it to your lunch, you can add it to your dinner, or you can, like, if you go to the gym, you can make
But
it
you
an
also know
ingredient.
that yogurts do have sugar, right?
Yeah, yogurt does have sugar.
So even the health part is being, not many health freaks will really have yogurt with its having sugar, although it has jamun.
No, actually, yogurt is a prevalent ingredient in the health community. They actually consume breakfast, like oats and all, they consume it with yogurt. So yogurt exists within the health community.
Okay, over to you, Sujata ma'am.
What would be the cost of your product?
Okay, so when we compare, firstly, we should look at the brands that have already cemented themselves. So brands such as Amul, Epigamia, stuff like that, who use exotic fruits. I've looked at a couple of papers, and apparently they can get their costs around 18 to 22 rupees per, I believe it's 80 ml. However, since we are using more indigenously available fruits, I am very confident we can get the price closer to 15 or 16 per 80 ml. Also, we can provide it in multiple serving packages. So you can obviously have the little carton, like how Epigamia gives, which is pretty standard within the yogurt community. However, we also believe that a package such as the one Paperboat provides, like a sippy bottle, is extremely helpful. It can be used by people who are elderly and by children.
Specifically, this yogurt or all these fancy things, you are looking in for people who are staying in urban community, or who are staying in metros, where they can afford to buy such fancy things. So now, if your objective is for getting rid of deficiency, it's not just the affluent class would want to use your product. So then how your product will be affordable to middle income or lower income people?
I'm glad you asked that question.
As well as availability of your product as well.
Regarding the last part, what do you mean by availability? Do you mean that with respect to the rural population?
Absolutely. And cost. Yeah.
Oh, and for cost. Okay. So we believe that an item such as yogurt, which is naturally fortified with really healthy fruits, which can provide the vitamins and minerals required, we believe it would be a really good addition towards stuff like midday meals. And if you produce it at that high of a quantity, it is very easy to get the cost down even lower than 15 or 16 rupees.
Just wait now.
So the rural community is not being shunned with this product, even though it sort of looks like we are taking the fruits from the rural community and then we are providing it to the urban community. That's not really the plan. We sort of believe that this product will work in the urban community. There are a lot of expensive yogurt places and expensive yogurt brands that do function within the urban environment. And we believe that we can take money from that side, that sector, and we can put it towards making the yogurt cheaper and include it in midday meals.
Okay. Any questions from your side, madam?
No.
Yeah. Thank you.
Thank you very much.
Thank you, ma'am. Thank you. Hello.
Yeah, we can hear you.
Yeah.
Esteemed judges, respected guests, participants, and all those assembled today. I stand before you with a deep sense of honor and privilege, representing the organizing team to express our sincere gratitude on the significant occasion of Student Research Congress 2023. First and foremost, we extend our heartfelt appreciation to our distinguished judges, Ms. Pooja Shah ma'am and Dr. Sujata Savarkar ma'am. Your presence not only elevates the prestige of this event, but also serves as a wellspring of inspiration for all of us. We genuinely appreciate the time, commitment, and the wealth of knowledge you have shared with us. Your insightful evaluations are sure to shape the trajectory of research endeavors presented here. To our participants, we extend our thanks for your unwavering enthusiasm and dedication to advancing research. Your contributions have added depth and diversity to this event, making it intellectually rewarding for all. Finally, a token of appreciation and gratitude is extended to our esteemed judges, Ms. Pooja Shah and Dr. Sujata Savak. This small gesture symbolizes our profound respect and gratitude for the invaluable role you played in the success of Student Research Congress 2023.
Thank you.
In conclusion, I express... Yes, ma'am.
So when should we submit our scorecards? Can we email it to you from where we got the link?
Yes, ma'am. You can email it.
Okay, fine.
In conclusion, I express my heartfelt thanks to each one of you present here today. Your presence has significantly enhanced the value of this gathering, and we are optimistic that the inspiration and knowledge gained will propel us into successful future endeavors. Once again, thank you, and we eagerly anticipate continued collaboration and success in the realm of research. I request the honorable judges to share a few words.
Yeah, so it was actually, great, participation by the students. There was something which even I got to learn. So really looking forward that, all of you go ahead and do some breakthrough innovations, do some research. Innovation doesn't really have to be something like very complex like robotics or something like that. No, not necessary. Even if you find some kind of a problem with the existing technology or existing product, you can just merely find a solution to that in your own laboratories, make a logbook, find the results, see some improvements if they are there. And if you find something breakthrough and novel, you can even patent that solution. It doesn't really matter if it's not the very big innovation, that's fine, but even a small solution can take you like everywhere. So basically, if you find some kind of toxicity, reduced efficacy, or to give you a simple pharma example, like for example, one tablet you're taking in like 200 mg thrice a day, And if you find something, innovate something which is just 100 mg probably, and you're taking once a day, so it is reducing the efforts of the patient to remember the dosage. It is basically patient compliance. So if you're finding something even as simple as that, it is innovative, it is worth patenting. So just open your mind, read lots of journals, read lots of free Google literature which is on Google patents. You can really read the patents and find the technology, the solution. Sometimes just by merely reading the patent, you realize that okay, we never thought of this. This could be done. And after that, you must notice that so many new ideas will come, and that is worth patenting. So I really wish you all the luck for patenting the solutions now and not just merely presenting it. All the best.
Second, the thoughts shared by Pooja Madam, and to take her thoughts ahead. See, novelty does not mean any time a fancy thing that you need to do. Anything which is addressing your unmet therapeutic need itself is also novel. And when you are thinking about novelty and patentability, also think of how it can be industrially feasible. Because any destination of any research idea should culminate into clinic. It should not be just for academic interest. It should culminate into your product reaching the consumer or the patient. So always think of industrial feasibility and scalability. Thank you.
Yeah, so just to put it in other words, commercialization is one thing that you should look for, not just merely having a piece of paper, which is a patent document. If you think that there is a potential for this particular technology, please find a solution for that, patent it, talk to the industry experts, talk to the patent attorneys. They'll help you commercialize it, and then, sky's the limit. Thank you.
Yes, ma'am. Thank you for your words of wisdom. The anticipation for the results is high, and we are excited to announce that participants will have the opportunity to discover their outcomes tomorrow during the valedictory function set to commence at 12 p.m. We understand the eagerness to learn about your achievements, and we invite you to join us in this celebratory event where we will recognize and applaud the remarkable efforts showcased throughout the Congress. For your convenience, the link to access the valedictory function has already been sent to your email. Please ensure you have details, and we look forward to virtually gathering with you tomorrow for the momentous announcement of results. Thank you once again for your active participation, and we can't wait to celebrate your accomplishments together tomorrow. Also, the feedback form for the event will be shared on email and WhatsApp group tomorrow. All the participants are requested to fill it to be eligible for the certificates. Thank you. Thank you, everyone.
Thank you. Bye bye. Have a nice day.
Yes, ma'am.